మేడం మీ పేర్లు నాకు టక్కగా చెప్పండి. చెప్పండి నాకు నేమ్స్ చెప్పండి. పెన్ కావాలి. అక్కడ ఏదైనా కవర్లో పెన్స్ ఉంటాయి చూడండి కనిపిస్తాయి.
మేడం స్టార్ట్ చేయాలి. అందరు గెస్ట్లు వచ్చారు.
స్టార్ట్ అయ్యే ముందు నాకు చెప్పాలండి. నేను లైవ్ చేసిన తర్వాతనే నేను స్టార్ట్ చేయాలి.
మరి ఎవరు ఏది ఎంసీజేరి ఇంకేం రాస్తున్నారు మేడం?
వాళ్ళందరి పేర్లు పిలవద్దా? ఐ రిక్వెస్ట్ ఆల్ ది
స్పీకర్స్ అని చెప్పండి మేడం.
స్పీకర్స్
రండి ఇక్కడ నిల్చోండి. మీరు అక్కడ నిల్చోపోతే స్టార్ట్ అవ్వదు.
लाइव स्ट्रीमिंग इन 5 मिनट्स
ये खराब है वो जो सही वाला है वो दे दिया
चलो स्टार्ट कर Hello. Hello. Hello.
यार उस पीपीटी में म्यूजिक ऐड नहीं किया है पर उसमें अभी मैं पीपीटी चला रहा हूँ ड्राइव से तो म्यूजिक नहीं चला एक बार डबल
No, see. Check, check. Once the music start, you start the PPT. All the best, all the best. Don't get confused. Okay? Don't get confused. Go with the flow. All the best to us. Team T-Series. Same T-Series. Oh, Tall Suresh. Tall Suresh. You are also here. Live start అవుతుందా projector?
laptop feed has gone upstairs. Hey, who is taking care of live? Bro, the upstairs na, that live feed is gone, which we are displaying on the screen na.
From there it will go. Not here, from here.
It was coming till then, but now it is gone. So
Now it is coming this picture.
अभी आ रहा है क्या?
ये आता.
अभी भी नहीं आ रहा है?
ये आता.
ओके, वो कह रहे हैं फ्रॉम पीसीआर टू हियर. अरे उनके लाइव में तो आ रहा है, उनके लाइव में आ रहा है. हेलो, look भैया, can I talk to him? The engineer here.
Hello? Yeah. Yes. Yes. Yeah, I did. They have removed laptop. From laptop. Yeah. I'm not getting laptop. I will show. Just ask him to connect the laptop. I will show you. From the super source. I will show you. Just there. I will show you. Already showed you. Not camera. Entire. Are given the any music? Hello? Are you given any music in PA system? ఆడిటర్లో ఏ ఆడియో వస్తుందో అదే లైన్లో వస్తుంది అది ఓపెన్ ఆడియో ట్రాన్స్ఫర్ చేయలేదు అక్కడ ఇచ్చేస్తాను స్ట్రీమ్ ఎండ్ అయ్యేది ఏం ఉండదు.
Okay. Check one. Okay.
ఆపాలంటే ఇక్కడ రెండే ఉంటాయి ఇక్కడ. కింద స్టాప్ స్టాప్ మొత్తం ఎండ్ స్ట్రీమ్ అవసరం లేదు. ఇంకా ఇక్కడికి వచ్చిన తర్వాత ఈ ఎండ్ స్ట్రీమ్ కానీ కొత్త ఎండ్ అంతే. ఆడియో ఉత్తరం అవసరం తీసుకో.
Check, hello. Check, check, hello. 1, 2. Check. Check.
అలాంటి కమిట్మెంట్ అవ్వదు. కూడా ఇవ్వలేదు. నేను మామూలు ఇది నొక్కితే ఇది వెళ్ళిపోతుంది. ల్యాప్టాప్ వస్తుంది, ఇక్కడ అలా వస్తుంది. కంటిన్యూస్ వెళ్తూనే ఉంటుంది. పర్ఫార్మెన్స్లు ఏమైనా స్పీచ్లు ఉన్నప్పుడు ఆపేసి కెమెరా ఓన్లీ ప్రజెంటేషన్ ఉన్నప్పుడే ఈ సోర్స్ సూపర్ సోర్స్. ఇంకా అసలు ఏం చేస్తే పని లైవ్ వెళ్తూనే ఉంటుంది. లైవ్ నైట్ డే అండ్ నైట్ ఉన్నప్పుడు లేదు లాగిన్ అవ్వదు. ఏదైనా ఇచ్చిరండి మరి నీకు డౌన్లోడ్ వస్తుంది. ఎక్కడ డౌన్లోడ్ వస్తుంది? ఇంటికి వెళ్ళి నువ్వు ఇక్కడ వైఫై కనెక్ట్ చేసుకోవాలి. నీ వైఫై కనెక్ట్ చేసుకోవాలి. వాట్సాప్ వాట్సాప్ డౌన్లోడ్ పెట్టినామనుకో వచ్చేస్తుంది. డౌన్లోడ్ వస్తుంది. ఇప్పుడు దీంట్లో వేసేసినాం అనుకో, దీంట్లో ఏం వేయకు. దీంట్లో వేస్తే ఇక్కడ వచ్చేస్తుంది. ఇప్పుడు ఎగ్జాంపుల్ ఇది ఉంది. ఇక్కడ వేసినాం
Good morning, ladies and gentlemen. I, Anantaparna Sumanjali, Faculty of Life Sciences from the Department of Microbiology, welcomes you all to BioMe'23, an international conference on advances in biology and medicine, organized by Telangana Social Welfare and Tribal Welfare Residential Educational Institutions Society, Government of Telangana, in association with Telangana Life Sciences, Federation of Asian Biotech Associations, Faba Academy, and Heartfulness Education Trust. Thank you for your presence today. The opening ceremony will begin in few minutes. I request everyone to be seated. I hope you have received a conference kit. The agenda contains the schedule for the day. Please go through the instruction sheet carefully for the important information needed during the event. We request you adhere to the timelines mentioned in the agenda so that we can organize the event smoothly. We have special guests who are about to enter the auditorium. We have volunteers spread on the floor wearing blue t-shirts. They will be happy to help you if you need assistance. And further, we request you to keep your mobile phones on silent mode. Use the side door marked as exit adjacent to the stage. The event is being recorded. For those who are not able, you can ask your friends to join us virtually on our YouTube live streaming named T-Sweries. And the audio file of the event, various presentations, and transcripts will be posted on the event website and our YouTube channel for future reference. Thank you all. For those who are not able, you can ask your friends to join us virtually on our YouTube live
An idea is not impactful until its practicality is demonstrated. Education is incomplete till its transformation facet is validated. Research is unacceptable till its prominence is substantiated. Good morning, ladies and gentlemen. On behalf of Telangana Social and Tribal Welfare Residential Educational Institution Society, we would like to extend a warm welcome to every one of you. We are delighted to receive all of you here in Telangana at BioMe'23, an international conference on advances in biology and medicine. We are aware that all of us are eagerly awaiting the commencement of the session. So, I, P. Sindura, and my colleague Aparna Sumanjali from the Department of Microbiology takes immense pleasure in initiating the proceedings for the day. This international conference is our attempt to delve into the deeper layers of life sciences, incorporating the various segments of pathology and unforeseen layers of life sciences. It will enrich us by providing many opportunities to discuss, critique, and share knowledge and ideas. This international summit attempts to connect the elite and the distinct features of biosciences to unveil the research work that revolutionizes the fundamentals. Without further ado, let's begin this collective journey at BioMe'23. I'm delighted that more than 800 participants from different corners of the world have joined us. On behalf of Telangana Social and Tribal Welfare Residential Educational Institution Societies, I wish you all tremendous learning, lifelong connections, and pleasant stay during the conference. On this wonderful morning, I take the honor of welcoming our eminent guests, distinguished speakers, delegates, researchers, academicians, and participants from across the globe who took their precious time for joining us today for this live event, BioMe, organized by Telangana Social and Tribal Welfare Residential Educational Institutions in association with Telangana Life Sciences, Federation of Asian Biotech Associations FABA Academy, and Heartfulness Educational Trust. We are immensely grateful to the Government of Telangana for its generous support. Federation of Asian Biotech Associations FABA Academy and Heartfulness Educational Trust. We are immensely grateful to the Government of Telangana.
Ladies and gentlemen, let us now take a look at the organizers of the entire three-day international conference. Let's take a look at a brief profile video of TS and TTWREIS. Let us give a big round of applause for the successful journey and more power to the organizers for enlightening the lives of the marginalized students. Thank you. Now, we are fortunate to have the presence of our esteemed guests with us, and I'm profusely elated to take this opportunity to invite our guest onto the dais. Please join me with a big round of applause in welcoming our guest onto the stage. And now, with great joy and immense exultation, I feel privileged to extend my warm welcome to our beloved, inspiring Secretary of Telangana Social and Tribal Welfare Residential Educational Institution Society and Secretary of Finance, Government of Telangana, who is also the Chief Patron of this conference. I welcome Shri Ronald Rose Garu, IAS, onto the stage. We welcome you, sir. I request our students to escort our guest onto the dais and present a welcome gift. Thank you, sir. I consider it a great honor to welcome Shri Panner Selvam Garu, CEO, Technogen, onto the stage. We welcome you, sir. Thank you, sir. I'm extremely honored to have this opportunity to welcome the Chief Advisor of this conference, Professor P. Reddanna Garu, Emeritus Professor at Department of Animal Sciences, School of Life Sciences, University of Hyderabad, founder member and executive president of FABA Academy, and also the founder and director, National Institute of Animal Biotechnology, Department of Biotechnology, Government of Telangana. We welcome you, sir. It's an immense pleasure to welcome Mr. Stan, who works for the French Embassy, Dhaka, Foreign Affairs Ministry of France, diplomat, and mindfulness and heartfulness certified trainer onto the stage. We welcome you, sir. Thank you, sir. It gives me immense pride in welcoming our guest of honor, Dr. Ram Kishan, Deputy Drugs Controller, CDS Co. India, who's a recipient of several prestigious awards, including Bharata Pratibha Ratna National Award, Dr. B.R. Ambedkar National Award, and many more. With over 25 years of regulatory service in Central Drug Standard Control India, we welcome you, sir. Thank you, sir. Now, I'm delighted to offer a hearty welcome to the Managing Director of Havel Life Sciences, founder of RAS Life Sciences, with over 20 years of experience in medical devices industry and expertise in molecular biology and building startups. I welcome Dr. Shashir Kumar Garu to grace the dais. Now, with immense pleasure, I welcome all the dignitaries and the guests, and especially the speakers of today's session. I invite Dr. Amit K. Tiwari, Dr. Scott Hall, Dr. Kamal Dua, Professor Sai Hanuman Sagar Boddu, Dr. Darshana Joshi, Professor Noruddin Khan onto the dais. Thank you, sir, and respected dignitaries for your gracious presence.
Thank you all the dignitaries for gracing the dais. Light guides us through the dark, clears great doubts with tiny sparks. To start on an auspicious note, may I now request the dignitaries on the dais to illuminate our path to glory by lighting the lamp and marking the ceremony open. May we all rise for a while. Thank you all. I would now invite the convener of BioMe'23, Dr. A.S. Pavani, OST Academics, Higher Education Wing, Telangana Social Welfare Residential Educational Institution Society, to elaborate on the objectives of the conference. We welcome you, ma'am.
Thank you, Sindhura. Good morning, everyone. On behalf of T-Sferries and T-Twerries, I welcome you all to BioMe'23, a 3-day international conference on advances in biology and medicine. It's a double delight for me today, actually, as a convener of BioMe'23 and also as an OSD working with the Higher Education Department of T-Sferries, to see our residential pharmacy college located in Mehboobabaad putting together a conference of this magnitude in just 2 years of inception. And did I imagine this while admitting our first batch of students? If you ask me, honestly, my answer would be no. But phenomena like these happen when you have a visionary leadership that fosters creativity and growth. And I would like to take this opportunity to thank our dynamic secretary, Ronald Rose sir, for creating that ecosystem at T-Sferries and T-Twerries. Thank you so much, sir, for always encouraging us and supporting us. I hope you're all enjoying the serene atmosphere of Kanha Shantivanam and appreciating our efforts to set up the ambience in this hall. But all of these are like the elements that add the extra oomph factor. You can say like the dance numbers in a Bollywood movie. But what draws the audience and stays with them is the essence of the film. Similarly, for any conference, it is the lectures, the networking, and the experiences made during the event that last for a lifetime, and often are stepping stones for a student's career laid at events like these. An internship with a connection made here sometimes might even translate into a tenure track position. The possibilities are limitless, but it all depends on how you utilize this opportunity of networking with the speaker delegation that is present at BioMe'23. As a convener, I'm very proud of the speakers we have here, with veteran scientists who have numerous international peer-reviewed publications to their credit, like Professor Scott Hall, Professor Frederick Williams, Professor Pia Vogel, Dr. Vinay Nandikuri, Director for Center for Cellular and Molecular Biology, Dr. Rakesh Mishra, Director, Tata Institute for Genetics and Society, and some renowned names in the pharma research like Professor Jayachandra, Dr. Kamal, Dr. Ritu, Professor Sai Hanuman Sagar, and emerging scientists like Dr. Gabriel. We have also taken care in balancing the scientific lectures with sessions on career sessions by Dr. Darshana Joshi, alumnus of Cambridge University, founder Vignan Shala, and entrepreneurship sessions by Bversity founders, the young entrepreneurs who are creating buzz in the life science ecosystem startup space of South India. But among all these wonderful speakers, we have one key delegate, who is also the keynote speaker, Professor K. Amit Tiwari, who deserves the credit for the speaker lineup I was taking credit for earlier. So thank you so much, Dr. Amit, for all the efforts and support to the T-Sferry and T-Twerry societies in helping us make BioMe'23 happen. Let's hear it for Dr. Amit, please. And to give you numbers of the conference, we received over 700 registrations with majority participants from the undergrads of pharmacy and life science disciplines. It's definitely a welcoming sign to see the young students showing interest in research and networking from such early stages of their career. I welcome you all. We also received 200 plus abstracts for poster and oral presentations, and the selected lot is displayed in the poster presentation area installed at N3 Satellite Center. BioMe'23 has the representation from over 50 colleges and universities from Telangana and a speaker delegation from 3 continents. Without taking much time, I now leave the dais to my colleagues and the speakers who have invested their valuable time in curating the lectures for the audience sitting in this hall today. I thank each and everyone who is involved in this event, and I wish and hope that this would turn out to be one of the best conference experiences for all the delegates. Thank you so much for this opportunity.
Thank you, ma'am. Seeking education is not a big issue for the privileged, but by being the pillar of support for the marginalized section, T-Sweries and TT-Rays has indeed set a benchmark by being one of the elite institutions to achieve the objective of shaping and sharpening young minds. I would now request the Honorable Secretary of T-Sweries and TT-Rays, Sri Ronald Rus Garu, IAS, to propose a warm welcome note.
Thank you. So very good morning to each and everyone who are present here. And a big welcome to all the dignitaries as well as the speakers who are on the dais also. So it's a very important milestone for the social welfare and tribal welfare societies, which are being in vogue for the past nearly 30 years, working for the downtrodden society. As you all know, most of our children are first generation learners, but the government is committed to provide world-class facilities to them, world-class education to them, and so born the residential institutions. These residential institutions are a bit different from the government-run colleges, degree colleges. The first one is residential. The second thing is end-to-end is taken care by the government. They are all granted aid institutions, run fully by grants from state government of Telangana. We could have also, like any other government institutions, could have stopped at giving the so-called textbook-based education, ensure that they clear their examinations, and then they can go back to the society. But in the society, all of them, it is not only the top management, including the faculty, including the non-teaching staff, my predecessors, everybody had taken it to heart that these children who have come to us and who is spending most of the time, that is nearly 90% of their time with us, rather than being in their home. And keeping in mind the trust which the parents had given to us, that is, they are willing to send their kids, trusting us to our institutions, hoping that the children will have a better future. So we have literally taken that to heart, and with active government support, we could achieve a lot of milestones in their undergrad and higher education. One of that fruit is what we are seeing it today. We could start the pharmacy college, and I am very proud to tell that this is the only residential, government-run, completely free pharmacy college in the country. There are many other pharmacy students who are here from different colleges, might be from the different states. I was told that more than 50 institutes or universities are represented here. Actually, the registrations were so fast that we had to stop registrations at one point of time and restricting the number of registrations from per institute. So we should not have more than some 20, 30. 30 is a number, I think, which they have restricted, saying that not more than 30 can register from one institute, so that we'll have a mix of students, and all other institutes also, universities also, can be represented in this conference. After closing also, we received a lot of requests that please allow some of us to come. So I was also very surprised about the response to this conference. So we also didn't expect that much of response, because we are doing it for the first time. But the caliber and the quality of the speakers, that pulled all the students and the other scholars who are present here. So I'm very much thankful to all the speakers and the scholars who are present here, especially Dr. Amit Tiwari, because he is the one who seeded this idea in our mind, and then he guided the whole society in how to go about it, including he has spoken to a lot of speakers and motivated them also to come to this space. I also thank Professor Scott, Professor Kamal Dua. They all flew from different parts of the continent, of the world, and it's a huge encouragement for us, and it's going to be highly beneficial for our children as well as the research scholars, because we are in the process of creating some sort of research mentality, and then some scientific temper we have to create in these children. Otherwise, these kids also, they complete their pharmacy and then they will start their pharmacy shops. They go and join some pharmacy chains. This is not we want for our children. And from our country and from our state and from our societies and all other colleges, we want to create high quality research, and we are willing to provide that platform. So we are in partnership with the Telangana Life Sciences. So now from FABA Academy, professor is also on board with us. We are in touch with the other big pharma giants also, how to have collaboration in research with the Dr. Reddy's Foundation. We had a meeting, how to give internship opportunities to our children. And we are also in touch with industries in understanding their requirements, what sort of talent pool they want, and we want to prepare our students accordingly. I also thank Professor Sai Hanuman Sahar who have come here, Darshana Joshi ji, who's going to talk about career opportunities in this particular field, and Dr. Stan, who's here, so who's going to combine interesting spirituality, is going to combine spirituality and science, and he has a lot of interesting theories. You will all hear that. And also, I also thank Mr. Paneer Selvam. He is the T-Hub ex-VP as well as now CEO of Technogen. And also special thanks to Dr. Ramkishan. He has come all the way based on our request. He is our Deputy Drug Controller of Government of India. So he's a very powerful man of the country, because he is the one who approves all the clinical trials which is happening. And also special thanks to Dr. Sishir Kumar, Director of Huvel Life Sciences, who had accepted our invitation in a very short notice, and he is here among us to share his experiences with all of you. So the whole objective of this conference, Dr. Pavani has already mentioned, but the major one is to create an academic ecosystem in the government education on par with premier institutions. So we wanted to dispel that, that sort of doubt or something which stigmatizes the government education. So we want to change that narrative in a way saying that these kids are no way less than those kids who study in so-called premier universities. And also we look for collaborative opportunities with similarly placed universities within the country and abroad, so that there is a lot of exchange, research ideas are being exchanged, student exchange programs happen, faculty exchange program happens, a lot of cross-learning happens among universities, so that this research is a combined thing. The COVID has actually taught us this particular thing. I was part of the COVID response team of the state. So the companies were willing to share their knowledge so that they can find a vaccine or a medicine to COVID pandemic. I also had bad examples where one company refused to share their formula, saying that you have to buy only from me, and they had a wait list for 6 months. I don't want to name that, but anyway, so that sort of parochial mentality should not be there. Cross-learning should be the way forward in this particular field, and I hope our speakers will sufficiently inspire all of you to take up research as one of your prime objective, and rather than limiting it to only your PhD thesis where you get one more added degree or a doctor name against you, I hope that you actually engage in proper research, and from the societies we can give you that platform wherein you can realize your dreams. And I also want to inform the speakers and audience there is that this conference has given us the confidence and also the courage to say that we wanted to make it as an annual event and ensure that this is going to be a premier event in the city of Hyderabad. So I wish you all the best, and I wish the conference a great success. And I also congratulate the organizers who have taken such pain to organize such an wonderful conference here. Special thanks to the kids who are behind the mics and the cameras, because they are all our degree college children of FAA, that is Fine Arts Academy, so who are going to be experts in that field. So I was just, yes, given a request about speaking in Telugu, so just 1 minute I'll speak in Telugu. Andarku namaskaram. So idi mana BioMe anadi mana Telangana Social Welfare and Tribal Welfare Residential Institutes Government of Telangana ద్వారా ఒక ఇంటర్నేషనల్ కాన్ఫరెన్స్ ఆన్ ఫార్మసీ అండ్ మెడికల్ సైన్సెస్ మీద ఏర్పాటు చేయడం జరిగింది. ఈ ఇంటర్నేషనల్ కాన్ఫరెన్స్లో చాలా ఇంటర్నేషనల్ స్పీకర్స్ వాళ్ళ రీసెర్చ్ గురించి మాట్లాడడం జరుగుతాయి, వాళ్ళ ఎక్స్పీరియన్స్ని పంచుకోవడం జరుగుతాయి. మరి మన పిల్లల దగ్గర నుంచి కూడా పోస్టర్ ప్రెజెంటేషన్ మరియు ఓరల్ ప్రెజెంటేషన్స్ కూడా జరుగుతాయి. దాదాపు 200కు పైగా మన పిల్లలకు సంబంధించినది, మన రీసెర్చ్ స్కాలర్స్ సంబంధించిన పేపర్స్ అండ్ పోస్టర్ ప్రెజెంటేషన్స్ ఏర్పాటు చేయడం జరిగింది. ఈ కాన్ఫరెన్సెస్లో మరి క్రాస్ లెర్నింగ్ అని మిగతా కాంటినెంట్లో, మిగతా రాష్ట్రంలో, మిగతా దేశంలో ఏం జరుగుతున్నాయి, ఈ అడ్వాన్స్మెంట్స్ మన దేశంలో ఉన్న పిల్లలు కూడా తెలుసుకోవాలి, వాళ్ళు కూడా ఒక రీసెర్చ్ మెంటాలిటీని డెవలప్ చేసుకోవాలనే ఉద్దేశంతో ఏర్పాటు చేయడం జరిగింది. 3 రోజుల పాటు ఈ కాన్ఫరెన్సు ఈ కళాశాంతివనంలో జరుగుతాయి. దీని ద్వారా మన ఫార్మసీ స్టూడెంట్స్ కానీ, లేకపోతే ఆ ఫీల్డ్లో ఉన్న రీసెర్చర్స్ కానీ, మరి దీంట్లో ఉన్న కెరియర్స్, ఫార్మసీ చదవడం వల్ల ఎటువంటి కెరియర్స్ పొందవచ్చు అని తెలుసుకోవడం కానీ, ఆంటర్‌ప్రెన్యూర్ స్కిల్స్ డెవలప్ చేసుకోవడం కానీ జరుగుతాయి. సో ఐ విష్ దిస్ కాన్ఫరెన్స్ ఎ గ్రేట్ సక్సెస్. I also want to thank the Heartfulness Education Trust, who are our partners in organizing this conference and to giving us such a lovely venue and such a lovely staying place and a wonderful hospitality also. So I wish you all the best, and I wish the conference a great success. Thank you very much.
Thank you, sir, for the warm embrace. Indeed, our students are blessed to have you as a dedicated and continuous support system for their overall empowerment. Thank you, sir. I would now request Sri Panneer Selvam Garu, ex-Vice President and Chief Innovative Officer T-Hub and CEO Technogen, to address the gathering. We welcome you, sir.
A very good morning to all of you. How are you all feeling? Hello? Good? Good. So first of all, I think it's an amazing conference. I think many congratulations to the organizing committee and the wonderful students who are present here, and my revered fellow speakers and dignitaries on the dais. A very good morning to all of you, and it's a pleasure to be with you all. And truth be told, this is the first biology and medicine conference that I'm speaking in. But the truth is, in any conference today, there's no conference is complete without things that we always speak about: innovation, entrepreneurship, and startups, right? That's the primary role that I'm going to talk, topic that I'm going to talk about, share my thoughts around it, and see how many of you want to be an entrepreneur or start something new when you pass out of your colleges. How many of you want to start a new business in your field? Show of hands. Okay, good. That's a good number. So in India today, we have an amazing opportunity to be an entrepreneur. And as the numbers speak today, we are at about 75,000 plus startups in the country. In Telangana alone, we have about 8,000 plus startups. Of the 77,000 startups, 7,500 startups are in the medtech space. So today medical technology is emerging as one of the most what you call revered technology in the startup space, and of course it is kind of creating an impact that no other technology sector is creating, because we have at least 7 billion people to take care in the world, and in India alone we have about 1.5 billion people to take care in terms of the healthcare and the pharmaceutical spaces. It's no wonder Hyderabad has many firsts to its credit, and I'm sure BioMe'23 will also be one of its first, and many more years to come. It will be one of the flagship conferences of biomedicine and biology. But in the most important part is it's about the students that you are here, who are here to kind of take this conversation forward. There are three things that I would like to share with you. One is it's all about today the emergence of technology has helped every last person is actually capable of doing something different if they really try out, right? So every one of you have an equal opportunity to kind of build your own dream, build your own company, and build and enable amazing startup ecosystem in the state. Telangana is the most youngest state in the country, but it has the most matured innovation ecosystem in the country. No other state in the country has the amazing innovation network that the Telangana has today. And in fact, it is the most, it has about 35, I was just talking over, it is 35 institutes of national prominence which can be leveraged by all of you, each one of you can have access to this innovation ecosystem. So my first thing is be bold. As young students today, you have every single opportunity and access to technology that you have. Make the best use of it. You can get nurtured by incubators like T-Hub. There are dedicated medical medtech incubators in the state. And there are amazing amount of talent and capability that is there in the state and city of Hyderabad. So the first opportunity is to think bigger and bolder so that you can build your dream in the medtech space. The second part, it's also about the whole the amount of research that happens in Hyderabad. How many of you know Hyderabad is the pharma capital of India? Right, it is the pharma capital of India. Almost, the Genome Valley that you might have heard about is filled with world-class pharmaceutical companies in the country, and it has amazing amount of research and development capabilities that is being run right here in Hyderabad. And you all are, welcome to access that. And if we are here to facilitate that access for each one of you, should there be a need that you want to build something bigger and better. So it is the third thing, and the last thing that I would like to share is all about entrepreneurship, because anything that that you need to do, you need to have a mindset of creating a job rather than asking for a job. Right, the whole idea as an entrepreneur today, Ronald was talking about pharmacy. Today, there are million-dollar pharmacy companies who are doing online businesses in the pharma space. The whole pharmaceutical and the pharma direct-to-consumer space has evolved so much that it has disrupted, the pharmaceutical trade and the pharmaceutical business. So there is immense amount of potential. There is immense amount of opportunity, and most importantly, there is immense amount of talent which I see in front of me, which is what we are here to kind of witness in the next 3 days, and see each one of you get inspired by this conference and to take home, that inspiration to build your business, build and do something innovative, and build an organization that will create jobs That's pretty much, if you keep the focus around those three areas of innovation, entrepreneurship, and starting up something new and solving a problem, pretty much there is no looking back for each one of you who are present here. So on that note, I would like to thank you and thank the organizers and wish you all the very best for the next 3 days of learning and immersive engagement in the BioMe'23. Thank you.
Thank you, sir, for your valuable information. I would now request Professor Reddanna Pallugaru, Emeritus Professor, Department of Animal Sciences, University of Hyderabad, and Executive President of FABA Academy to deliver his valuable message. We welcome you, sir.
Good morning and welcome you all to this BioMe'23. Let me first thank the organizers for including the Federation of Asian Biotech Association to be associated with BioMe'23. We are really delighted to participate and then also help in the organization of the event. The Federation of Asian Biotech Association is a non-profit organization which we started in 2005, mainly to promote academy and industry interactions, not only in India, but also across various Asian countries. In fact, we have now expanded to the African countries also. So as part of this activity of promoting academy and industry interactions, the main event that is being organized is BioAsia, which is starting in the next couple of days. The 20th edition of the BioAsia is happening in Hyderabad, in HITEX, on 24th, 25th, and 26th. So during our journey of the Federation of Asian Biotech Associations, promoting academy and industry interactions, we found that there is a big gap between the academy and industry in terms of human resource development. The kind of skills that are being imparted in the colleges are not enough to be absorbed in the industry. And FABA has initiated FABA Academy in 2020. And as part of this FABA Academy, we have been organizing seminars, webinars, workshops, conferences, mainly to promote the career guidance and skill development activities. A number of webinars, starting from how to write your CV, what are the career opportunities for higher studies in India or abroad, and what kind of opportunities that are there in the industry in different disciplines, traditional, conventional, and then other opportunities also. And it is not only just promoting this academy activities, but we also felt that a number of startups have come up in the country. As just now Mr. Paneer was mentioning that there are 75,000 startups that have come up with the support from the state as well as the central governments. But for them to graduate to the next level is the major lack. And that is where FABA has started FABA Entrepreneurship Division, mainly to promote the startup ecosystem. And in that, where we have brought the venture capitalists not only from India, but around the world, and the promising startups and the venture capitalists meetings were conducted, which was one of that flagship event was the whale tank event, which we conducted last year. And this is mainly to promote our startup companies to graduate to the next level and then flourish. I am really delighted to see many of you are interested in starting your innovation activities and entrepreneurship activities, the FABA Academy, FABA Entrepreneurship Division is ready to guide you in that direction. At University of Hyderabad, we have 3 incubation centers, and in this, around 65 companies are located right in the University of Hyderabad campus. And we have been promoting these activities. Not only that, as part of this academy industry interactions, in fact, on 23rd, we are organizing Hyderabad Innovation, Life Sciences Innovation Cluster Meeting, where we have invited all the academic institution heads like IIT Hyderabad, University of Hyderabad, CCMB, CDFD, ISCT, NIN, all these academic institutional heads are brought to the Genome Valley where a number of pharma and biotech companies are located. And in fact, this is the first of its kind that we are organizing in Genome Valley, a cluster meeting of academy and industry so that they will be able to come together, interact, and come up with innovative product development. As the previous speaker was mentioning, Hyderabad is the pharma capital of the world, vaccine capital of the world. But we are very good in generics and biosimilars. But not innovative products are coming up. Innovations will emerge when there is a interaction that is happening between the academy and industry. In order to promote that, we have taken that initiative to bring them together with the hope that there will be more fruitful interactions and more innovative products also will come from our pharma and biotech industry. So these are various activities, and for the children, there is a greater opportunity for you to guide, to take advantage of various programs that we are organizing. In fact, all the previous events are there in our YouTube channel, and you could go through, and we are in fact planning to interact with your educational society, mainly to go visit the colleges and then guide them in career guidance and skill development activities. So these are various programs that you could avail. Let me once again thank the organizers for a wonderful event and inviting us to be associated with this conference. I would like to congratulate Mr. Ronald Ross and his entire team, and specifically Dr. Pavani, who is our one of our proud alumni from University of Hyderabad, who has put together this wonderful meet. I once again welcome you all to this BioMe'23, and I urge every one of the students here to interact with the experts who have come from different parts of them. This is the great opportunity to build collaborations or interactions with the leading scientists around the world. Thank you once again.
Thank you, sir. We thank you for extending your support in all the ways possible for organizing BioMe'23. It gives me immense pride in welcoming Dr. A. Ramkishan, Deputy Drugs Controller, Ministry of Health and Family Welfare, DGHS, Central Drug Standard Control Organization, India, to address the gathering.
Respected Sri Ronald Rajji, very young and dynamic bureaucrat. Just 4 days interaction with him. Please give a big clapping to him. The way he has taken lead with respect to this important BioMe 2023 conference in this important ashram, that is Shantivanam, Professor Redanaji and Professor Tiwari ji and Darshana Madam, Shishirji, the young entrepreneur for the in vitro diagnostic kits he is developing during the COVID time. He developed a lot of RT-PCR test kits on molecular basis with respect to important diagnoses for important SARS virus, etc. And other dignitaries invited on the dais, and Dr. Tiwari ji also, and Dr. Kamal Duha, and because I could not remember, because I am not having agenda, so at least I can remember through the various speakers, and all important invited speakers invited across the globe, and special invitees, and all important organizing committee members involved for BioMe 2023, special invitees, ladies and gentlemen, all invited personnel from the electronic and print media, and all 800 budding pharmacists, life sciences sector students presenting in this important hall. So good morning to one and all. On behalf of Health Ministry, it gives me immense pleasure to share some of my good thoughts for the betterment of vulnerable population. You are all blessed. BioMe, B-I-O-M-E. B means bright, I means innovation, O means opportunities, M means you are all working for the men, means females and gentlemen, and in an excellent way. That is the terminology I got today, and you are all blessed with respect to the Shanti Vanam. Shanti Vanam is headed by our Daji. Daji is nothing but Kamlesh D. Patel. He is our proud pharmacist, did his pharmacy in the year 1961 from Lallubai Motilal College of Pharmacy. I am also fortunate to do my research, that is PhD program on type 1 diabetes for development of novel drug formulation, clubbing with some trace elements with recombinant derived insulin molecule like Mixtard we have developed. I got patent for that. I am proud to be there in Gujarat Technological University, and I am from regulatory side. The main gap is, friends, here the academia and industry people always discuss various important initiatives, maybe science and technology, but regulators also very, very important. If you connect regulators, academicians like top Nobel laureates like Amit ji and Kamal Duha ji and our Panir Selvan Saab taking care of all the startup, more than 8,000 startup companies in government of Telangana, but we need to have good harmony. That's how our Shishir Saab, when he came to our office, we have given same day 10 approvals for the IVD kits. So very, very important. I'm sorry, friends, you are having bright future. India is going in different direction. Honorable Prime Minister of India taking lead with respect to identifying all important key sectors. He has identified 25 key sectors across the country. Out of that, pharmaceutical sector is one of the key sectors where innovation, drug development, novel formulations are taking place to serve humankind with respect to the important diseases or disorders in human hospital or clinical community setting. So therefore, friends, life sciences students sitting all here, and first Gurukulam started in Mehboobaad in our country, means tribal residential pharmaceutical college situated in that Mehboobaad. I am also proud. I belong to Mehboobaad region, and my father was teacher, and today I am blessed through our Ronald ji. Friends, आप लोगों को पूरा उज्जवल भविष्य है. Either here, Telangana Social Welfare Residential Educational Institute, Telangana Tribal Residential Educational Institute. इसमें देखो TS, T means talent, S means skill, T means tribal, S means social welfare. So क्या कम है? You are having bright future. Honorable our Chief Minister of Telangana is taking lead with respect to new important curriculum activities in order to impart quality education through all invited speakers like this. Please give a big hand to all the speakers. And they are taking lead. And we are all here to support as a regulator, friends. Dr. Saab, Ronald Saab, we are all here. I worked in Mumbai, Gujarat, Delhi, and Northeast Calcutta. So I know the difficulties. We have supported more than 300 people, 17 students. I have encouraged to carry out cancer research in different countries across the globe. So we are all having close coordination. This is the gap. If we can identify, we will support you. We will facilitate you. Telangana is going to take lead in near future with respect to innovation, or drug development, maybe biotechnology products, or biosimilar products, or cell therapy, or gene therapy, or xenografts, or stem cell product in international commerce. So huge potential. If you see the globe, the pharmaceutical sector, across the globe, we are having $1,200 billion market. Out of that, India is contributing $49.55 billion market. So out of that, $25 billion we are doing for export business, covering more than 205 countries, 205 countries, and domestic $24 billion market. So huge potential, and medical devices, cosmetic products, traditional products, routine allopathic products are going to take lead. Now we are all concentrating on chemistry. If you see the percentage, 70% is involved for the chemistry and 30% is biology. By 2030, by 2040, when Ronald Saab is going to retire as the chief secretary, his superannuation is the 2040, then biology is going to take lead for 70%, chemistry is going to remain with 30%. So huge potential and lot of bright chances for young mind people sitting off the dais, pharmacists, microbiology, biochemistry, biotechnology, and other important areas where you are having. So I want to congratulate you all, and your parents are really very, very fortunate to encourage you for choosing the life sciences graduation in different discipline. You are very happy, and also please try to concentrate on important activities. Don't keep your mobile round the clock up to night 2 o'clock, 3 o'clock. Aane wala samay mein you are going to have the marriage and this thing. Please concentrate on important textbooks. Gurus, because of gurus you are all here. Because of my father, because of my gurus, I am taking lead for the all important innovation and regulatory evaluation with respect to different approvals across the country. So we are also having limitation, but friends, I am also from a small remote village. Chota Tanda se aaya hum, but we never faced any difficulties. Only hard work. Hard work remains, because people have different community politics, but no. Every human being, every student is having their own important brains and important thoughts. Left brain, right brain. Left brain takes care of the innovation, right brain takes care of the creativity. So you're all blessed in this important Shanti Vanam, our Kana Ashram. So you can take lead, and with this important forum, please try to absorb all important scientific inputs and take lead. And Gurukulam people, Gurukulam students, globally competent, they can do wonders. And when we are having more than 163 colleges across the state, Telangana state, more than 263 schools, where we are missing excellent teaching staff, excellent faculty, excellent government, excellent bureaucrats like Ronald, where we are missing. Means missing is we have to put our constant hard work with respect to your innovation or your career opportunities in order to settle in your career for sustainability and stability, and also to satisfy your parents. They are having lot of dreams. Mera beta America jayega, mera beta Germany jayega, mera beti Korea jayega. वो ड्रीम को आप कभी भी धोखा नहीं देने का. So this is my appeal. I am also from a Telugu this thing, but you have to work hard. This is my appeal. And pharmacists are having excellent culture. CULTUR. C means communication, U means urge, L means leadership. Again, T means teamwork, and U means understanding, and E means we are all working for the efficacy, readiness of all global community people, bureaucrats, all human beings, scientists across the country, putting for the safety, purity, efficacious, important medical products in order to keep desired pharmacological effect in clinical pharmacy is the main objective. So therefore, you have to work hard, bright future with this forum. From every year, sir, irrespective of my position, I'll come and dedicate to your student, and they should get maximum opportunities for their internship. Maybe pharmacology, biochemistry, or clinical pharmacology, toxicology, or laboratory, any service is required, any support is required. We are all here to facilitate, not to harass people. So regulators' mindset will be changing. Next week, we are going to have Chintan Shivir on drugs, quality regulations and enforcement 2023 in this hall. All commissioners, all principal secretaries, Honorable Minister for Health and Family Welfare, they are all visiting. I will be the nodal officer. We are going to have the complete new regulation for the new new drugs, medical devices, cosmetics 2022 bill is in the draft stage. We are also going to work in similar line of ICH countries, European Union, Canada, Australia, USA, United States. So India is going in different direction. So India is going to be the good destination for all novel research and scientific research to serve vulnerable population across the globe. Thank you very much for giving me the opportunity to share my thoughts. We are all here. Darne kane Telugu medium nachina mak English raadu anukodu. Telugu medium people, rural students can do wonderful things. I wish at least out of 800 students, I want to see at least 6-7 Nobel laureates from this hall to the global level in near future, and we are all here. That is my appeal, and I am always with you. Whatever support, whatever samples required with respect to your research project, I am there to support. With respect to imports, exports, we are monitoring in Shamshabad Airport, Krishnapatnam Port, and Visakhapatnam Port. So I got the opportunity to facilitate students, not as a regulator, but as a friendly way. I will support you, irrespective of your problem. Thank you very much. But isme 1 minute my time leta sir, because already 11 minutes I took. మనం చనిపోతే మనకి ఏం రాదు, ఒక చిన్న పాట వినిపిస్తా. Thank you very much. Value as a value, only if its value is valued. So my special thanks to our young IAS officer. मैं 2 दिन में मैं पूरा पता कर लिया what is Ronald ji, a very simple man and scientific personality, utilize his services for the betterment of Telangana and betterment of country. Thank you very much.
Thank you, Dr. Ramkishan Garu, for your informative talk and valuable suggestions to our young participants and pointing us on the importance of hard work. Thank you, sir. Now, we are delighted to call upon Mr. Stanislas Lajouzi, Mindful and Heartfulness Certified Trainer, Diplomat and French Embassy, Dhaka, Foreign Affairs Ministry of France, to address the gathering. We welcome you, sir.
Good morning, everybody, and good morning, dignitaries. I'm very happy to be here, and I will share a few thoughts which are dear to me, especially when I see students. To start with, I would like to quote the writer of science fiction, Isaac Asimov. He says that the saddest aspect of modern society is that science gathers more knowledge than wisdom. So what I would like to think over is then how to create wisdom and how to live with the life of wisdom. I'm sure we can get a lot of knowledge in schools, universities, and so on, but how do we use it? And we will see how it can be helpful for the students to cultivate such wisdom, but also for the beneficiaries. And I would like to warn the students, wisdom cannot be learned just by reading books. I may know information, I may get access to a lot of information. I may know knowledge how to make medicines, but how to use it, adapt it to the people for their greater welfare. That is not given by books only. There is no ready-made recipe. And for me, that wisdom can only or mostly cultivated if we learn to tune the mind and the heart. And one of the most efficient means for that is meditation. And I would like to come down to practical things and see then what the heck was this? How are we going to do this? How is it going to help in our student lives? But let's speak about decision making. We make decisions every day, hundreds of them maybe. And later on, we will be also paid to make good decisions, wise decisions, useful decisions. But how many of us know what is the biology of decision making? Do we know what is the process of decision making? Very strange enough that we have never thought about that and how to improve our capacity to make decisions. Have you heard about this movie, Forrest Gump? Maybe it's not popular for you at your age or in your culture. Forrest Gump was supposed to be not a very intelligent person. He just liked running and chocolate box. And he was so simple that everybody was making fun of him. He was not considered as an intelligent guy. But if we look at his whole life, he only took good decisions in his life. He followed his heart. And we cannot say that someone who only took good decisions in his life is a stupid person. But daily, every single moment, we see very intelligent people taking very bad decisions. The moral of the story is that decision-making is not only about thinking, but it's about feeling. And it's not just a movie, it's science. There are 4 steps in decision-making. First one is security check. Am I going to be eaten or am I going to eat? If I am going to be eaten, fight or flight, no thinking, I just have to run for life. I am stressed, I behave the way I can. If I am going to have an ice cream, then I move to the second step, using the prefrontal cortex. It's about thinking. This is where I can show all my intelligence, analyze, and evaluate all the pros and cons. But just be aware, this process is very slow and energy-taking, energy-consuming. And interestingly, it is not there that we take the decision. The process sends the information back to the central part of the brain, the basal ganglia, to the mammalian brain. And the function of this step is to tap into our entire life experiences and emotional wisdom. It says, in that context, that works for me, in that context, it doesn't work for me. Is it important or not important? And interestingly, this part of the brain is also associated with moral values. The crux is that when this part of the brain, which will give the decision, it is there that the decision is taken, it will not send the information in a form of thought. It will send the decision in a form of feeling. So if we are not trained to regulate our mind and to pay attention to our feelings, how are we going to tap into our inner wisdom and take the best decisions? And here I think that meditation can take us to higher levels. It is shown that when people are meditating on the heart, they develop heart-brain coherence and they are better in connective activities. And when we meditate on higher points, higher subjects like compassion, care, kindness, people are not only able to take good decisions for themselves, but also for their community, for the greater good. And they are able to capture intuitive informations. So that was for my first point. We need wisdom as much as we need science. And that cannot be learned just by reading books. And practically, it can help us with meditation to develop this wisdom and take better decisions. Another point, the last one that I would like to raise is, again by quoting one person you know probably, Albert Einstein, we cannot solve a problem from the same level of consciousness which has created it. We need to move to higher levels. It is good to have medicines, but it may be not curing everything. I may be healthy, I may be materially well, but am I happy and fulfilled? Not necessarily. The mental aspect of taking care of people somehow needs to be also taken into consideration. So the invitation here is to consider health with an integrative approach, not only medicals, but also sports, yoga, alternative medicines, and meditation. And why meditation would help then? Let me ask you a question. Do you think that happiness is given, that it is just natural and I will be happy like that? Who thinks that happiness is natural? Do you understand my French English or? So anyway, let me ask you another question. 10 things happened to you today. 9 were good, 1 was bad. When you go back home, which one are you going to speak about to your friends? Right. And why? Because our brain is wired to be negative. Because it is more profitable to be negative than positive for survival. And that has tremendous psychological impact. Negativity sticks to the brain. Positivity flows through the brain. It requires 5 positive experiences to neutralize 1 positive experience. So somehow, to be happy, and here again, a warm invitation to the students, we need to learn to take in the good. It's not sufficient to be positive. We need to learn to take in the good. Positive strategies may just be, leading to short-term results. How to take in the good? First, we have to pause. Detach from the external circumstances. 2, focus on something positive. But that is not sufficient because we create a neural connection, and as we think about something else, this neural connection is pruning. So we need a 3rd step. Feel, absorb, savor good experiences. Savor the condition which is in your heart. And that will teach, that will weave, that will create a body, brain, mind experiences which will weave our subconscious mind with positive values of the heart. Love, care, kindness, generosity, appreciation, harmony, courage. And that will support a mindset which will be positive and caring in whatever activities that I do. 2 messages. Learn to cultivate wisdom. That is not sufficient to read books. It requires a mental training. 2nd, let's elevate our consciousness and take perspective and integrate our work, our researches, by having a holistic and integrative approach of life. And meditation can surely help us to imbibe our mind with the hard values, which are the best qualities of the human mind, and lead us to happiness. I'm very happy to have been given this opportunity to share this with you. If I am permitted, I would like to make a short experience of 30 seconds. Let's take a moment to close our eyes. Breathe naturally. Close your eyes and bring your attention to your heart. And feel the source of light which is in your heart. Connect with your deepest self. And invite feel the longing which is there to be one and the highest you can be. And invite this deeper self to guide you in all your decisions and actions. Thank you. You can slowly open your eyes and remain connected with your heart. Thank you for your patience listening.
Thank you, Mr. Stan, for the wonderful, nourishing session. I now take this opportunity in welcoming Dr. Shashir Kumar, Managing Director at Havel Pharmaceuticals Private Limited, to address the gathering. We welcome you, sir.
Good morning. Respected dignitaries, and thank you for inviting me. Mr. Roland Ross, thanks a lot. Maybe it's a last-minute entry, but I'm enjoying it. I'm happy to be in this gathering. I represent startup side of life sciences. So I think out of all panel, what you have seen regulators, you have seen administrators, you have seen academicians, you have seen career counselors. So I'm the other part, startup side. So I'm to give you a little bit brief, I probably that might encourage you to become a good entrepreneur later. So I'm from a small town in Nizamabad, as part of Telangana. I did my most of the education. Till graduation, I was in government schools, government college. Subsequently, I left to master's in JNU, and then subsequently I did PhD in Usmania and CCMB. So objective of things is like we started bootstrapping, selling our house, I didn't have any financial background, so did lots of efforts to get things together, and especially where we are not from IITs or IIMs, but still gathered lots of energy, and together with my wife, we started a company. And subsequently that emerged into a good startup, and then it was sold to a company, French company, BioMe'23 took over. And subsequently we started, that encouraged us to do more to society. See, I think the best thing what we felt is giving back to society is generation of employment. So once you employ one direct employee, and there are lots of people who are dependent on them, and you get more, that's the way what I feel is giving back to society. So in 2017, we started back again this HUWEL. HUWEL is acronym for human welfare. So that's how we started. We are into diagnostic space. So I would encourage more students to focus on your basics more. At this point, I would not suggest you to go and do all exploring entrepreneurship right away. Entrepreneurship is a big journey. It's a long journey. It's struggle. It's hard work, as others said. So I wish you focus more on the basics of science and understand more and more the basics of pharmacy and whatever course which you select, and understand more about the subject, maybe fine arts, maybe pharmacy. So you need to focus more on your science. And then once you are solid in your basics, as somebody said, doesn't come by Googling a specific subject, it comes more by involvement, your involvement in the subject. So once you understand more about your subject, develop your basics, and then you can think what is the unmet need. See, the country, unmet need, country is vast. Now we overtook China with the population. So country is vast. So once you think of any product which is applicable to masses, would naturally generate your revenue. So focus on what is unmet needs, and then try to focus on how to do it. And do lots of planning around it, then you take up the entrepreneurship more. And that's a hard journey, so you need to have proper, every step very well worked out before plunging into entrepreneurship. And I think Telangana has provided lots of great ecosystems, like you have, that is demonstrated by the people on dignitaries on dais. Like we have all together right now, administrators, regulators, and incubators, and all career counselors. So we are all in one platter right now. So I think utilize this opportunity to understand where you stand and how you want to proceed in life, and take more opportunities. And Telangana as such have very big, I'm in the medical devices sector, so there are lots of, 80% is still imported. So we are trying to replace more and more and get more technologies. I think this platform should also be utilized for more collaborations, more tech transfers, more understanding about how to work together. I think the concept of Vasudevaka Kutumbakam should be more towards globalization of indigenization. So come together, understand, do more collaborative research, and do work together. When the ecosystem is very favorable, I think Hyderabad can offer you everything what is to become an entrepreneur. Thanks a lot. Thanks for the opportunity given to me. And I'm there for any of the, see, I think I would wish all the students do lots of while being students only, you can visit lots of industries. Once you're out of studentship, it's very difficult to approach any industry to look into. So please do visit lots of industries. Any industries in the medical devices park, I can connect them. So please explore as much as possible before you plunge into your regular life. Thank you. Absolutely, absolutely. I'm more than welcome. We are into chemistry. We are all around DNA-based research. We are now making DNA therapeutics as well. We vaccine adjuvants in terms of around nucleic acid-based. Apart from nucleic acid-based diagnostics, we are also into chemistry part, mostly in terms of point-of-care testing in the chemistry side. So anybody at Medical Devices Park, please do visit us. It's a budding startup company, which is well spread and built in good CE mark or FDA compliant facility. Do visit us. It's at Medical Devices Park, Sultanpur. Thank you.
Thank you, Dr. Shashir, for an enlightening talk. We are grateful to have you. And now, as a token of gratitude for gracing the inaugural ceremony, we would like to felicitate our esteemed guests present on the stage with an amazing artwork done by our fine arts students. Now, I would like to request Dr. Amit K. Tiwari to come forward to felicitate our guest of honor, Dr. Ram Kishan. Thank you, sir. Now I request Dr. Scott to felicitate Professor Redana, sir. Please give a round of applause. Thank you, sir. May I now call upon Dr. Kamal to felicitate our guest of honor, Dr. Shashir Kumar, sir. Now, I would like to request Dr. Sai Hanuman Boddu sir to felicitate our guest of honor, Sri Paneer Selvan sir. Please join us as we felicitate our guest. Please give a big round of applause. Thank you, sir. Now, I request Dr. Darshana Joshi to come forward to felicitate our guest of honor, Mr. Stan. Thank you, sir, and thank you, dear students. I once again thank respected dignitaries for gracing this inaugural ceremony by sparing your precious time despite your busy schedule. We are indeed blessed to have you today. Thank you all once again. Dear ladies and gentlemen, before we begin the keynote address, the attendees are welcome to a tea break which is served at N3, the satellite beside the exit. And I request the dignitaries on the stage to have a tea beside the stage, the room beside the stage. Thank you all. And we'll join the session back in 10 minutes. I request all the participants to join us back in 10 minutes. Thank you all. And one more announcement. Participants, please notice that we'll have poster presentations during the tea break. Thank you. possible because we have come to the core of the meeting. I request all the participants to be seated. Welcome back, ladies and gentlemen. We have come to the core agenda of our meeting, which is to hear from our keynote speaker. The keynote speaker will be rendered by Dr. Amit K. Tiwari. May I now request Ms. P. Sindura, faculty in microbiology, to introduce our keynote speaker.
I consider it a great honor to introduce our keynote speaker, Dr. Amit K. Tiwari. Unable to understand where to start from and what to include or exclude from innumerable noteworthy achievements of Dr. Tiwari, I am presenting here just a glimpse of his splendid personality. Dr. Amit K. Tiwari did his MS and PhD from St. John's University, New York, which was followed by a postdoctoral training from Mount Sinai School of Medicine, New York. He is currently a full professor in the Department of Pharmacology and Experimental Therapeutics at the College of Pharmacy, with joint position in College of Medicine at the University of Toledo. He is director of the Cancer and System Therapeutics Program and also serves as the director of International Engagement at the University of Toledo. He is also a Common Breast Cancer Fellow and UFRI Fellow from National Science Foundation, USA. He is a board-certified toxicologist and is a diplomate of American Board of Toxicology. Dr. Tiwari is a world-renowned expert who has made substantial scientific contribution in discovering, designing, and developing targeted anti-cancer therapeutics against advanced multi-drug resistant cancers. In these areas, Dr. Tiwari holds several patent applications, has authored over 165 peer-reviewed publications and over 200 presentations in national and international conferences. Dr. Tiwari's work is widely cited and he has received several awards for his excellence in research and teaching. Dr. Tiwari serves on a number of grand panels and is a reviewer of numerous scientific journals. He is an editor-in-chief of Cancer Reports and an associate editor for Frontiers in Pharmacology, Frontiers in Oncology, Journal of Clinical Medicine, and is on the editorial board of over 20 prominent scientific journals. He is a reviewer of over 225 journals. Dr. Tiwari's research is supported by grants from federal agencies, including US Department of Defense, National Cancer Institute, National Science Foundation, Common Breast Cancer Foundation, international agencies, private foundations, and several pharmaceutical companies. In short, he has set a mission to save human beings from one of the most fatal diseases on the earth, cancer. His current research projects in the CAST are related to developing more efficacious and safe anti-cancer molecules, overcoming apoptosis and other forms of drug resistance. With this brief introduction, I would now request Dr. Amit K. Tiwari to deliver his talk titled innovative approaches to induce unconventional cell death in multi-drug resistant cancers, leading us to better hopes based on his evolution. We are truly blessed to have you here, sir.
It needs to be turned on. Okay. I think you can hear my voice now. Good. I think you can pull up my slides if it's possible. First of all, let me say thanks for giving such a lovely and long introduction. I'm not used to such a long introduction. Thank you so much. And I feel honored and privileged to be standing here and talking to such a lovely audience. You know, having this conference of such a scale is not easy task, right? So I really want to thank and congratulate the organizing committee for supporting this conference, the government of Telangana, so the Society of Tribal Welfare and Social Welfare for putting this together in collaboration with Life Sciences, Telangana Life Sciences, FABA community, and also Heartfulness Education Trust, right? So a lot of people have come together to bring this conference together. So, you know, we always talk about speakers and other people who come here on the stage and talk to you about you know, what are new developments and in biology and medicine that we will be talking about. But putting this together has not been an easy task, right? So it's students, staff, faculty, visionary leaders like your patron, Mr. Rose, and also the conveners of the conference. They all came together, and I really want to thank, and let's put in our hands together to congratulate and appreciate their effort. Wonderful. So let's begin this conference by saying that there are a lot of new development that is going on in the area of science, life sciences, particularly in biology and medicine. So you will, in next couple of days, you will learn about a lot of new discoveries, a lot of new innovation that people will be talking about, and how they're approaching these discoveries. So we will learn a lot about those. And so I will be talking about some newer discoveries in the area of cancer research, particularly in the area of multi-drug resistance. And so you will be hearing a lot of different you know, professors and scientific leaders in different areas to talk about this conference. But let me tell you that I was just like you, you know, sitting in the back as a pharmacy student, okay? So I have come from here to this place, and I'm talking to you. And the goal of this conference is really to see if we can inspire few students, if not all, to take up research and you know, go forward with this journey. You are the next generation of scientists, right? So we are looking up to you. And so please feel free to interact with investigators and professors here. They really love when students come and talk to them. Okay, so this conference is dedicated and solely is targeted to pharmacy students and life sciences students. And I hope that you know, you will enjoy you know, the conference that we are going to have for next few days. So my talk is on, you know, what I'm going to tell is about two new innovative technologies that we have come forward with to target drug-resistant cancer, okay? And I'm hoping that I can teach you what is drug resistance and the process of drug discovery and drug development to all the way to, you know, conducting the preclinical stages. And then you heard previous dignitaries talking about, you know, how to move them into commercial space, entrepreneurship, setting up companies and all that. But there's a lot of things goes you know, before that. And so one of the speakers actually also, told that learn the basics first before you can actually begin the journey of setting up companies and entrepreneurship, right? So here, my hope is that you will learn about the drug discovery process, you'll learn about multi-drug resistance, and you'll learn about a new cell death mechanism that we have identified, okay? So before that, let me just tell you where I'm from. This is the University of Toledo. I don't think this pointer will work. So as you can see, University of Toledo is on the border of Michigan and Ohio. It's one of the public university. It is quite large campus, just like Ghana Shantivanam, spread across in 15 to 1600 square acres. This is the main campus, and on the right-hand side, you are seeing my building where I work on Health Science Campus. We have 450 different programs, from undergrad to graduate programs, and one of the speakers from University of Toledo is going to talk about some of those programs later. Okay, so that's where I'm housed, and that's where most of my research is done. Okay, so this is my research team, and I wear a number of different hats. Right, so my lab is focused on precision medicine. We try to design and develop new anti-cancer molecule against, difficult to treat cancer, especially drug-resistant cancer. So we have certain targets that we are designing our molecules, so that is precision medicine. And our approach is mostly pharmacokinetic and pharmacodynamic driven, and you'll hear those terms as you go through your pharmacy journey. Okay. In addition, part of my lab is synthetic chemistry. We do design and synthesize those molecules. Then we work with collaborators, and some of my team members are very interested in delivering those molecules to the right target using innovative approaches. And so I'm very interested in drug delivery and drug-drug interaction. And again, as a transporter biologist and transporter person, our lab also does a lot of molecular biology. So you heard about genomics and other omics. We do a lot of molecular biology. You will get some flavor of those in my talk today. So that's a part of work in the lab. And the newer area that we have gotten into is development of new devices and new assays. So we have some posters on that. I encourage you to visit those posters where we have come with new devices. But today's talk, I'm just going to focus on discussing about a very complex area, multi-drug resistance, and how we have approached it. So the idea is that I hope that you will be able to understand what is multi-drug resistance after this talk, what are people doing to overcome multi-drug resistance, and what have we done and come across the new technologies that we are very excited about. So I hope I can excite you about some of those resistance research that we are doing. And so you'll also be learning about some new term, and I'm hoping that you are sitting here to learn something new, okay? So if you have any questions, please feel free to ask, after the conference. So can we get the slides back, please? So let's take a step back. Cancer is the second leading cause of death, as we all know, right? There are a lot of ways we can treat cancer, but basically surgery, radiation, and chemotherapy is the mainstay, and now immunotherapy is the mainstay. However, in most cases, when patients don't respond to this chemotherapy, they develop what we call drug resistance, right? And that's when chemotherapy does not work in most of these patients. What is drug resistance? Really, it is, you're treating a patient with a particular chemotherapy or immunotherapy, and it is working at a certain dose. And after over a period of time, these cancer cells are very smart, right? They develop different receptors, different factors by which they resist those drugs to produce their action, right? So you have to give more drug to get the same action. When we talk about giving more drug to get the same action, we say that it's getting drug resistant. It's resisting those drugs to target those sites, right? Unfortunately, they don't only get resistant to a particular drug the patient is being treated, they get resistant to other class of drug, which is structurally and mechanistically unrelated. We call it multi-drug resistance. And if you talk to an oncologist, right, a cancer doctor, almost everybody will say that patients usually don't die of cancer when the cancer is localized. Only when it spreads to different part of the body, and that spread we call it as metastasis, right? When the tumor metastasizes to different part of the body, that is when patient is in stage 3 and stage 4 of their cancer, and that's when they actually die. And now it is, well sort of accepted that this metastasis usually occur when tumors do not respond to chemotherapy. In other words, in 90% of the cases, these tumor spreads after the patient stops responding to these chemo drugs, or they get multi-drug resistance. So it's a major, major problem, right? We need to find a way to really address this multi-drug resistance. But before we do that, as I always say, learn the basics. We have to understand what is multi-drug resistance before you can actually treat it. So if you talk about a drug, right? A drug needs to be absorbed in the system. They have to be distributed. Then they should go to the target site, whichever cancer we are targeting, breast cancer, colorectal cancer, or prostate cancer, depending on which organ the cancer is in. It should go at the target site, then it should go inside the cancer cells. Once inside the cancer cells, the drug has to be activated. Then it should go and bind to its cellular target, and then it should produce its action, which is to kill the cancer cells, because we really want to kill the cancer cells when we talk about cancer therapy, right? Once it has done its job, the drug needs to be metabolized and then excreted from the system. Very simple pharmacokinetics, right? What drug does to the body, or what body does to the drug. Now, in this process, oftentimes what happens, how the drug becomes resistant is, what if the drug is not absorbed enough, or not distributed enough, or it does not enter the cell because of these different carriers and other transporters, which is not letting the drug come in. And even if the drug comes inside, what if the drug is deactivated? Means it is metabolized inside the cells. Or what if there is a mutation in the target it is trying to target? Drug will not work as it's supposed to work. We call it drug resistance. So 95 to 98% of the cancer drugs kill the cancer cells through a process called apoptosis, right? Apoptosis is a programmed cell death, and there is a cascade of event that occurs to produce apoptosis. So if there is a mutation in that pathway, or we call it dysfunctional apoptosis, the drug will not do its job. So patients get drug resistant. Or in some cases, there are some transporters called drug efflux transporters, and we will talk about it. They pump the drug outside the cells and do not allow the drug to accumulate inside the cell enough to kill the cancer cells. Okay, that can also lead to drug resistance. Or the drug can be just simply quickly metabolized in the systemic circulation and excreted very quickly. That can also lead to drug resistance. Overall, there are so many things that can go wrong to cause drug resistance, okay? And when I started my pharmacy journey, and I was interested to become a pharmacy student, my motivation was that drugs are like magic, right? They are like magic. You give it to the patient, they are treated, I didn't know the mechanism of action earlier, but that's what I thought about drugs. They're like magic, just you can treat anybody with drugs. But then I learned eventually that these drugs eventually fail in many, many patients, and not all patients can benefit with the true action of the drug. And so I was very intrigued, and I wanted to understand why that is so, right? So if you see, there are so many different things that can happen to drug. They can be less or more absorbed or bioavailable in the systemic circulation. They can produce toxicity or side effects. They can have drug-drug interactions, or they can be multi-drug resistant, right? Multi-drug resistance can limit that. And early in my career, I learned that ABC transporters, these are efflux transporters, which has been implicated in all these factors. So that's where I began my journey. I really wanted to understand, wow, these transporters are so important that we need to really, really understand them and see how we can find ways to target those transporters. So let's learn about these transporters quickly, right? These are ATP binding cassette transporters, that's what they are called. These are membrane transporters. So there are 49 of them which are present on the cell membranes throughout our body, okay? They are generally good people, right? They actually protect your body from xenobiotics or any foreign substances, okay? That's their role, right? But when it comes to drug, they also think that drugs are xenobiotic and they throw them out of the system, and I'll explain you how they do that. But looking at their importance, you should know that almost all the drugs that are in the clinic, including cancer drugs or hypertension drugs or cardiovascular other diseases drugs or addiction drugs or other infectious drugs, 50% can be pumped out by these transporters, meaning ABC transporters can cause drug resistance to these many drugs. They can alter their pharmacokinetics for these many drugs, right? Other thing that you will appreciate is that these transporters are overexpressed in more than 70% of the tumor, meaning that they are one of the leading cause of drug resistance, right? So just to understand their mechanism for you guys, if you just understand this cartoon and see, this is a cell membrane, this is a bed of cell membrane, right? Just a portion of it. And as you know, the drug enters the cell either through diffusion or carrier-mediated transport, right? But because of the presence of these transporters, ABC transporter called as ABCB1, popularly known as P-glycoprotein or PGP, that I'm sure that you will read in your literature, in your curriculum. Because of the presence of these transporters, ABCB1 or ABCC1, they pump the drug outside the cells, and unfortunately, they pump these anti-cancer drug outside the cell. So there is less drug that is available inside to kill the cancer cells, right? So that is what we know about these drugs. So to improve chemotherapy, we came across a very simple hypothesis, right? Now these transporters are so important, they are doing bad things to anti-cancer drug. What if we just block its ATP function, because they need ATP to pump out the drug outside the system? And so we tried with that hypothesis, if you can block that transporter, it will allow more accumulation of the drug, will lead to, reversal of multi-drug resistance. And that was our goal, to treat patients who are not responding to chemotherapy. We used that approach and we came up with so many different innovative ideas. We published a lot of public papers on this ideas, and every time we thought that we are coming close to it, we have made a very little progress, okay? So far, there is no FDA-approved ABC transporter inhibitor in the clinic, okay? So a lot of work has been gone into this area, and there has been failed outcomes, and I can tell you about a lot of reasons why these ABC transporters did not make it to the clinic. The clinical trials failed because the inhibitors were non-selective. They were causing pharmacokinetic damages. But my hypothesis, and few of people in ABC transporter field have come to believe that most of the people who were targeting these ABC transporters were only targeting one ABC transporter at one time. While we know that there are so many ABC transporters that are available, right? I told 49, but out of them, 9 are very important in drug resistance. So, bottom line is there is no FDA-approved drug in the clinic, and so we started looking into this problem a little bit more in detail. And as I said, that these transporters are expressed or co-expressed at the same organ sites. You talk about the main organ type like brain or blood-brain barrier or kidney, liver, small intestine, and you can see that plus sign means that they are overexpressed on the same site. So there are more transporters who are present on the same site. Also, they have similar functions. They have functional overlap, right? So they can pump a lot of drug. More than one transporter can pump a lot of drug. So if you're inhibiting only one transporter, how are you going to cure the problem, right? So one of the approaches that we thought was, okay, let's look at a little bit more deeper. So, just to make matter a little bit more complex, not only these transporters are co-expressed and co-localized and have overlapping function, if you look at the intracellular level at hepatic or kidney level or intestine level, you can see these transporters have different orientation. Some transporters pump, apically, into biliary and allows for excretion, while other transporters pump the drug inside the blood. I don't expect you to remember any of this. However, I hope that you are just following a bigger picture that drug resistance is a major problem. ABC transporter is another major problem which people have tried to target and have limited success because of complexity of the system. And they're only one of those multi-drug resistance factors. What approach we took to target is that we came across newer drugs and inhibitors that can target more than one transporters at the same time, okay? So we are still working in this area. We are still trying to come up with permutations and combinations of how we can inhibit more important transporters that are expressed in particular cancer that leads to drug resistance. All good, but ABC transporter is only one small drug resistance. I should not say small. It's a very important drug resistance factor, but even bigger than that we thought that, okay, we are still working on this. Let's understand this drug resistance problem a little bit more clearly. So we now started tackling even more important or even a bigger problem, which was mutation in apoptosis signaling pathway, okay? In that, I'm going to show you some really, really cool stuff that we think is very cool because we developed it, of course. But also, I think you will appreciate how much work it goes to bring those drugs from the bench side to all the way where we think that it can go to do more preclinical testing to clinical side, okay? So you will learn about some new things today. Methuophagy, I don't think anybody have heard this term before. Even some of the faculties might not have heard this term before because we coined it, right? So it's a very, very new concept. And so we'll talk about this, and you will learn about this funny face in a few minutes, okay? But as I said, let's go back to basics. Before I tell you about this new cell death mechanism that we have identified, let's learn about what is cell death. So as you know that when cancer cells die, they can die of two ways, mainly two ways. One is apoptosis. So when you give anti-cancer drug to a cancer cells, they actually, there is membrane blebbing. The cell shrinks in shape. There is chromatin condensation, and they form these apoptotic bodies, which is then phagocytosed. Okay, this process is programmed cell death. It's a very regulated process, right? There are a lot of important markers that are available to control this process, but there is no inflammation in this process, so it's apoptosis. So as I said, 95 to 98% of the anti-cancer drug kill the cancer cells through apoptosis. In some rare situations, you can see if the cells are infected by bacteria or viruses or they're injured, they can go through this another cell death process called as necrosis, where the cells swell and then they become leaky and rupture, and they leak out all these debris, and there's a lot of inflammation in that process, okay? So these are the two main type of cell death mechanism. Again, 95% or more clinical drug out there kill the cancer cells through apoptosis. And so as I said earlier, mutation in apoptosis signaling pathway leads to drug resistance, multi-drug resistance, which is one of the major problems, right? So our goal was to discover new drug to find a way to overcome drug resistance. And I'll tell you a story, how we came across a new class of anti-cancer agents and a new cell death mechanism. And I'm going to tell you, I'm not going to go deep into the data. I don't expect you to understand or really go into the methods and material methods of the data. I just hope that you can enjoy the story. And I'm going to relate it in a story format only, like I tell my 11-year-old daughter, okay? So here we go. So once upon a time, not so long time ago, okay? One of my students, as pictured here, Haneen, we designed and synthesized close to 40 plus compounds, okay? And we were these were dual tyrosine kinase inhibitors, meaning they were precision medicine. We were trying to target two very important targets. And so she was trying to do and understand how they were killing the cancer cells. And out of that 40 plus compounds, many were killing exactly how we expected them to kill. And they were very selective and all that. What she came running to my office one day and says, Dr. There's a very unusual thing I'm seeing under the microscope. Can you come and check And I wait for these kind of moments because I get excited. That's why I'm into research, right? New discoveries. And this was an accidental discovery. We never thought that we will bump into something so cool. So this is what we saw under the microscope. She showed me these cells. These are colon cancer cells. And you can see a lot of gunshot wounds if somebody has like shot them multiple times. A friend of mine called them like Swiss cheese model. But these cells were actually drinking themselves. There were a lot of vacuoles that were forming, and as if they were like drinking themselves, which is very unusual. So these vacuoles were forming. They were like coming close together. They were forming bigger vacuoles, and then they were detaching from the surface where they are cultured. And then they come up, and then pop, they were like, dying. This is a very, very unique phenomenon. And we being scientists, right, we don't really believe everything that we see right away. We have to really, really make sure that that is replicable and it is what we are seeing. So I made her like do that through multiple different ways, and every time we got the same data, right? So it was very cool, and the IC50 was around 24 micromolar. So just so you know what 24 micromolar is, generally speaking, very general speaking, it is still high on the higher side of killing the cancer cells, okay? So when we observed this, we had no idea what we have bumped into. So we started looking into the literature, and we came across Dr. Bill Maltese' work. He was a professor and a chair at University of Toledo, and was such a coincidence that we looked into his work, and he published a new cell death mechanism. He called that cell death mechanism as methoosis. Now, what is methoosis? Okay? It's a tongue twister, but you should understand what is methoosis. In Greek, methoosis means drink to intoxication, meaning if you keep on drinking, and your urinary pathway is blocked, what will happen? You will burst open. That is exactly what was happening to cancer cells, right? So they worked on a molecule called MOMIP, where they saw that as soon as the MOMIP was given, there were ruffles that were forming, anything that was extracellular was coming in, and vacuoles were forming, they were fusing together, then they were forming large vacuoles, which should go and bind to the lysosomes. Now, lysosomes are digestive body in our cellular system. They will digest anything that is either eaten or being drunk, or there was no autophagy. So what he observed was this very unique class of compounds that were making the cancer cells drink themselves, right? Not eat themselves, because there was no autophagy, no autophagosome formation. Our first thought was, wow, looks very similar to what we observed from our new class of drug that we have designed and we have developed. So our first thought was that we have come across a new class of methoosis inducers, which is making the cells drink themselves to death. So I put them to work. We all work together to design and synthesize even more potent compounds, assuming that these are the new class of drug which is making the cancer cells drink themselves. From high micromolar, we came to a nanomolar potency from BAP13, BAP27 originated, which you can see a lot of vacuoles are formed, and they become bigger and they die. Let's understand this a little bit more. So not only these vacuoles were forming, but they were actually killing the cancer cells, right? So if you look in the colony formation assay, as you can see, as you treat with different drugs, these cancer colonies were gone, right? That shows that they were very effective in eliminating these cancer cells. So if you look in the mechanism, how do they actually work, right? So if you treat the colon cancer cells with BAP27, within 1 hour, you can see a lot of vacuoles are filled inside the colon cancer cells. These vacuoles will become bigger and bigger. They will become one giant vacuole, and they will detach, and they will burst, okay? And here you can see the cell bursting phenomenon, how they are bursting. All the cellular content is leaking out, okay? And this is transmission electron microscope. Like, you can zoom in 5,000 to 10,000 times more inside the cell, and you can appreciate these vacuoles forming and how they were fusing together by rupturing, becoming a large vacuole. So this is very cool process. We call it macropinocytosis. Cell drinking process is called macropinocytosis. Or when the cells die from this dysregulated macropinocytosis, we call them methoosis, okay? That's what we were observing also. And, you know, and you will appreciate this because this is not enough. This observation is usually not enough for us. We have to really show that really mitosis is happening, so we have to do multiple different assays and look in through multiple different ways to really prove that yes, what we are seeing is exactly what is happening. So we did a bunch of other assays. I'm not going to go in detail, but you can see these vacuoles were forming. They were actually taking dyes from outside, which shows that this is actually a macropinocytosis process. And there are some markers like RAP7, RAP5. These are getting upregulated. This band going up means they are getting upregulated, meaning that their expression is going up, telling you that there's a lot of vacuoles forming, a lot of macropinocytosis going on. That's all it means, right? And you can do more assays to confirm this, you know, from microscopy studies and other studies. You know, but what interesting thing we observed was this dye, like called lysotracker dye, it actually dyes lysosomes, right? And it was shining red, which suggested that there's a lot of acidity going on, okay? Which then we started looking much more closely, and we looked into our electron microscopy data, and what you are seeing here is a very zoomed in image of a single cell, and you're looking at lysosomes, which is like 10 to 15 to 20-fold more swollen compared to the normal lysosomes in a cancer cells. And here are like double membranous structures and other things, which kind of suggested that looks like the cells are also eating themselves. Just wait a minute. We were designing compounds that can make the cells drink themselves. Now, not only they are making the cells drinking themselves, they're also eating themselves, okay? When something eats themselves, that process is called autophagy, self-eating process, right? And when does this happen? When you do fasting, for example. When there is lack of nutrients, there are some signaling pathway that is activated, or when there is too much of stress, these pathways activated, or when there is these organelles that do not function well, like mitochondria and other organelles or proteins and protein aggregates occur. There is this formation of double membranous structure called autophagosomes, right? Which will then digest everything here through lysosomes. So lysosome is a digestive body like our stomach, right? Once these autophagolysosomes are formed, they will release all these metabolic products like amino acid, nucleosides, and fatty acids and sugar, which will allow for more energy for the cell, which is contradictory than what we are actually looking into. Autophagy or self-eating, cancer cells do that all the time. The reason they do that is they want a lot of energy for producing new cancer cells. Remember, cancer is when cells are actually replicating and producing new cancer cells. They are like rapidly dividing, right? There's a lot of autophagy going on. So autophagy is a cell survival mechanism. Here, in our case, we were seeing that cells were actually dying because of too much of autophagy. So you are overwhelming the system so much that they were actually dying from autophagy. So this was very cool that, okay, not only they are drinking to death, they're also eating themselves to death, right? So we started looking into biomarkers. Again, I'm not going to go in detail, but you will appreciate that we looked into a number of different markers, and they were upregulated, showing that autophagy was definitely happening. We looked into other markers of autophagy, and these are different markers of autophagy which showed that, yes, autophagy was going on. In science, then, you can do different other ways to really look at the same thing. For example, you can give other probe to see if you give, what happens if you give more autophagy inducer, something that can do more autophagy along with your drug. If you do more autophagy, naturally the cells should die more, right? And that's what we saw, that if you give autophagy inducer, cells are dying more. Or if you give autophagy inhibitor, such as 3-methyladenine, cells were actually surviving, okay? That suggests that autophagy was leading to cell death, okay? We further probed this, and we looked at other ways, like we added chloroquine, which also allowed cell survival. The reason I'm showing you all this data, I can just simply tell you that, yeah, autophagy was happening and cell death was happening, and the cells were drinking to death, and they were also eating themselves to death, and end of story. But I'm showing you all this data. I know that many of you will not follow it right now because you need to really understand the different markets and other thing. However, you will appreciate how much work it goes to really, really tell the scientific community that yes, what we are observing is true, okay? It's not like, you know, you just say something and they will believe you. The data actually let them believe whatever we say. So yes, the autophagy was happening. So basically what I'm saying is, when we learned this new mechanism that cells were drinking to death and they were eating themselves to death, methoosis was happening, autophagy was happening, we married the two terms together, and methoophagi was born, okay? So that's how we call the new term methoophagi, okay? So you can appreciate that, there's autophagy happening and this methoosis, a lot of vacuoles collecting, and they all lead to cell death, okay? But this is not enough. Okay, you observe the phenomenon that, yes, some drug is killing the cancer cells through doing a cannibalism sort of an effect. They're making them drink and eat themselves to death. But how does it really work, right? So our first goal was to go back to the basics, whether it is apoptosis or not, right? In apoptosis, as I said, cells shrink, there is a chromatin condensation, the cells are packaged after forming these apoptotic bodies, and they die by suicide. In our case, what we saw was that this methoophagi is a non-apoptotic cell death mechanism. Very cool, right? Instead of cells shrinking, they were actually becoming bigger, filled with vacuoles, but they were actually becoming bigger. Nucleus was happy and healthy. There was no shrinkage or anything. They were like good. So chromatin condensation was not seen. In fact, we looked at different markers, and there was no apoptosis happening, right? There was no cleavage of caspase 3, caspase 7, pro-apoptotic markers, back BCL2, there was no change, which suggests that there was no apoptosis happening. And I'll tell you the significance of this finding in a few minutes, right? You can give pan-caspase inhibitors, and there was no rescue for cell death, which also tells that it is non-apoptotic cell death. Then you can look at other cell death mechanism. There is one more cell death mechanism which is non-apoptotic called necroptosis. It's a programmed necrosis. So you can give necrostatin, which is a specific inhibitor of necroptosis, to look at that. But methoophagi is not like necroptosis because it did not rescue the cell death. So methoophagi was non-apoptotic, non-necroptotic cell death mechanism. Then we looked into endoplasmic reticulum, mitochondria, and other things. Why we are looking into all these things? Because there are proven anti-cancer drugs that kill the cancer cells through specific mechanisms. So first thing is, you always go and look at what other people have done to see that if your drug is behaving like any of the other drug that is in the market, right? Or any of the drug that is in the clinic. So there was no ER stress. Mitochondria swelling was seen at a very, very high concentration, so we don't think mitochondria was as much involved. Reactive oxygen species, and some people will talk about inflammation. So reactive oxygen species goes up when the cancer cells are dying or when there is apoptosis, so there was no reactive oxygen species generation. There was beta tubulin was not affected, and you can see a lot of vacuoles forming here, but there was no change in beta tubulin. So there are a lot of anti-cancer drugs that are tubulin inhibitors. They either stabilize it or destabilize tubulin. I'm not going to go in that detail, just so that you know that we checked multiple different ways how cancer cells can be killed, and whether this methoophagi process is aligning to any of that. So it is not a beta tubulin inhibitor either. Then we looked at RAS and P53 mutation, which is very important in colon cancer, right? Colon cancer, why, now you can ask me all those questions, why colon cancer actually begin in the first place? Why, what are the factors that leads to cells becoming cancerous? So to check that, we looked into different mutations. RAS and P53 are very important. And how do you check that? You have multiple cell models and other models where they have different expression of RAS and P53. No matter which different model we choose, methoophagi was happening in almost all of them. So in other words, methoophagi is independent of P53 and RAS status, okay? Now, this was very cool to us. Okay, we found a drug which is doing something very, very cool, killing the cell through a non-apoptotic mechanism, making them drink and eat themselves. We call them methoophagi. Okay? So can we treat those patients who are getting apoptotic resistance? Remember, I told you, right, apoptosis is where most of the drug kill the cancer cells. And if you look at the colon cancer at different stages, almost 100% of the drugs are killed, use their kill the cancer cells through apoptosis, right? So to check whether these class of drug is going to be beneficial to overcome apoptosis resistance, we created new models, and you can create this colon cancer with different knockout models. You can delete some genes to see whether there is apoptosis or non-apoptosis, and we can see here that 5-fluorouracil that kills the cancer cells through apoptosis, this curve shifts to the right. That means that you have to give a lot of 5-fluorouracil to kill this double knockout resistant line. Whereas when you give BAP compound, it doesn't matter what kind of mutation it is, because it is killing through non-apoptotic mechanism, it will still kill those resistant cells, and it does kill at the same concentration, right? So that's what it means. Then you can combine these drugs with different anti-cancer drugs. So this graph shows you that when you combine these drugs with other anti-cancer drugs, they were producing synergistic activity. So that is what you would want as an adjuvant therapy, okay? You can design a drug, you can develop a drug, you can have a drug which is very effective in cancer cells, but if it is not safe and it is toxic, then it's of no use. You know, you cannot give cyanide to a patient even though if it is it's killing something, right? Because it will have other side effects. So we checked, whether they are safe into different cell models, and there was no methodophagy happening in normal cells up to a very high concentration. We looked at their safety in mouse models, and there was no change in weight, which suggests that they were non-toxic. And of course, we checked on other markers of toxicity. We looked into zebrafish up to 100 micromolar, which is a very high concentration to look for toxicity. There was no toxicity. Then we looked into more detailed mechanism of toxicity, and you can look at organ toxicity. So you can look at different organs, liver, kidney, brain, heart, and all that, and you can section them in these animals and look deep inside if there was toxicity. So we did not observe any toxicity with this class of compound. You can look at liver function, kidney functions, other blood markers to see whether there was toxicity. We looked into that. There was no toxicity, right? So we are still working on understanding this class of compounds. Now we are giving patient-derived xenograft models to understand how these drugs are working, right? Now, even though I told you that there is a very cool class of drugs which is killing non-apoptotic cell death and given, producing synergistic action in apoptosis-resistant cells. But what is its mechanism? I have not told you its mechanism, right? So how do we go about hunting for mechanism? There are a number of different approaches you can take. One of the approaches that we took was looking at in the genomics, so you can do sequencing and look at 50,000 genes. What gene is upregulated, what genes are downregulated when you give the drug to the cancer cells, right? So we did that. And we came across certain pathways, and you can do all these pathway analysis to look at what pathway is most disturbed, right? So we looked into that. We also did some kinome arrays and other arrays to look at the targets, and there are a number of different targets that popped up. So we are still trying to verify those targets, if those are the targets that are leading to it. But one of the very interesting things that I told you earlier is what was popping up again and again, which was lysosomes. So we were seeing that lysosomes were swollen, they were more in numbers, and they were actually there was some homeostasis differences in lysosome that was resulting in cell death. So lysosome, as I said, is a digestive body, right? So when autophagosome, those autophagy forms, they should go and bind to the lysosomes. Lysosomes should digest it and produce those energy, which should help in survival. But what was happening is that these autophagosomes were actually confined in the lysosomes. The lysosomes were taking them, but were not converting any energy out. So there was some homeostasis differences between signaling from lysosome to the nucleus. And then we looked into that signaling pathway. That signaling pathway is called mTOR-TFEB signaling pathway. It's called transcription factor EB. And we looked into that pathway, and it really, autophagy was definitely disturbed, and these autolysosomal markers were all upregulated, and this TFEB was significantly downregulated in cytosolic portion, which suggested that yes, it is killing through some of the lysosomal to nucleus signaling pathway. Okay, and I can go on and on and on, going into molecular pathway, but bigger story is, let's learn about what we learned so far. We talked about a drug class of molecule that was producing cell death. So autophagy is generally a good thing. It leads to cell growth and cell survival. But when you give these BAP compounds, it leads to formation of these vacuoles, we call them methoosis. Like a lot of vacuoles form and they can become bigger and bigger, which can lead to cell death by disrupting this called lysosomes, right? So these lysosomes are disrupted through this mTOR TFEB signaling pathway. That's what we think. There is some signaling changes that is happening between lysosome to nucleus, which is leading to all cell death, okay? If you even remember what is methoophagy, I think I'm good here, okay? I know it's a little complicated, but this is a new area of research. Okay, so after that, we designed more compounds, more class of these methoophagy inducers. So far now we have drugs that can produce only methoosis. They can make the cancer cells drink themselves, or can cause only to eat themselves, or can do both, right? Methoophagy. So methoosis, there are some drugs we have designed. So now we are trying to understand what are the structural differences in the chemistry of the molecule. If you can change that, that will lead to different cell death. Okay, so that is what we have done with this project, and so a lot of work is going on. So we have, I'm not going to go in the data, but yes, most of these compounds are affecting lysosomal trafficking. Okay. All right. So I think that was a marathon, and I don't know how much we are doing with time. Okay. So I'll just take another quick project. It's not more than 15 minutes. We will come to the questions later. Otherwise, I will definitely not be able to finish my talk. So this was methoophagy in colon cancer, right? I just quickly want to share another technology that we have discovered, a new class of drug, which is very effective in triple-negative breast cancer. So as you know, breast cancer is a very problematic cancer in women. And even breast cancer, there are different subtypes. There are hormone-positive breast cancer, progesterone positive, estrogen positive, or HER2 positive. When all these three receptors are absent, there's fourth type of breast cancer. It's called triple-negative breast cancer, okay? This is one of the most lethal, most aggressive form of breast cancer that we don't have so many treatment options. So we were very interested in working in that area because these cancers also become drug resistant very quickly, and there's not much that is done. We use a phenotypic screening approach, and we have a poster that you will learn about phenotypic screening approach. Through that, we came across a new class of compound. So we were looking at those drugs that is not making the cells shrink, but instead expand, and they were killing through a programmed cell death. So we identified a new class of drug called TPH compounds, or TPH104 compounds. These drugs were very effective, very selective in triple-negative breast cancer patient compared to the normal cells. And you can do a bunch of different assays. These are colony formation assays where you can see how the cells are dying at different concentration. You can do other assays, MTT assay, cell titer blue assay, SRB assay. These are different cell types, cytotoxicity assay to confirm the same thing. So it's very selective in triple-negative breast cancer compared to non-triple-negative breast cancer, or compared to the normal breast cells, right? So very effective class of drug. We looked into its mechanism. So you look at how the cells are, if you just let the cells grow, they will proliferate and become bigger. But when you give TPH compounds at different concentration, it will lead to cell death, 100% cell death in many cases. You can look at their cell cycle, at what stages the cancer cells are dying. So you can do cell cycle analysis. We did that. We also then wanted to understand that this new class of drug, how are they killing the cancer cells? Are they killing through apoptosis or some other mechanism? So these drugs are very, very cool, right? So if you look into these drugs, I don't know if I can play this. I don't think I can play this here somehow. But this is a very nice video. I know that sometimes the video does not work. So I captured different slides of this video at different time points, right? So you can see these cells, when they're treated, they become bigger and bigger and bigger, and then they burst, which is a very cool programmed necrosis-like cell death mechanism, right? So we have screened this, and we have done a lot of work understanding its mechanism. But what other cool things I want to tell you about this class of drug is, first, it's a non-apoptotic cell death mechanism. And how do you know it's non-apoptotic? You have to run a lot of different assays, a lot of different markers to look at whether it is apoptosis or non-apoptosis. So we did that. We did pan-caspase inhibitor, ZVAD. We looked if the nucleus was shrinking or not. Then you compare it with the positive control, and so on. It's a very safe class of compounds. As you can see, TPH is very effective in in triple-negative breast cancer. Also, it's very safe. You can look at different organ toxicity, brain, liver, heart, pancreas, kidney, spleen. We looked at all this toxicity. And it's very effective in killing the tumor. So you can induce tumors in mouse, right? And then it can keep growing, but when you give TPH compound, it's even more effective than the drug that is used in the clinic, which is doxorubicin, at least the dose that we tested, right? It has some solubility issues that we are trying to overcome, so there are some issues with this class of drug, right? So that's where we need formulation scientists to overcome the solubility issues. But overall, this class of drug is also very safe in zebrafish and animal studies that we have tested so far, okay? So what is the mechanism of this new class of other drug? In the past, you learned that there is a lysosomal signaling pathway for methylophagy. In this class, what we found is mitochondria is somehow disturbed. So if you look at the mitochondria, which is a powerhouse of the cell where all the energy, ATP, is generated, they were very elongated. I mean, because of light, you cannot see this figure very clearly, but they were very elongated. And as you give this compound, total mitochondrial content was going down. So mitochondrial is in constant phase of fission and fusion. So when you learn about mitochondrial biology, you will understand that. So it has to be cut down by the seizures called DRP1 or dynamin-related protein 1, which leads to two daughter mitochondria. But what we know that DRP1 is highly upregulated in triple-negative breast cancer, okay? So what we observed with this new class of compound, but before that we also checked DRP1 status in human patients, and we saw that DRP1 is really, really upregulated in triple-negative breast cancer patients. These are human patient data, okay? When we looked at our drug and their effects on DRP1, they were actually binding to this DRP1 with a very high affinity. This is a molecular modeling assays that we can done on computational systems, right, in silico modeling. But you can also confirm this with binding affinity assay with surface plasmon resonance that we use in the lab. And we found that they were binding to this target, DRP1, very, very effectively. So before this, there are not many DRP1 inhibitors out there in the market. Whatever is available kills cells with a very high concentration, like MDV1 kill the cancer cells at 50 micromolar, whereas TPH compounds kill the triple-negative breast cancer cells at 0.1 micromolar, 500-fold less drug you have to give to kill the cancer cells. Very, very cool. Right? So then we looked into this DRP1 mechanism, downstream signaling pathways, and I'm not going to go in detail, but so that you know that this is a new class of drug that kills the triple-negative breast cancer by DRP1 inhibition. Okay. It can also overcome triple-negative breast cancer chemoresistance, and you can create these different chemoresistance models, and we have created many of them. And we created this taxane-resistant model, which is very resistant to paclitaxel, which is another drug that is used in, for example, triple-negative breast cancer therapy. But when you give TPH-104, it's very, very effective in overcoming taxane resistance. Pia Vogel is going to talk about her research tomorrow on drug resistance, but she also worked on our TPH compounds, and she found that these are very effective drugs to overcome drug resistance. So right now we are designing and developing this class of compound. As you can appreciate, there's a lot of chemistry, there's a lot of work goes to actually understand and optimize the drug molecule to actually be able to make it useful and effective in treating cancer. Okay, so this is all I have to talk today. I've given you two new technologies. I don't know how much you knew before about this methylophagy and this new necroptosis-like cell death mechanism, TPH class of compounds, but very effective. And this is not possible without right team, right people. So I'm very appreciative of all the students and staff members and postdocs who have worked to achieve this at University of Toledo, and collaborators at University of Toledo, and also collaborators that I collaborate outside of UT, who have all helped me in my research. And this is only one portion of work that I've shown you. You know, as I said, we have a number of different projects going on in a number of different areas, but I thought this was very cool, and I really wanted to share with you. It's not even published, so you know, we have to do a lot more work to really make sure that it's reproducible and can be publishable, right? So with that, we have some more new techniques and technologies that we are discovering. So I'm not going to talk about any of those. We have posters out there. Please go and visit those posters when you get chance, and you will see we have developed some new microfluidic devices. We have also discovered new microscope. This is 3D printed microscope, which can do holographic microscopy, and we can do phenotypic screening through this new microscope, patent pending, right? so you have shown all of that, so you should be appreciative of this new class of drugs. So we have a few posters about that. We have also posters on anti-cancer drug discovery, how we design and develop new anti-cancer molecule. We have some poster on the thing that I talked today on DRP1 inhibitor, so you can read about it, you can ask more questions about it, and I'll be happy to answer them. If I don't have answer, I'll try to find some for you, right? And lastly, I want to quickly talk 1 minute about our initiative that we have with the T-Sweries and Government of Telangana. So we have an MOU with Social Welfare Residential Educational Society, and we have been working past 5 years now almost, but last 4 years more effectively. So the first cohort of students came to University of Toledo to do their internship. Some of your previous past colleagues were here, and there were 5 girls who came through this association at UW Toledo as a summer undergraduate research fellows. They did a lot of research. They were met with faculty one-on-one. They actually went into the lab, did a lot of research, bench work. They got a lot of benefits talking to other students, other graduate students who are doing research in the lab. They met a lot of community people, had community sort of time with people, how they live, and people were very nice to have all these students come to them and explain them how the system works. All of these students came back. Unfortunately, COVID hit at the wrong time. They have to come back just before their program was about to end. They reached home safely, and now all of them are doing very, very, very well, and we are very proud of them, right? So now we are working on next cohort of students, and I'm very thankful to T-Sweries and T-Tweries, both tribal welfare and social welfare, to be willing to work with University of Toledo and get you out there so that you can have that experience firsthand. You should be very thankful and blessed to have this kind of opportunity. Not many people can even think about, getting this kind of opportunity to work and learn, things firsthand. So we have been doing that. We have been still working on this. So if you have any questions, please feel free to ask. And again, I want to thank T-Sweries and T-Tweries for organizing this conference, and the convener, Dr. Pavani, has been, I should have a few words for her, but her team, it's never one person, as you know. The visionary leadership of Mr. Rose has been phenomenal, and every single member, volunteers, students, staff, and fellows who have been supporting this, other faculty members who have been participating in this, and it's amazing how we have come together to witness this scientific forum, right? With that, I will stop here. This is my favorite slide. You can ask any question that you have. This is a new data we just got like 2 weeks ago, doing microscopy, transmission electron microscopy. We gave one of the BAPT compounds looking at how they are killing the cancer cells, but this is something very, very cool phenomenon that I can share later with you. Okay, so I'll stop here. If you have any questions, you can ask now or maybe later. So yeah, you can ask me questions. I will be here for next 3 days, so please feel free to come and talk to me, and I'll be happy to discuss about any of these projects. Thank you.
Yeah, thank you so much. I'm sure the students have learned about 2 new terms. For sure, I have learned. I have never heard, I'm a life scientist myself, but I've never heard metamorphosis before Amit explained. So thank you so much for teaching us all a new concept of cell death. We have been learning about apoptosis and necrosis since ages. Today we learned a new thing. So thank you so much for that. And due to paucity of time, we are not able to take any questions from the audience, but feel free to interact with the speakers when the other sessions are going on, or during the networking session time during the tea break and all other parts. Okay. Let us have one more round of applause for Professor Amit for the wonderful lecture. Yeah. Amit, please stay back. Yeah. So now may I invite our Additional Secretary T-Sweries, Mr. Hanmant Naik sir, Additional Secretary T-Tweries, Sarveshwar Reddy sir, and our OSD T-Sweries, Chandrakant sir, to come on to the dais to felicitate our keynote speaker. I request both the additional secretaries to present the shawl to our speaker. Shawl. Thank you so much. So, Professor Amit was mentioning about the team at T-Sweries and T-Tweries, and these are the three leaders at their head office, next to Secretary Sir, who are guiding us continuously in all the projects. Thank you, sir. Yeah. Yes. Thank you so much, Amit. We have a couple of announcements. Because we are running behind schedule, Professor Scott's lecture, which was earlier scheduled before lunch, will be scheduled now after the lunch. So I request all the participants to attend lunch, which has already been arranged at the N3 dining facility located just next to the auditorium. And since we are already running late, we have given only 40 minutes. We have made sure to arrange multiple counters so that there is no big rush for you at the lunch. So all the students, all the delegates are requested to proceed to the N3 area for lunch. And before that, I'm sorry, just this is an announcement. Please be seated. And then for all the officers and the speakers, we have the lunch arranged in the VIP dining area in the lobby. Before we move for lunch, I request our MCs to conclude the inaugural session with a vote of thanks.
I would now request Ms. Purnima, Faculty in Life Sciences from Department of Zoology, to propose vote of thanks for the inaugural session.
A very warm good afternoon to all of you. It is not happiness that makes us grateful, it is gratitude that makes us happy. After taking this quote, I now, at the outset, I express my profound gratitude on behalf of T-Sweries and T-Tweries BioMe 2023 to all the dignitaries, eminent speakers, distinguished guests. We welcome you all onto this platform of innovative learning. We are truly enriched by your inspiring words, sir. This goes to the Secretary of T-Sweries and T-Tweries. And we understand the strategic planning, implementation in steering us, sir. A big round of applause to Ronald Ross, sir. I am indebted for your gracious presence in spending your valuable time to all the dignitaries who are present here. And big thanks to the panelists, the speakers that we had this morning, all flown from different parts of the world to make it more meaningful and for being a solid support system to this conference. I genuinely appreciate your flexibility and your spirit of dedication. You have done this fantastic job, the organizing committee. We are more honored by your esteemed presence, without which this accomplishment would not be possible. This goes to the faculty of both T-Sweries and T-Tweries. Thank you for being here. Special mention for handling the media, who had immense effort to make it possible. Ladies and gentlemen, put your hands together for the unsung heroes who have made it this inaugural session a productive one. I honestly feel that this conglomeration of expertise is put forth by our students whose work portrays the confidence and courage by accepting the unending challenges to maneuver the life, transforming into more constructive citizens. The relentless support offered by Kanha Society to make this BioMe 2023 International Conference a world-class experience. Thank you very much for joining us on this warm note of inaugural session and spending your valuable time. Thank you. Thank you very much.
Ladies and gentlemen, the next session is going to start in a minute. Please be seated. On behalf of Telangana Social and Tribal Welfare Residential Educational Institutions Society, Government of Telangana, in association with Telangana Life Sciences, Federation of Asian Biotech Associations, FABA Academy, and Heartfulness Education Trust, we are glad to welcome you once again for the post-lunch session of the International Conference on Advances in Biology and Medicine, BioMe'23. Distinguished guests, ladies and gentlemen, we are going to continue our session with Dr. Kamal Dua, and I request Professor Amit K. Tiwari to chair the session. To introduce the speaker, may I now call my colleague, Kala Jyoti ma'am, to come forward. Thank you.
Thank you, ma'am. I take this opportunity to introduce our distinguished speaker, Dr. Kamal Dua. Dr. Kamal Dua is a senior lecturer in the discipline of pharmacy at the Graduate School of Health, University of Technology, Sydney, Australia. He has research experience of over 13 years in the field of drug delivery systems targeting inflammatory diseases. Dr. Dua is also a node leader of drug delivery research in the Center for Inflammation at Centenary Institute, UTS, where the targets identified from the research projects are pursued to develop novel formulations as the first step towards translation into clinics. Dr. Dua researches into two complementary areas, drug discovery and immunology, specifically addressing how these disciplines can advance one another, helping the community to live longer and healthier. This is evidenced by his extensive publication record in reputed journals. Dr. Dua's research interest focuses on harnessing the pharmaceutical potential of modulating critical regulators such as interleukins and microRNAs and developing new and effective drug delivery formulations for the management of chronic airway diseases. Since 2019, Dr. Dua's research has resulted into more than 300 publications. He has also published 7 books with reputed publishers and more than 70 book chapters. Dr. Dua is also an adjunct assistant professor, Faculty of Pharmacy and Pharmaceutical Sciences, University of Alberta, Canada, core member, Australian Research Centre in Complementary and Integrative Medicine, UTS, research associate at the Australia-China Relation Institute, adjunct fellow, NICM Health Research Institute, Western Sydney University, Australia, adjunct senior lecturer at Taylor University, Malaysia, adjunct visiting academic at Manipal University, visiting academic at Pune Uni, and consultant at ULTI Pharmaceuticals, New Zealand, and D. Aurora, Australia. He also held a conjoint lecturer position with the School of Biomedical Sciences and Pharmacy, the University of Newcastle, Australia. He is an active member of many national and international professional societies. With this brief introduction, I now welcome Dr. Kamal Dua and Dr. Amit onto the stage, please.
There you go. I don't think it's reflecting there. Okay, thank you. Can you tell me how it works? Slide will move with this. Beautiful. And the pointer is this. Beautiful. Thank you so much. That's great. Let me know once it goes there. Okay, great. All right.
It's okay. All right. A very good afternoon to everyone, and thank you so much for listening to me, the post-lunch talk, which means half of the people will sleep. Okay? Well, before I start my talk, let me tell you a little story which I tell everyone in my every single class, the first class of every subject. I was having a friend who just passed away, actually, and it's a reality, and it's a real story, okay? I was having a friend in New Zealand. One day he came to me and he said he's not getting sleep. I said, what exactly happened, Michael? He said, I had a loss in business, and also I have tried. He was a pharmacist. So he knows everything in and out of medication, so I need not have to tell you take that or what you take and what you don't. So he was still very concerned, and he said, Cam, it's really getting bad every day, and it's almost 5 days. I haven't had a proper sleep at all. I'm not getting. I've tried all medications. I've tried yoga, I have tried that meditation and medications and whatnot. What should I do now? You know what was my reply to him? I said, just come to my lecture, within 15 minutes you'll sleep. And I think this is what exactly will happen, and because this is after the post-lunch, so please don't sleep, okay? Well, all right, so I'm not going to take too much of time. I'll quickly wrap up my slides. I'm not going to make you too much bored. But what exactly I do, in my laboratory back in Sydney, I work on 2 areas. I work on immunology and microbiology, and I work on drug delivery. And I combine both of these areas, and with the major focus on the respiratory diseases, which is the airway diseases. I work on 3 main diseases: asthma, COPD, and lung cancer. These are the 3 main diseases I work. Now, in terms of where I'm coming from one of the biggest states in Sydney, Australia, which is the New South Wales, and Sydney is just on the corner. The pointer is not working. Sydney is just on the corner. And I believe everyone knows these 2 beautiful hallmark or the icons of Sydney, the Sydney Opera House and the Sydney Harbour Bridge, where the fireworks is one of the main fireworks around the globe is well known. And the school where I am based at is hardly 15 minutes walk to these 2 icons. So our university campus is just in the center of the city, next to the central station, and these icons are very not far, even from the Darling Harbour. This is a school where I am based at. It's called Graduate School of Health and with the discipline of pharmacy. And I'm also associated with various research centers. This is one of the center called Hunter Medical Research Institute, which is a part of the University of Newcastle and next to the John Hunter Hospital. So a lot of patient biopsies samples we get from there. Now, in terms of my study, before actually I show you some data, let me show you some statistics. This is one of the paper which was published in Lancet, and it clearly shows the burden of the respiratory diseases in country like India. It is prevailing all around the globe. The reality is this. But when you look at this Lancet paper even, the more the red color, the pointer is not working, so the more the red color on the top, the chances and prevalence of the cases of the respiratory diseases and burden is very high. So if you really look, the top-notch part, which is, thank you, the top-notch part, which is the North India, it is having highly prevalent all the respiratory diseases as compared to the lower part of India. And this is not just the, the minimal data which I'm showing you. When we go further, this is one of the paper which was done, the sports study, particularly in India, and they clearly show that 45.4% of the outpatients presenting to the Indian respiratory services, they were having asthma and COPD. And this is not still the story ends. This is another news, which is just in, I think there's no date here, but I can show you, oh yeah, it's here there, 2022. It's in 2022, just last year. COPD is one of the highest hit respiratory diseases in India because there's a lot of smoking. And it's not only just smoking, I mean the cigarette smoking. With the household, the smoke sometime in the rural areas, people still, they use the cow dung and the wood fire to cook the food. The females and the family members indirectly as a passive smoker, they still get the COPD. And these diseases, asthma and COPD, has recently been discovered. It's called overlay, which is called ACOS, asthma COPD overlay. Now, so these are clearly showing that the respiratory diseases burden are quite heavy in India. There's another news, which is also in 2022, that clearly shows that India is almost accounting for 42% of the global asthma deaths. Is this statistics close to 50% reaching is not alarming? Is it not alarming? It is alarming. It is absolutely alarming. And this is nothing. There are so many other news, which I'm not going to read. The kids are also one of the highest level of the category which are hit with asthma. And in Western societies, a lot of kids have a peanut allergy. And when the peanut allergy comes to, it also may further lead to the cascade down to conditions like asthma. So all these things would clearly demonstrate that there is a significant need of research in this area. Being an Indian, no more Indian anymore, but being an Australian Indian, I proudly say, I still focus on the disease state which are highly prevalent in this region, and I highly collaborate with various researchers across India to mitigate such problems. Now, this is another article. This was a bit old when I presented in the Indian Science Congress. I think it was in the same year in 2017 when I came to India to present in Indian Science Congress. Look at that. These all news which I'm not going to read for you, they clearly show that it's very alarming. And we just came out from one of the respiratory diseases. Not exactly we are out of that, we are still coming out, which is COVID-19. So similarly COVID-19, influenza was one of the virus, which in 1918, almost 1/3 of the world population was hit with this. And look at the number of deaths it has caused. It was almost around 500 million people around the globe. And it's not very far what exactly has happened with the COVID across the globe as well. And the estimated death toll is around 50 to 100 million people, and seasonal pandemics which come every year. There are many types of virus, rhinovirus, and influenza. It's just, there are plenty of virus because they change their virulence. And that's why there is no single treatment. And in Australia, which is still a very small country, there are hardly more than 18,000 of the hospital admissions and more than 1,000 deaths per year. But in India, it is almost 43 million episodes of the influenza infection, and that has also led to a high mortality rate. And influenza is nothing, but this is just your common cold. And why there are so many treatments are there, but still there are limitations to it. What are those limitations? The most important limitations is because any virus, it mutates very rapidly. And when it mutates, it changes virulence from year to year. And because of that, we cannot make a single robot vaccines which can kill all the strains of virus. The current vaccines are only effective when the patient, listen carefully, they are only effective when the patient is being presented to the clinic within the first 48 hours of the symptoms. And what we do when we get the common cold in the first 2 days, we try to treat as much as we can with our own remedies. And by the time you've been presented to clinic, the conditions had led to the exacerbated condition. And that's not easy to treat. And the last thing is there are so many antiviral therapies, and particularly people are being heavily relying on steroids. But in any respiratory disease, you go to a clinician, you just tell them that I have asthma, I have COPD. What they will do is they will immediately give you antibiotics. They will put you on steroids. And what these steroids will do, at the end of the day, these steroids will bring steroid insensitivity. It means the steroids also at a certain point of time, they're not going to work. So what we need some new research and therapies. Now what I'm going to show you is the biology part of my laboratory. I'm not showing you drug delivery part of my laboratory. However, I have been working on significant drug delivery with medicinal plants, and we have shown that it has been quite effective in mitigating influenza, asthma, COPD, and lung cancer. Now, as you know, asthma is actually, according to WHO, there are around 300 million people worldwide, they are affected with asthma. 2 million Australians, in around 1 in 10 adults, and 1 in 9 or 10 kids. In Australia, don't worry about that, the stat is really very low, but look at the India. It's almost 18 million asthmatic patients, and which is costing around 2.7 billion Australian dollar. What exactly happened in asthma? It is a chronic inflammatory disease. In a simple way, when you, let me just fast forward it. So what happened? Your airway it's a normal airway. They really quite widen, so the exchange of airs are quite normal. When the asthma, COPD, or lung cancer happen, your epithelial cells, which are the main target cells in your respiratory disease, they change their morphology. And once they change their morphology, they secrete a lot of mucus. And that's called remodeling. They secrete a lot of collagen. And because of that, your airway becomes narrow. And once it becomes narrow, it is very difficult for a patient to breathe. And the story which I'm going to present today is around this. So most of the Th2 cells, like IL-13, IL-4, IL-5, they are the one which majorly plays role in asthma. I think due to the time constraint, I'm only going to show you the asthma story. I'm not touching base on the COPD, which is after this. So there were 3 questions. When a people, or even kids, when they get asthma or COPD and they get flu on top of that, their condition really exacerbate. It's become very difficult for them to breathe. Why? What was the reason behind that? When a people, they are having asthma or COPD or any lung condition, but what I'm showing you is a story of asthma only. So our first question is, why the people with asthma, they are more susceptible to flu? Why they easily get flu? Second, why their condition exacerbate? And can we get that into our mice model? So what we did, let's look. Now, if everyone understands this slide, everyone will understand the whole story. There is one common model which is used across the world. Everyone uses for asthma. Anyone who is doing any study in asthma, they use OVA. Okay, how it works, what we done, we have sensitized the mice in the beginning with OVA intraperitoneally, and then we challenge the mice intranasally, and when we challenge, on the last day, we give infection to the mice. And the 3-day post-infection, we kill the mice. Now let's see what were the results. Now, if you see PR8, PR8 is virus. And if you see OVA plus PR8, it means the mice which is having both allergy and infection. Now you can see, this is exactly the viral titer is the one what you do with your COVID qPCR and any other test. This is called viral titer. How much of the virus load is in your lungs? Look at the mice which is having the virus and the allergy. Their viral load was very high as compared to the infection alone. When we looked at the inflammation, which is the cell count, you can see the last bar, which is the infection plus allergy, they are having quite significant number of the inflammatory cells as compared to allergy, infection, or control. And then what we did looked at the interferon level. Now interferons are your magical moieties. They give you immunity. Look at that. This is the last group, which is the OVA plus PR8, which is the mice with infection and asthma. Their interferon level, both beta and lambda, they are down. They are very down. It means they don't have immunity. And when we looked at the airway hyperresponsiveness, it was too high in the asthma plus flu mice as compared to asthma and flu alone. We looked at the eosinophils. Eosinophils is one of the major cells which are involved in asthma. Anyone can ask you a question simple, which are the cells involved in asthma? Is eosinophils. In COPD with the smoking, you smoke cigarette and you come out and you do your blood count, your neutrophils would be high. So in COPD, neutrophils, in asthma, it's eosinophil. And look at this, the eosinophils number in the OVA plus infection are too high as compared to OVA alone. Means allergy alone was having less as compared to the infection plus allergy. Then we looked at the IL-13 level, because just now we looked that Th2 cytokines play an important role. And that the miraculous thing was the IL-13 levels were very high in the mice, which is having both infection and allergy. So we thought IL-13 is something definitely doing something here. And when we looked at the receptor of IL-13, that also shown the similar trend as IL-13. And we looked at the mucus. So you see, this is the airway, and in the airway, the more the pink color cells, that is mucus. So you can see the lower picture, which is infection plus allergy. It is having a lot of mucus secreting cells, which is through the past staining as compared to the allergy alone. Then what we did, we thought, now this miraculous thing has clearly shown IL-13 is doing something. And I think I'm not the one who discovered IL-13 has been doing something. It has been already reported that IL-13 is well known in allergy, but no one has studied in allergy plus flu. We were the first people, those who have shown, and what I'm showing you is an all unpublished data. The COPD is published with flu, but this is not published yet. So what we did, we thought IL-13 is doing, why don't we just get from the market recombinant IL-13, which is a small model. So we just got recombinant IL-13 straight from the market, and we gave that to the mice. It is such a short model. We gave the recombinant IL-13, and on the day 0, we gave the infection, and 3-day post-infection, we killed the mice. So what should happen? It is only recombinant IL-13. It means we should get all the same features. And we really got all the same features what I showed you in the previous slide. We got increase in viral titer. We also got increase in the inflammatory cell count. We also got reduced level of the interferon beta and lambda. We also got increased in the airway hyperresponsiveness. We also got increased level of the eosinophils. We also got IL-13 and IL-13 alpha level receptor, both were up through ELISA, and we also found that the mucus were high in the mice, which is having recombinant IL-13 and flu. That is very clear that IL-13 is playing role. So what should we do now? So we approached our collaborators in United Kingdom, and we gone on to the house dust mite. House dust mite is one of the more clinically relevant model. What in humans we get asthma is because of the house dust mite, not of ova. In the mice model, if we have to create asthma, we usually use ova because it's quick. So we thought, why not before we try anything else for IL-13, we can just check on a house dust mite. So we got this from Imperial College London, our collaborator, 2 things, which I'm showing you in the next slide, and also in this slide. So we got house dust mite, and we used the same model, what we did in ova. What we did, we sensitized the mice, we challenged the mice, and with 3-day post-infection, we actually culled the mice. I'm showing you half a data because half a data we're still analyzing. We got increased viral titer, which is exactly same resembling with the previous slides. We also got increased airway hyperresponsiveness. We have just finished this endpoint, so we have to analyze a lot of data. We also found increased eosinophils, and we also found increase in mucus. We have to still run IL-13 and IL-13 alpha, ELISA, and interferons, because we have just finished these studies. Then, because this one is still been analyzing, we thought in parallel we ran anti-IL-13. So this also we got from our collaborators, and we used the same old OVA model, and we gave the anti-IL-13 to the mice. So when we gave the anti-IL-13, with the same model we used, we sensitized them, we challenged them. On the last day of the challenge, we actually started giving the infection. And 3-day post-infection, we culled the mice. And when we culled the mice, we also found increased viral titer. We also found increase in airway hyperresponsiveness. We also found increase in eosinophils and also in the mucus. Rest of the data we still analyzing. So the story was very clear that IL-13 has really been playing a significant role in asthma with IL-13. Then we further cascaded down to the microRNA level. and because we were already having a published literature, which I'll show you quickly in the next slides, in a running show in COPD, we really looked at the microRNA 21. So when we ran microRNA 21, we clearly see the microRNA level were quite high, with the mice having recombinant IL-13 and infection as compared to the other groups. And same with the microRNA 21, when we started giving them antagonists, it started reducing. Now what we are currently doing, we are actually encapsulating these inhibitors into nanoparticles, so that we can give it to mice to see whether the controlled effect can be achieved in terms of reducing the severity or the exacerbation of the asthma with COPD. So now to conclude this study, what exactly happened and what we're trying to show is, any patient with asthma, they have increase in the influx of the IL-13. We have also done facts, and I also have a facts data proving IL-13 is quite high. And then that can increase the levels of BioMe'21 in epithelial cells, and down the track it is been regulating through the PR3 kinase pathway and leading to the exacerbation of the disease. Now, this is asthma. I have only 2 minutes left, or 3 minutes I think. So I will quickly show you in COPD. In COPD also what we have done, I'm not going through the rates. In India it's almost 22 million people having COPD, and what it happened is your airways, which is just like a web-like structure, it start breaking. So once it start breaking, you cannot actually have a common influx of you cannot breathe normally. And the similar study, we have also shown that in COPD, when the patient gets flu or infection, which is PR8, there is an increase in inflammation. And that is also having reduction in the antiviral responses, and that is because of microRNA 125A and B. And that is increased due to the level of NF-kappa B and A20, and that leads to the inflammation. And also we have shown through the MAV signaling. Now this study is done in both animals and humans. So thanks to Peter Walk, who is our clinician collaborator from the John Hunter Hospital. And this study, actually, I'm not running, I'm just going to the conclusion slide, which is done in both humans and mice. So when we treated our COPD PBEX, which is the human cells obtained from the patients of COPD, the smoking and non-smoking, and we treat our flu, what we have seen that there is an increase in level of microRNA 125A and B. And that is because the A20 level goes down, and that increased the level of NF-kappa B. And also with the reduction of the mitochondrial antiviral signaling. And that actually leads to the influx of the inflammatory cytokines and leading to the exacerbation of the disease. But when we are giving inhibitors, it actually reversed the whole cycle and identified the new therapeutic moieties. So that's the whole kind of the study what we do in my laboratory. And as Amit has also been getting a couple of students, from here, I'm more than happy to also collaborate and see if some students can come and do a bit of research with me as well there. I would like to acknowledge some people before I end my presentation. I would like to acknowledge especially Phil, Malcolm, and Alan. They were quite a core part in this, and also running significant projects with other diseases as well, like pulmonary fibrosis, because we got a funding from NHMRC, which is the National Health Medical Research Council. And to UTS, Charlie, who was the founding dean of the Graduate School of Health, Kylie, my immediate head of the discipline of pharmacy, and Mary, my another collaborator. And also to the organizing committee, who has really invited me and giving me the best hospitality and been experiencing really very high. And I can see that you guys as a young scientist have a lot to do. And what exactly the other speakers been saying from the morning, and what Amit was saying even, is just keep striving. As you keep striving, you'll definitely attain your goal. In particularly with, oops, that's just the preview. That's the extra slide. So I would also like to thank Dr. Pavni and also Dr. Mamidala, who've been in touch with me for such a long time, along with Amit. Thanks to him as well. And so that has allowed me to fly all the way from Sydney to here to be with you guys. But I'll be around tomorrow, as I think, yeah, I'll be around. I don't know about tomorrow, but if you need me, you can easily found through my name, my email ID, and feel free to contact me. I would be more than happy to answer your queries. I know it was a bit of a rushy talk. In Australia, before I end this, we have, I'm sure I don't know whether in other countries, in states you have or not. We call it as a 3-minute thesis, 3MT. It's a 3-minute thesis means you have to present your thesis in 3 minutes. What the student normally do, they come up with one slide with no animations, and they present their whole thesis in 3 minutes. And what I did, my presentation in 20 minutes. So thank you so much, everyone, again. Thank you. Thanks for being patient.
Yeah. Thank you so much, Dr. Dura. So now we can take few questions. I think there's a time for 2 to 3 questions from students. Don't feel shy. You can ask very basic question. It's a learning session for you. So anything from what is asthma to COPD and what is the role of 125, MEAT 125. The floor is open for questions, and you can ask anything that comes to your mind.
Yeah, it's a very good question. So I don't directly work on allergies, but so we have a big center called Priority Health Research Center for Lung Health. So there's another person called Paul Foster. He all works on the peanut allergies and other food allergies, particularly lactose. So what they have shown is there are certain other microRNAs, such as microRNA 234, and also they have also shown, I think, through the certain interleukins which have been gone up with the allergies in those diseases, but I don't directly work on any kind of allergies, sorry. Yeah, so MEAT 125A and B, actually these kind of microRNAs, they are very well known more in cancer, as compared to the inflammation. Inflammation area has started slowly evolving over the time from cancer to inflammation. So MEAT 125A and B, we have published already. You can easily Google with my name that paper. It's in a very, very good journal. And what this particularly microRNA do is, these microRNAs actually directly trigger your influx of the cytokines. And cytokines are the main moieties which actually brings inflammation in any disease state. It doesn't matter, I'm saying only in the lung diseases or anything. It can be a cancer, or it can be any kind of the disease, any other disease. When there is an influx of cytokines, it is always called a cytokine storm, and that particularly leads to the inflammation. So microRNA 125A and B, when their levels are up, automatically cytokine levels are up. So when we give inhibitors of those microRNAs, this is what currently one of my PhD students doing with UNSW, we are preparing dextran-based nanoparticles. Now, why we are preparing dextran-based? Macrophages are one of the cells which actually are always increased in levels when any disease happen. And macrophages always love sugar-based things to attract. So what we're doing, we are encapsulating our inhibitors, microRNA 125A and B, because we know they go up. So what we're doing, we are just been encapsulating in dextran-based nanoparticles. We have done already that study. One of our paper is just accepted in Journal of Drug Delivery, Science and Technology of that. It's about in a galley proof at the moment. So we encapsulated in a dextran-based nanoparticles, and we treated raw cells, which is most of the macrophages 264. And what we can clearly see is there is a very reduced level of the microRNA 125A and B, and a very reduced level of these cytokines. I can show you, if I go back to my slides, I can show you quickly with one slide.
Okay. While he's pulling up, there's one more question that has come up.
Okay, can I get that? Yes. Sorry? IL-17, look, I'm showing you. In COPD, this is what we have done. No, IL-17 is not a part of a Th2, I think. So we have looked definitely in COPD, but it has not shown, sorry? Yes, I agree with you.
It has a direct regulation with IL-17. So
correct.
IL-17 regulates IL-13.
Because the only focus of our study was on IL-13 and MEAT-21, so we never jumped onto the other cytokines to make the access more complicated. But I agree with you, IL-17 actually regulate 21. But in COPD, the statistics was not really showing any changes in IL-17. Look, what we looked at, IL-6, CXCL8, TNF-alpha, IL-1 beta, IFN beta 1, IFN lambda 1. These were the main what we looked. But this was being done in the humans, but I can show you what we done in the mice. So we also got a very unique mice model, which is the nose only. And what we have shown is, in mice, it was only showing the differences of IL-6, CXCL8, TNF-alpha, and IL-1 beta. It was rest all the cytokines, they were down-regulated. So that's not captured.
All right, thank you so much. I think
No
we are
problem.
running out of time. So let's have a round of applause for Dr. Deva.
Thank you.
Thank you so much.
Thank you, Amit.
Just hold on for a second. We will have a felicitation, I think.
Thanks, everyone.
Thank you, sir, for the elaborate insight into the respiratory disorders and novel interventions. Thank you, Professor Amit Kitiwari, for chairing the session. I would now request Additional Secretary Hanmant Nayak Sir Garu and Professor Amit Kitiwari to felicitate our speaker. Moving on, we have an interesting session by Dr. F. Scott Hall. I would now request Ms. Kalajyoti, my colleague from Department of Microbiology, to introduce our speaker.
I now introduce our next speaker, F. Scott Hall, an Associate Professor of Pharmacology and Experimental Therapeutics in the College of Pharmacy and Pharmaceutical Sciences at the University of Toledo. Dr. Hall has researched alcoholism and drug addiction models for 20 years at the National Institute on Alcoholism and Alcohol Abuse and the National Institute on Drug Abuse. His work majorly focused on genetic contributions to addiction using genetically modified mouse models, assessing the lethal and toxic effects of synthetic cathinones, among others. Dr. Scott holds many awards, grants, and honors, such as the International Behavioral Neuroscience Society recognition for outstanding contributions and dedicated service as IBNS President, elected President of the International Behavioral Neuroscience Society, elected to membership to the Sigma Phi Sigma Pharmaceutical Sciences Honor Society, to name a few. He is also the editorial board member to Advances in Drug and Alcohol Abuse, associate editor to Brain Sciences and Behavioral Pharmacology. He has authored and co-authored many research and review articles in reputed journals. His most recent book was Negative Affect States and Cognitive Impairments in Nicotine Dependence, published by Elsevier in 2016. With this brief introduction, I welcome Professor Scott Hall and Professor H.S. Poddu onto the dais, please.
I request Hanuman Sagar Buddhu sir to chair the session.
Good afternoon, everyone. I would like to thank the organizers and also all of the students who did so much work to put this together, staying up late into the night to make everything look beautiful. I'm going to talk about something quite a bit different. In my career, I have studied different models of psychiatric disorders and also focused on drug addiction. And part of the reason for that is that everyone who has an addiction problem also has some other psychiatric problem. There are reasons that people take drugs of abuse. They may have suffered trauma, experience anxiety or depression, and for these reasons they take those drugs. But unlike most people who can take drugs, drink alcohol, smoke, other things, they cannot control that intake over time. Now, in the last few years, my research is focused on a slightly different area, and that is drug overdose. So when drug intake becomes uncontrolled or sometimes just accidentally, people overdose from different types of drugs. And I'm going to focus on some changes that have happened in my research over the last several years. For most of my career, my research subjects were rodents, a very typical research subject for pharmacological studies and drug abuse studies. But we've started using a different model organism, in part to accelerate our research. And I will talk about the reasons for that a little bit as well, hopefully. But to begin with, I'm going to talk about what we refer to as the drug overdose epidemic in the United States. So this is a graph over time of the number of yearly deaths from drug overdoses in the United States. It is broken up into parts, and you can see how they're sort of, you can get this to work, not so much. There have been increases over time and certain inflection points where the number of drug overdoses in the United States went up substantially. This began in the 1990s when a lot of prescription opioids started to be used, and then as the regulatory authorities clamped down on the use of those prescription opioids, heroin started to be used, and then finally fentanyl. And the lower portion of the graph that sees the large increase over the last few years is related to fentanyl overdoses. So fentanyl is an opioid, but it's a different type of opioid from the others. It is very, very potent. It is 500 times more potent than heroin. And for that reason, tends to have a greater contribution to overdoses overall. But the reason we refer to this as an epidemic is because of the very large number of deaths that have been associated with it. So last year, in 2021, there were over 100,000 deaths from overdose in the United States from illicit drugs. So it has become a very, very large problem. It's by no means a new one, but it has become a larger problem. And part of the reason it got so bad was because of the high potency drugs that people started to use illicitly, in particular fentanyl. However, another thing has happened. If it'll come back, there we go. At the same time, hidden within that data, over the last few years, there have been large increases in the number of stimulant drug overdoses. So stimulant drugs have different types of effects. It's in the name, they activate you. So most of us use mild stimulants. So when we drink tea or we drink coffee, they're minor stimulants, they wake us up. There are other stimulants that people use under more extreme conditions. So these include especially methamphetamine and cocaine. They have been used for a long time and have been major drug abuse problems for a long time, including overdose. However, in the last few years, the number of overdoses in these have increased dramatically in the United States. In particular, amphetamine-like drugs. Now, one of the interesting things about this is over the last 20 years, there's been a change, and part of it's due to the internet and the free availability of information. anybody can look up how to synthesize different types of drugs. So this includes novel drugs, what we call novel psychoactive substances. And these novel psychoactive substances, we don't know what their properties are yet. We're still just researching them. And we're not certain whether they might have greater effects than these other drugs, including greater toxic effects. And that's where my research really started a few years ago, because nobody was really studying these drugs and whether they posed a greater risk than other drugs that have been used for a long time, like methamphetamine. So here are the structures of methamphetamine and amphetamine. Amphetamine can be abused as well. People probably know it more commonly from the drug Adderall, which is used to treat ADHD. Methamphetamine is the most commonly abused amphetamine. A structurally related drug is cathinone, and it actually differs from amphetamine only by the addition of that oxygen. And traditionally it had been used in many parts of the world, in parts of the Arabian Peninsula and northeastern Africa, traditionally, and it would be used in typical ways. You would chew it, you would have it in a tea. The effects tended to be mild. What happened when that drug made its way to Europe and the United States is we isolated the active ingredient, made it into a powder, and took it in other ways, and it started to produce abuse problems overall. Now, I've been talking about the United States. One of your questions might be, well, does this really apply to us here in India? And I'm afraid that we have drug abuse problems everywhere. There's no country that really escapes it. The particular drugs that are a problem might be different, but no country escapes it, including India. So last night I looked through the internet a little bit to identify just some examples. So this is from Times of India a few years ago, saying that manufacturing centers in India were exporting a large number of these cathinones to Europe. And earlier somebody was saying, in particular in Hyderabad, about it being one of the centers for big pharma in India. And part of what this means, it has the capacity to make a lot of different drugs, and it was making these drugs as well too. This is the seizure of ephedrine from a few years ago, 21,000 kilograms. Ephedrine is the primary precursor used to make MDMA, a commonly abused amphetamine, usually referred to as ecstasy. Here's just a statement that there was an increase in the popularity of synthetic drugs within India. One of the things that happens when there's a large supply of drugs, even if they're originally made for export, they come to be abused in the local area as well too. So, in fact, the seizure of amphetamine-type stimulants between 2012 and 2016 increased from 40 kilograms to around 1,600. There was a large seizure of cat leaves, which is one of the precursors of cathinones. And here there was a seizure of the cathinone called mephedrone from 2 labs in Hyderabad of about 24 kilograms, which is several thousand doses of the drug. So there is, again, the chemical structures of methamphetamine, methcathinone, the cathinone version of methamphetamine, and mephedrone that you can see is also quite similar. So for a long time throughout the world, most of these new novel psychoactive substances were in fact legal because nobody had seen them before, so nobody had made them illegal. And in fact, they were sold in packages like these in stores, in gas stations, convenience stores, until just a few years ago. Over the last 10 years, most countries in the world have outlawed them overall. But the questions that I became interested in centered on stories like this in the popular press about all of the horrible things that happened when you took bath salts. And I wondered whether the press was exaggerating the risk here. We don't really know. That's the core problem. It reminded me of a lot of government propaganda from the 1950s in the United States surrounding marijuana about how horrible it was. And basically most of that was made up as well. However, when you look at clinical studies of overdose from cathinone-like drugs, these amphetamine-type-like stimulants, they have clinical effects that are very similar to other stimulant drugs, so methamphetamine and cocaine, when you take too much of them. This can include death overall. And in addition, a lot of people take these drugs without knowing it. They are sold these drugs in place of other drugs like ecstasy or methamphetamine. So this is a study from New York where they went to clubs where people were using ecstasy and asked them if they'd used ecstasy in the last 3 months and if they could take a little bit of their hair. And they took their hair and tested it for various cathinones and found that these people who thought they had been taking another drug, in fact, had been taking a lot of these cathinones. So for a lot of novel psychoactive substances, one of the problems is a lot of people may be taking them and not even knowing it. So we started to study this in the classical way. I'm going to run through the data quite quickly, and hopefully somebody's keeping track of time for me. Just give me the message when I should be getting up. So we started studying it in mice. This is my student Don's work. And first of all, we showed something very different from other amphetamines that actually lowered temperature rather than increasing it. And then we started to see that they really weren't very toxic. These graphs show the amount of lethality with dose. And you can see that it was not actually killing very many of the mice overall. There was some evidence of organ toxicity, but these drugs simply did not appear to be very dangerous. So why? Well, there could be a lot of explanations for that. It could be that there are other ones that are in fact dangerous. It could be that it takes more than one dose. It could be that it involves interactions with other drugs. Because people take these drugs in social circumstances, they're very often consuming other things. They are smoking, they are drinking, they're doing other things. So that may actually contribute to the toxicity. And then finally, some individuals may be more sensitive than others. That's always a question. And another thing here at the bottom, because people tend to take them under circumstances in which there are social circumstances, they very often take them at clubs or at concerts. So these tend to be places where the ambient temperatures are very, very high, and this is known to contribute to the lethality of amphetamines already. So one of the next things we studied is, again, we did in mice, and we in fact showed that that was in part true. When you increase the temperature, so the HT conditions here are the high temperature conditions, all of a sudden doses that were not lethal are lethal. So I'm just going to skip through this quickly. So this is just some of the evidence from mice showing that this might be a problem. Now, what I really wanted to talk today about was zebrafish studies, because one of our problems was there's so many of these cathinones. There are several hundreds of them that have come into use, and different variants of the original molecule that we can't keep up with them to study them to find out if they're truly dangerous or not. So we decided to use zebrafish as an experimental system because we thought the research would go more quickly. Thank you. So we also wanted to study more closely related molecules in order to develop structure-activity relationships. Altering some parts of molecules has huge effects and really changes the way they act and how potent the drugs are. Changing other parts of the molecule makes no difference. So what we really want to do is understand which parts of the molecule contribute to their actions. So we were studying all of these closely related molecules that are similar to MDMA. So these are just our first observations in zebrafish. We studied with methamphetamine, and we showed that it was in fact lethal, but not at a relatively high concentration. So, and then we showed that they have stimulant-like effects, but also at a certain point, instead of stimulating the heart, they inhibit it, and they appear to damage it. And that may be actually the more important part of why they're actually dangerous drugs. Again, I'm going to skip through that quickly. There's also some evidence of liver damage, And when we looked across a range of drugs, they differed in their ability to produce these effects. And we have the beginnings of a structure-activity relationship here as well. So these are just the same sort of effects upon heart rate, where at low doses, at early time points, you get elevations. At later time points, at higher doses, you get the opposite effects. So let's see if I can get this to work. We were interested in this cardiac toxicity. I'm hoping my video will work, but it doesn't seem to be. That's a shame. It's really cool. So one of the reasons we went to the zebrafish as a model system is that when they're young, we're using zebrafish larvae. They're 5 days old. First of all, we can do it in a 96-well plate, so it's very high throughput. But we can also see through the animals. We can see the internal organs. So we could literally see the heartbeat in these animals and measure it visually. And when we gave them the cathinones, so here's a normal heart rate, and again, fortunately it doesn't seem like I can play the video, but we start to get very abnormal heart rates with the others. So cardiac toxicity seems to be really important for these drugs in the zebrafish like it is in mammals. We also showed that when you lower the temperature or increase the temperature, you make the drugs much more lethal. So this is a whole range of these cathinones and the increased lethality. Now the last thing I'll throw at you is something that's relatively new. Like I said, when people take these drugs, they tend to take them with other things, including alcohol, which universally makes these drugs worse. So these are measures of toxicity for ecstasy and methadone plus ethanol. So there are shifts in all of these curves to the left. They become more toxic. But then when we looked at nicotine, we got a very different type of effect. So nicotine protected against the dangerous effects of these drugs. So this raises the possibility that we could develop a drug to treat these types of overdoses using a nicotine-like drug that works through acetylcholine receptors. All right, so this is a summary of this overall, including the last two main findings. The main thing here is the zebrafish really worked as a model system to study these types of drugs. It was not something that, it was something that a lot of people really doubted because they are so different from us. But one of the mistakes we often make, particularly scientists, is we look at the differences between things rather than the similarities. And there are far more similarities across the whole range of animal species than there are differences, which means that we can use these lower level organisms to really study these types of things very effectively. So what we need to do in the future is really start studying this wide range of drugs that people do abuse to get a better understanding of what threats they present, and then also try to look for a treatment. So when somebody has an overdose from an opioid, we know what to do. They're antagonists of opioid receptors, they will treat that. There are no equivalent treatments for stimulant drug overdoses. All we can do is treat the side effects. We cannot treat the underlying toxicity. So it is, in fact, a major need within the field overall. So I would like to thank all of the people involved, especially Don, Sydney, and Logan, who are still in my lab, who did parts of this research, but especially Yu Chen, who did a large portion of the work. He's now a postdoctoral fellow in Tennessee. And my collaborators at the University of Toledo, especially Isaac Schaefer and his laboratory that synthesized all the drugs for me. And the grant funding were funded by the National Institute of Drug Abuse and the University of Toledo. And that was very quick, but thank you very much, and I'll be happy to talk to you about any issues in addiction or overdose.
Thank you, Dr. Scott. Now the floor is open to questions. Audience? I know the talk is kind of, Hello. On a little higher level, but still any basic questions related to the talk would be, welcome.
Hi, sir. This is Aparna. I have a question. Nicotine is itself a dangerous addictive chemical, and how come it is protecting the MDMA?
That is a really good question. First of all, nicotine is not dangerous. Nicotine's fantastic. It relieves anxiety and stress, it improves memory and attention. nicotine actually is not a problem. Tobacco is a problem overall. Or vaping, the things that you do in vaping introduce poisons into the body as well too. It's all the other stuff that's associated with which is really bad. Nicotine itself really isn't too bad. Now nicotine is the addictive component, and that's an issue as well too. In terms of why it might be protective, we don't really know, but ironically, there is some evidence that nicotine may reduce the production of reactive oxygen species. So it may reduce inflammation, which is interesting because generally when people think about smoking, they think about it being pro-inflammatory, which is true, but it's not the nicotine, it's all the other stuff in tobacco that produces those inflammatory effects. Or even from vaping, some of the other ingredients that are produced in the process of vaping produce inflammatory effects as well too. But the nicotine itself might have some of the opposite effects, and that may be why it's protected, but the real answer is we don't know. That's as much as we know is the graph that I showed you there. It was very surprising, but Amit was talking about, Dr. Tiwari was talking about this earlier, and his student brought him something surprising. And the part he left out of that story is he really didn't believe it. So he had to replicate it like 3 times, which is what happens when you get a surprising result. And my student, the same thing happened. He brought me the first data on nicotine. I said, do it again. He got the same result. And then he did methadone. He got the same result with that. And he replicated it a second time just because he thought I would doubt it. So now we've seen it 4 times. So I'm sure it's true, but I don't know why.
Thank you. Other questions?
Thank you. Thank you so much for a lovely talk. I know you expressed a lot of benefits of zebrafish models, but are there any limitations that you think using this model?
We're still trying to evaluate that. So one of the ways you think about a model, because remember, we don't care about zebrafish. We don't really care about mice. We care about people. We use these model systems because we can't do experiments in people that get us the same information. So the big question is, are they valid or not? Do they give us results that translate to humans? So part of our ongoing work is to validate that. One of the reasons we did all the temperature studies is because that is something that you see in people. Higher ambient temperatures make the drug more toxic. So the fact that we saw it in the fish validates the model to a certain extent. One of the things that we're trying to do currently is to evaluate LD50 values in humans from clinical studies, from mice, from our own mouse studies, and in the zebrafish, and they should all predict each other. They should all be correlated. So that will help validate the model. But there are always points where it might differ. The mechanism of toxicity could be different in a different species. So that's another way, are the details of the toxicity the same? And that gets into all the aspects of mechanism that Dr. Tiwari was talking about in his studies for the different ways that toxins can kill cells or kill organisms. There are different ways. You want the way to be the same. And then it's true, it's valid.
One last question.
Scott, so what about this FDA? They recognize this zebrafish toxicity data?
Sorry about that? Oh, FDA?
FDA, yeah.
No.
Okay. So,
but the thing is, it's not necessarily something you would present to the FDA. One of the things in drug development is that very often you do things early in testing as part of the development process, not just to go to the FDA. So if you wanted to go to the FDA, Food and Drug Administration in the United States for drug approval, or the equivalent agency in India or Europe or any place else, you would have to have data from higher species before you go there. However, in the drug development process, if you want to, one of the things you want to decide is, do I want to move forward with this drug mechanism? Do I want to move forward with this new class of drugs? If the drug has any kind of toxicity, you stop right there. So it's very valuable information early on in the process, even if it's not something you would present before the FDA.
Yeah, so you require a mice as a preclinical data.
Yes. So if you're going to the FDA, you would have to go into mammals. And that gets to Dr. Tawari's question as well, too, about validity.
Yeah, so one of the questions from some of the audience, totally don't understand the question, but in market, most of the addiction killers are available. Are they helpful to drug alcohol abuse persons, any side effect? I think what they're trying to ask is there any addiction, is there drugs that are available there to treat addiction?
Oh, to treat addiction.
And if people with alcohol abuse, people who use alcohol a lot, and they use them for the treatment, what would be the side effect?
Yeah.
With your drugs?
So for addiction treatments, pharmacological treatments, it depends upon the drug. Very common, what we use are what we call replacement therapies. So it's not the exact drug the people were abusing, it's a slightly different drug. So for opioids, we have different types of opioids we can use as replacements that people take instead, and it reduces their craving for the other ones. For nicotine, it's the same kind of thing, where you can provide the person with nicotine, but they're not smoking. As I was saying, most of the bad effects actually come from the smoking. So you can have a nicotine lozenge or a patch. But what we really want is something that treats the drug cravings themselves. One of the things that happens when people develop an addiction is they have this intense desire to take the drug. And what you want to do is block that. So there are some treatments to do that, but not for all classes of drugs. There are no approved treatments for stimulant drug addictions at all. That's a huge need overall. And one of the things to remember is that it's not just about the addiction itself, but it's about whatever was driving the addiction to begin with. So part of what you have to understand, and for the individual person you're trying to treat, is what were their problems to begin with? What problems do they have besides the addiction? Do they have a history of trauma and you need to treat their PTSD? Do they have anxiety or depression and you need to treat that? Do they have a sleep disorder? There can be a large number of other problems, and if you treat those, very often you find that the addiction may still be there, but it might not be as bad. So treating the whole person, I guess is the way to put it, is an important part of that.
Thank you, Dr. Scott. I think we are done with the questions.
All right.
Thank you, sir, for sharing your enchanting knowledge with us. I would now request Jagadishwar Reddy Garu, OST Tribal Welfare Institutions, and Professor Hanuman Sagar Buddu to felicitate our speaker. I extend my gratitude to Hanuman Sagar, sir, for chairing the session. In a couple of minutes, we'll start with another session by Dr. Nuruddin Khan. I request all the participants to please be seated. The session will commence shortly. In the next coming sessions, we have a career counseling session by Darshana Joshi. I request the volunteers at the exit door to please stop the participants from leaving the hall. The tea break will be given shortly. May I now request Ms. Kalajyoti ma'am to introduce our next speaker, Dr. Nuruddin Khan.
Thank you, ma'am. I take great delight in introducing our next speaker, Dr. Nuruddin Khan, an associate professor from the Department of Animal Biology, School of Life Sciences, University of Hyderabad. He is also a courtesy assistant professor at the Department of Microbiology and Immunology at the College of Veterinary Medicine, Cornell University, USA. Dr. Khan has numerous ongoing projects funded by DBT, BIRAC, ICMR, BINS Bioproducts Limited, and University of Hyderabad. He has authored and co-authored numerous research papers and review articles in prestigious journals, including Nature and Science. Dr. Khan has delivered many invited lectures, oral and poster presentations in national and international symposia and conferences. Dr. Khan is a member of many professional bodies such as the Indian Immunological Society, Molecular Immunology Forum, Indian Science Congress Association, and Asian Council for Science Editors. He has been bestowed with prestigious awards such as the University Chancellor's Award for Excellence in Teaching and Research, Shakuntala Amir Chand Prize for Clinical Research, and Chaturvedi Ghanshyam Das Jayagopal Memorial Award for Outstanding Research in Immunology by ICMR, and many others. His areas of specialization include molecular immunology, infectious and metabolic diseases, and vaccine and adjuvant development. With this brief introduction, I welcome Dr. Nuruddin Khan to please deliver his lecture, and I request Professor Scott Hall to chair the session.
Thank you, Kala. Am I audible, sir? Okay. Yeah, thank you, Kala, for a kind introduction. And I'm fortunate that she's my student and she has to yet complete my PhD. So she gave my introduction. Okay, so first of all, I would like to thank Dr. Pawnee and the organizers for inviting me to deliver this very important topic. But my lab at University of Hyderabad, since last almost 12 years, we are trying to understand how immunological cells, they senses various biological perturbations like infection, inflammation, and metabolic alterations. And the basic knowledge what we gain from that we are trying to extrapolate for the development of vaccines and therapeutics. And I will give you a case study how our basic science, how our basic information have led to development of first Indianized drug, which is under Atmanirbhar initiative, which I will be sharing some of the data. That drug, we have finished phase 2 clinical trial, and we are waiting for DGCA approval. So keeping in this mind, I had chosen this very important. This is not working, can I? It's not working. Oh, this, okay. Let me, okay, fine. Yes, okay. So this, I took this topic, which is very important in today's world, the vaccinology in COVID era. And by this cartoon, you can, it's not working somehow. Yeah, so this cartoon, if you see the importance of vaccine in our life. So whenever you have any pandemics or infectious outbreak, the first question we ask, do we have any vaccine or not? And we all have experienced that during COVID-19, when we all were in a house arrest, the first thing which came in mind, when the vaccine will come? And we know throughout how big queues were there for taking a COVID vaccine or Covishield. Even I used to get a call from big short people, can you arrange a vaccine for not? So this cartoon really emphasizes this, it's pointers. Okay, this is very dim actually. Okay, anyways, yeah, so I'll try to take you very fast because I know this is afternoon talk and most of people are in sleep mode, but I will keep you energized by showing this type of cartoons, okay? So see the cartoon, don't see see the contents, okay? So you'll get energized. It's a very good cartoon. So this explains why a vaccine is important. And the vaccines has been one of the greatest strengths of modern medicine, and it is used to save millions of lives. And if you see the vaccine industry, it is growing at a phenomenal rate, and by 2026, it is estimated to go to around $100 billion market, and all this credit goes to the discovery of vaccine this credit goes to this giant person, Edward Jenner. So you know that in 70s, 80th century, that the big problem of smallpox. So during that time, the smallpox was considered to be a very dreadful disease, and it leads to a loss of mortality as well as scars, and so many problems were there. So the giant discovery of Edward Jenner and Louis Pasteur made a smallpox. They laid down first laid down the basic of the vaccinology, and that vaccinology also led to the understanding of immunology. So vaccine and immunology, they have evolved together, and that's how, if you see today, as a child is born, we have n number of different type of vaccines available. And before the child is born, still the pumping, so many women, the mothers, they are pumped with so many different vaccines. So this is the immunization schedule we have as a child is born, and we have a multiple different type of Can you arrange this? It's not working, please. So this is not working actually. Your pointer is not working. This light, this laser. Light is very less, yeah. The advent of so many vaccines is there in the market, and from the child, for the first day on this earth, the series of vaccination immunization schedule, a child is pumped up. Okay, so today, if you see many of the vaccines available in the market, either they are different type of vaccines. We have a viral-based vectors, virus-based vaccines are available, which are either inactivated. We have a killed or weakened live attenuated vaccines. We have a viral vector-based vaccines, which is replicating, non-replicating. And then we have a DNA, we have nucleic acid-based vaccines are available. we have subunit vaccine that is a protein or peptide or virus-like particles. So I will not focus too much on vaccines, and I will just share some of the basic discovery which came from my lab and how we have extrapolated for other adjuvant therapies. So the basic problem today, what we have, so we know that many of the vaccines that were developed was empirically developed. If you see the Edward Jenner vaccine, it was developed with what we call as the astute observation, which was combined with the serendipity, and that leads to the development of a smallpox vaccine. But many of these vaccines, whatever live attenuated we had, the mechanism was not known, okay? And with the proper mechanism, we didn't have. So basically what happens, most of these traditional vaccines, whether it's a live attenuated, killed, inactivated, they have certain disadvantages associated. So that's why new generation or next generation type of vaccine, which is coming up, we call it subunit vaccines. And the challenge, one day we are thinking that, okay, a child does not have to go through so many series of vaccination program, and they can have only one vaccine. And that how it can be achieved is through development of multivalent subunit vaccines. But multivalent subunit vaccines, if you see, they show a poor immunogenicity. So when they come to clinics, many of them failed. Why? Because of poor immunogenic responses, weak B and T cell responses, and they require a lot of boosters and immunostimulators. So in immunology, we call today a 3A challenge. Okay, so what is the 3A challenge is, so we know that vaccine has two important components, which call as antigens, and other is the adjuvants. So we have to play around this antigen and adjuvants. And there comes a 3A challenge. It's not an ice bucket challenge. Likewise, we have a 3A challenge that includes, first is the antigen selection, what type of antigen we are going to use. And once we have selected antigens, how we are going to deliver to the immune cells. And once we have delivered the immune cells, whether they are going to recognize, they are going to respond or not. So we require a proper antigen delivery, antigen selection, and adjuvanting system. So basically, our lab is totally focusing on 3A challenge, and I will be sharing some of the data to show how we have yielded. So long-term goal of our lab is to cut down number of syringes, and we hope that long-term we'll be able to have one vaccine that is targeting multiple diseases. So how this can be achieved? So basically we started in our lab, this is a platform technology we use. One is that we identify through reverse vaccinology approach. So we know that many of the genomic sequences of many of the pathogens are available. So you can, like you have a 28 alphabets, you can take one alphabet, you put into a proper expression system, put in a delivery system, use in a right combination, and use as a vaccine formulation. So this IDBI approach is not a bank, it is our vaccine discovery. IDBI, it's a bank, well-known bank, but this is our vaccine discovery approach we use, and this is to make a new next generation vaccine formulations. So today what I'm going to focus, I'm not going to talk a lot about the upper antigen selection and antigen delivery system, what we have been focusing, but I will be talking about another third A, that is a adjuvanting part, and how we have been able to develop a new adjuvant. So how this reverse vaccinology approach we are continuously using. So as I told you that most of the potential, the vaccine, the genome sequence of many of the pathogens are available. We can take one of these alphabets, we clone it into expression system, put into a proper delivery system like nanoformulation, put in mice, and then we do all preclinical, clinical assessments, and then we take into clinical trials for human studies. So this is the long-term approach we follow for developing a particular vaccines. So because we cannot use this, if you take a recombinant protein and you put into humans, what happens, there are a lot of proteases in our body will chop into small, and they make it less immunogenic. And so to make into a strong immunogen, what you have to encapsulate into a proper delivery system. And there comes role of nanotechnologies. So we have a collaboration. We have a multidisciplinary collaborators across the globe. We have a chemist, we have nanotechnologists, and with those we decide a lot of formulations. I will be keeping, I'll just, what type of formulation we are using in a lab is a PLJ nanoparticle-based formulation, which is a chitosane-based formulation for making oral vaccines. Apart from that, we have a lot of metal-based, and in metal-based, we use zinc nanoxide, because zinc is, we know that many of us, we take zinc of it as supplements. Apart from this, we use a silica nanocapsules, and then we have engineered one of bacteria, and this bacteria can display our desired antigen on its surface, and that we call as outer membrane vesicles or exosomes. And this we are using to target for poultry for development of poultry vaccines. And you know that poultry is a big challenge because many of the AMR, antibacterial resistance genes or the strains which are coming is from the poultry industry. So they are responsible for many of the AMR strains. And that's how we have been heavily funded by DBT to make a vaccine against poultry pathogens. And we are using this engineered outer membrane vesicles, which is from the probiotic bacteria. And this bacteria component cut down the requirement of adjuvants, and this is coming to be very effective formulations. So, so these are some of the publications we have gone through. I'm not going to discuss about, I told you, antigen selection and delivery system. So these are the couple of papers we have reflects our research work, and these have been published in the various reputed journals. But the third part, what we have been discovering, so I'll directly go to the adjuvant parts. And why adjuvant? Because adjuvants are, they are the sentinel of the immune system, and they are required to awaken the immune system, so they act as a depot. So, there are few adjuvants available in the market, like if you see alum. Okay, there are a lot of adjuvants, but still we require a lot of new adjuvants are required. And we started asking question that if we are able to understand how a proper vaccine works, maybe we can get a certain clue, and we will be able to design a small molecule that can be used as adjuvant. And with this concept, we took yellow fever as one of the ideal vaccine. So yellow fever, one shot of yellow fever, if you take, it provides protection for almost 30 years. So yellow fever is one of the safest and one of the most effective vaccine ever made. And if you take this one shot, it provides protection. It has a neutralizing antibody titers for more than 30 years. So this is one of the most successful vaccine. And we took this as a model to understand how vaccine works. Okay. So to know that, what we had immunized in healthy volunteers, and then we used all these high throughput omic technologies, and we wanted to understand globally how this vaccine works. And fortunately, these are a couple of papers we made and at that time, and we identified one molecule. I'll just run faster because of time limitation. So this, one of the molecules that popped up across all the volunteers, healthy volunteers which took the vaccine, is one of the gene which you call as EIF2AK4. This is a EIF2AK4 or GCN2. This gene was it was popping up in most of the vaccines, and this was correlating with the predicted T cell and B cell responses. So we were wondering that how this gene, which at that time it was very novel, nobody knew about this, and then when we back search our literature, at that time, same time, I think this cartoon appears. So you can see why I'm showing this cartoon, because this gene is a sensor of amino acid restriction. So we were surprised that how a vaccine is able to trigger a molecule that is sensing some nutrients, why nutrient perturbations or why nutrient sensing is required, and how a vaccine works through this pathway. So at the same time, research was going in Wisconsin, where a lot of macaques were used, and we know that caloric restriction is beneficial for our health system. And caloric restriction, when I talk, a lot of people, a lot of girls, a lot of people are taking intermittent fasting, so-called. Okay, so this caloric restriction, today, if you know, this is involved in increasing longevity, preventing against a lot of cardiovascular diseases, age-related disorders like diabetes, etc. And today's research suggests that most of the benefits of this caloric restriction goes to the amino acid restriction. So many of the reduced intakes of protein and amino acids is somehow it is able to program our body. And one of the morning you talked about autophagy, Professor Amit discussed about autophagy pathway. So when there is a reduced amino acid and proteins, there is an induction of autophagy, and autophagy is a cleansing mechanism through which it cleanses, it brings about all the proteins and whatever pathogens inside the cells, and it presents to the T cells and the B cell responses. So this is the technique that that yellow fever vaccine was employing, and this was the first time we published in a Science, and we showed that the old trick was applied by one of the most successful vaccine to work and to provide a better protection responses. So I will just take you, and this is how it happens. So when there is a shortage of amino acids, there is a change in the translational, the translational machinery. So you have so many PTMs takes place, and as a result, some of the mRNA transcript, they are translated or they are kept in storage, and you see a punctuates like this. So we started experiments with that, whether the yellow fever vaccine is indeed it's activating this type of pathway. We can see that it's activating the integrated stress response pathway that is a downstream of GCN2. So the basic question, when you want to know the importance of somebody in life, either you kick out of that particular person or particular gene, or you put that particular gene or person inside your life. So in biology, what we say, we make a knockouts, where we delete that particular gene and see the effect. And how we do is that we made a knockout mice where we deleted this gene, and then we infected with a yellow fever vaccine. So when we delete this gene in this mice, and we challenge this infected with the yellow fever vaccine, this whole protective response has gone down, suggesting that the whole machinery was working, the whole yellow fever pathogen was working through this gene and this whole amino acid restriction pathway. And if you see this data, some of the data I'm just showing, how you can see that in wild type, this is a wild type mice, and this is a GCN2 knockout mice. And in GCN2 knockout mice, the T cell response were almost in all we took a lot of different organs, and we find that there's a drastic reduction in the T cell responses and the B cell responses, suggesting that GCN2 was a crucial player in a yellow fever induced model system. And then we are extrapolating to other different diseases. I'm not going to, because of time limitation, I'm not going to discuss about all these things, but this was a very significant data we got, and we were able to publish in Science paper. And this is the mechanism how so this is how it happens. I will brief you the mechanism. So yellow fever vaccine, it mimics, it induces amino acid starvation life conditions. As a result, what happens? Autophagy is induced. And when autophagy is induced, all the long-lived and the dirt materials in the body, they will be chopped up and they will be presented with the T cell responses. That's how the fasting or intermittent fasting is good for our body, because it will clean all trashes in our cells. So if you consider a room as a cell, okay, and you want to activate the cleansing process, this is one of the processes is happening. And indirectly, you are triggering your innate immune system. So this this study for the first time provided the molecular evidence of molecular basis of fasting or caloric restriction or amino acid restriction. So now we know that to translate the caloric restriction or fasting is very tough because there is not a baseline study or common study. So we thought that why don't we make a small molecule that can mimic this pathway and we can use for treating various diseases. And this how we had made a n number of molecules we have designed, and I'll take shortly. So the parent molecule, we took it from the febrifuge, which is one of the, and this is derived from Dichoria febrifuga. And this molecule, so this we synthesized with our collaboration halofuginon, and halofuginon, it mimics the condition of amino acid restriction. So you don't have to do fasting, but it mimics these conditions, and it can trigger these responses. So apart from halofuginon, we made a different compounds. I will just take it fast. So with our collaboration in chemistry department, so this is a parent molecule. You can see this halofuginon, and this is AR activators. So these are the chemistry part. I don't know about the chemistry, and we have synthesized almost 300 compounds which are showing better responses than the existing compounds. So I am not going to discuss about this. And so we started asking questions that can we extrapolate this finding for making an adjuvant? So adjuvants are required for vaccine development programs and new vaccines. So halofuginon is AR mimetics. And we tested this halofuginon adjuvant for making a tetravalent dengue vaccine. So we know that dengue is one of the biggest challenges today. And dengue is it's more bigger than what you have a COVID. And this, it's, please remind me timing, I'll just go through. Yeah, sir, just remind me about timing, how much time is left? Yeah, so with your permission, I can, if I, oh, I will wrap up, okay, yeah, good. So thank you so much. So, so we wanted to use these small molecules for making a tetravalent vaccine formulation, and we took dengue as a model. And so one of the antigen that is used is ED3, envelope domain 3 antigen, and that is a lot of neutralizing antibodies are generated against it. So we purified these antigens, and so you can see this, there are 4 serotypes to dengue, so we purified this, and then when we immunize the halofuginon, the AR mimetics, along with the antigens as a tetravalent vaccine formulation, we were able to see, so this is our immunization schedule. So once we immunize, we take on 7 days, 28 days, we do the bleeding, and then on 28 days, we euthanize the mice, we take the different organs, and we do the T cell and B cell profiling responses, whether it's able to trigger the immune responses or not. And these are the various assays which performed. And you can see that when we have used this HF, the AR mimetic as an adjuvant, there was an enormous increase in the T cell responses. And not only T cells, you can see the IF-1 gamma producing CD4s and CD8 responses. We can compare that with HF, and with the soluble antigen, there was a drastic increase, 1.17 to 4.72, and these were found to be drastically upregulated in different organs and tissues, and that's how it boosted the immunity. So I'll just take you quickly to this. I'll take you some of the slides which are important. And apart from CD4s, we also did CD8 responses, and similar result is evident. So these are some of the data which are required to monitor the immunological assessment, whether whatever your antigen, your formulation, or your adjuvants are working or not. So we have to do all these immunological profilings, and one of them is the T cell scoring, and then we have to the polyfunctionality of the T cell responses. And if you see this, the polyfunctional T cell responses were highly upregulated during HF. So how this programming has taken place, I have already told how this enhances the T cell responses. So that means it is able to induce a strong antigenous responses against dengue. And similarly, the B cell responses we can see. And finally, I will take you, important thing is, yes. So we have seen that when you use this AR mimetic, the overall antibody and T cell responses were enhanced. So whether these are effective or not, we do a neutralization assay. And this, when we use this as adjuvant, the tetravalent formulation was able to neutralize all the 4 serotypes. So this is one of the effective vaccine which we developed, and now it is in a preclinical assessment, and we are going to have a tetravalent vaccine against it. So, so these are the mechanisms I'm just going to skip. So when in 2019 and 20 that COVID started, so we asked question, can we use this adjuvant discovery model and we make a therapy against COVID-19? And we got a generous funding from DBT and other sources, and we started working on COVID-19 and to have an alternative therapy apart from vaccines. Okay, and we use this as adjuvant, and we thought, because there were so many companies who were making vaccines. There were Bharat Biotech, Serum, all they were in, they are big shots, and we were a small fish, so we cannot compete with them. So we thought, why don't we have alternate strategies? And we thought we'll use our adjuvants, and we'll make this whole inactivated virus as an antigen, immunize in a in a higher animals like equines, purify the antibodies, make a polyclonal antibodies, and use as a therapy. So we made a polyclonal antibody against COVID-19 using new adjuvants, and this is highly effective. If you see, so this is we got a phase 1 trial where this is the product in the market, it's going to the market, it's a COVID-19, and so that's what I'm saying to you, how to translate a basic science data into clinic is very important. And you have to explore the opportunities. So we thought COVID-19 is opportunity for scientists like us. So we had adjuvants, we had antigens, and we thought, why don't we make some new drugs? And this is how the successful collaboration between clinicians, basic scientists, we were able to make this COVID-19 as one of the first drugs under Atmanirbhar Bharat initiative, and this has completed a phase 2 trial, and we have applied an emergency approval. So this is some of the success story from our lab, and just I will take a long-term goal is to have so we have another tetravalent vaccine formulation, dengue. We are also making vaccine against poultry pathogens. We are we have a lot of diseases, Gumbarus disease, Ranikhet, and all those. We are trying to make multivalent vaccines against poultry diseases. So this is our long term. And just if, with the chair permission, 2 slides, I'll just show that another work. So this is how that the application of AR mimetics can be used for generation of new vaccines. And with this important talk, that best of all medicine is resting and fasting. It was Benjamin Franklin, who is the quote from Benjamin Franklin. And this is a very important quote. And I'll just take a small, another important work which is coming up from our lab is psoriasis. I know it is very tiring for you people, but it's very interesting, and just see how if you approach with a good question, we can take it to a lot of clinical practices. So psoriasis is a big problem in India and abroad, and using a high throughput technologies, we were able to identify one of the gene signatures, that is 12-hour lipoxygenases. And in morning, Professor Redena, we have an active collaboration with Professor Redena, and so we almost 1,900 different patients we screened, and we found that one of the genes that was playing important role is 12-hour lipoxygenase, and that is responsible for causing psoriasis. So these are the screening procedure we made, and so once we know that this gene is important, we made a transgenic knockout mice. So we made a humanized mice where the gene was expressing, and this, when this, you can see this data, when you overexpress this in made a transgenic, there was a clinical symptoms that is equivalent to psoriasis. So that means this the gene plays critical role in causing psoriasis. I'm not going to discuss about this mechanism because this paper is just, we are in its progress. So once we had this model, we designed a lot of inhibitors, and we have several different compounds. We screen different compounds, and we have, when we applied this drug on this model, we were able to see a reduction in the clinical scoring of the psoriasis, suggesting that this gene, that this can be one of the major therapeutic targets for developing a drug against psoriasis and other diseases. So with this, we have so many patents, and I don't want to discuss about this. And so all this could not have been possible without my students, without my collaborations, without postdocs and scientists. So these are, I have a lot of collaboration across the globe, and our lab is generously funded by different funding agencies. I have different collaboration. We have industrial grants, and I would like to thank all of my students, including Kala Jyoti, who has, you can see her photo, where she is, okay, she is here. Okay, so I'll thank everyone who has been involved in this project. Thank you so much. I'm happy to take any questions.
Is mine on? Ah, there we go. Do we have time for questions?
I don't know. It's to chair.
How about if we just take one question? We have someone right there. Please go ahead.
I know it's a COVID, so a lot of people will have questions, but because of time limitations, okay. Yeah, go ahead. Myocarditis, huh? Yes, so this is a question which...
heart palpitations and cardiac problems. Is it myth or it's fact?
See, I cannot, at this floor I cannot say, okay. Why? Because mRNA vaccine is the future, okay. And the problems what we are seeing in the newspapers, it could be because our delivery system, okay. So, see, most of the time the vaccine, new discovery of a vaccine takes a lot of time. But because all these vaccines have given emergency approval, many of the vehicles which are being used, okay, they cause a lot of inflammations, okay. And that could be one of the cause for the myocarditis, what we have, but I don't have any clinical data to say. I have not seen a clinical data. Only they are doing a correlation study between hospitalization versus, but the clinical data are not available, which really says that it is because of the vaccine that myocarditis people are seeing. Okay, so it's difficult to comment right this.
Thank you, sir.
Okay.
And one more, sir, is there any relationship between calorie restriction and telomere replication?
I have not done that, but it's an exciting question. Yeah, definitely. Yeah. So, but yeah, that could be because aging. Okay. So longevity, caloric restriction is associated with longevity. There is a lot of papers where longevity is associated. So is there any paper on that? I have not checked actually.
I'm afraid any other questions will have to wait until after the session is over. Let's thank our speaker very much.
Thank you. Thank you, sir.
Thank you, sir, for your insightful lecture. And I thank Professor Scott for sharing the session. I now welcome our Additional Secretary, Social Welfare Societies, Hanuman Naik, sir, and Professor Scott Hall to felicitate Dr. Nuruddin Khan. Yes, sir.
Hello.
putting this. Okay. Yeah. Left arrow. Cool.
Right?
Okay.
This is the one.
Yeah.
You use this.
Yeah. Thank you, sir.
Collar mic.
We'll introduce you when we do his profile. No problem.
Moving on, I request Ms. P. Sindhura ma'am to introduce our next speaker, Dr. Darshana Joshi.
I'm very much glad to introduce Dr. Darshana Joshi. Dr. Darshana Joshi is a passionate physicist, mentor, and social entrepreneur, founder of Vigyanshala, committed to recalibrating the gender balance in STEM disciplines. She is presently building India's largest digital ecosystem for affordable mentoring and coaching at scale for every aspiring, committed, talented girl in STEM at Program Kalpana. She is keenly interested in designing low-cost pedagogical tools to promote interdisciplinary thinking and problem-solving among youth from the most marginalized communities. Dr. Darshana is also the first Indian woman to be elected the President of Graduate Union, the primary representative body of postgraduate students at the University of Cambridge. She is a winner of several excellence awards and scholarships, including the Women Transforming India Award by Niti Aayog, Government of India, and United Nations Mission in India, and is endorsed by the Royal Society as an exceptional talent. We feel very much honored to have you here, madam. We request you to take over the session.
This is the pen drive, I guess. Sorry, not that one. Hello? Can you hear me? Hello, how are you? So, how many of you are enjoying the day so far? Come on, bigger hand, yeah? Why no?
So, good. So while we get the laptop up and running, can we do a small activity? I know you've been very tired sitting here, and so why don't you just get up from your seats? Just jump a bit, like 3 jumps on your seats, okay? With your hands up. 1, 2, 3. Okay? So now please sit down. Okay, good. Sit down, please. So, hello. So please settle down. See, while the slides are going up, let me tell you, I'm not going to talk much science today. I'm going to think like a girl or a boy who is in their first or second year of their bachelor's and thinking about what life holds for me, right? Where do I go from here and how do I think about a career in science, right? Science or technology or engineering. The good thing that we have had today is we have heard a lot of different views. We've learnt about entrepreneurship, that we can start our own startups and think about solving some of the pressing challenges that our country is facing today. Other side, we have heard about very interesting scientific developments through various model systems. We have had scientists from different corners of the globe who have come here and spoke about what they are doing at science, right? But as a young student of your age, how can you think about a career, say, 10 years from now, right? So let's do another small activity. Can I ask you all to close your eyes for a minute and settle down? Please close your eyes and just follow the instructions that I'm doing, okay? Think about the you that you will be, say, in 10 years' time. Think about the she or he, how they will look, how they will walk, where they are standing, what is their personality like, how they are engaging with people. Think about every smallest detail. Somebody may say, oh, they have a very good accent in English, or they are sitting in, a remote village in Telangana, just like the Three Idiots, what's his name, Ranchor Das Chanchar, right? So he's building these things. So you may be sitting somewhere and building those kind of innovation. Just think about every small, tiny detail that you can think about this person. Just bring it to a shape. That person should be standing right in front of you. Now, for each one of you, this is the person you should be having a conversation with every single day of your life, at least for 2 to 3 minutes in the day. Find that time, talk to this person, and see how they are looking, what they are doing, and what have you done today to get closer to this person. This is your dream. This is your dream career in science or engineering. Just think about how much money this person has, any attribute, like literally any attribute, how healthy they are, what prize or medal have they won, are they not being nominated for Nobel Prize or whatever, like, literally every single detail. And every single day, you should spend between 1 and 5 minutes talking to this person. And this will inspire you to push beyond your limits and your boundaries that you thought were stopping you from being the version that you want to be. Now, you may all open your eyes. Thank you for doing this exercise. This is an activity that I have done since I was in class 10th, and that was in 2004. And today is 2023, so almost 20 years now. And this is an activity that keeps giving me the peace and the push that I want. That inspiration only comes from within and not from outside. So my talk today is structured around few things that I'm just going to talk through my own journey. I come from a family which many of you may be coming from similar backgrounds. So I will talk more about it. But before I do, you see, is the picture clear to the people at the back? Right? So we all imagined where we will be in 10 years or 5 years' time, right?
But there's one thing that is more important than where we will be in 5 years' time, and that is where this world will be, right? So the world that we are living, how it is being shaped by our collective actions as humanity is more important than where we will be, and how we can create a better environment to solve for the challenges that this world is facing is very important. So let me ask, I have written some cues on the slides as well, but so the first slide on the left you see, right? You see lots of people putting their pipes in the tankers. This was the day when Chennai hit day zero. Day zero means there was no water left in the city. People had no drinking water, access to drinking water, and this was a city which 30 years ago had lot of industries purely because it was very rich in water resources. And it came to this day a couple of years ago. What is the second image on the right? Can somebody tell me? Forest fires, right? We heard a lot about forest fire from Amazon to Australia to even our own very India. I'm from Uttarakhand. My state faces forest fires every single day. And we lose millions of natural resources to forest fire. And most of these are caused by human actions. But science can stop this. Your training in science can be utilized to develop things that can solve for forest fires, that can solve for water scarcity. The bottom left image is the polluted Delhi that you all see during winters, air qualities and particulate matter above the detection limit, also way above the detection limit, like above 1,000 parts per million. And it's just so difficult. We saw a presentation about how asthma affects our body, but every single child in our polluted cities are getting affected with respiratory disorders and challenges. So we, of course, on one side, we need to study the drugs that can help us solve for this, but we also need to solve for air pollution. So this is another very pressing challenge, not just before Delhi, but before every other city in India that is going to face this challenge, right? So we need people like you to think of innovative solutions, design your own startups that will solve some of these problems. The fourth one is plastic. We all have seen what plastic can do to us, but this is a particular case. This is a huge patch of over 15 square kilometers of plastics off the coast of Japan and US. So in that area, there's a huge, it's almost like an island that is just plastic. It has killed all the marine wildlife there, and it is all because of human actions. And oceans are our Jeevan Data, right? They give us life. If they die like this, humanity wouldn't even survive, say, another 200 years. So we really need to solve for these things. My presentation has nothing to do with this, but this is a thing that I want to leave with all of you today, that think about how are you going to devise innovative solutions to solve for plastic degradation, or solve for air pollution, or solve for water crisis, or solve for wildfires, or anything that a farmer in Telangana may be facing, anything a farmer in the northeast or north of India is facing. Those solutions will come from you, because you are the first generation that is facing climate change on its face, and you are the last generation that can do anything about it. So you have to start thinking every single time, and innovation cannot be unidirectional. It cannot be only designed by people who are sitting on high on resources. It has to have a participation of people like you. So I am Darshana. I am a scientist by training, but today I am working to nurture future innovators and scientists such as yourselves. I'm sorry, I'm addressing only the young people in the audience here, so teachers, please pardon me. So people like you, so that you can build, design, use your creative juices to solve for these challenges, okay? So let me take you on a brief journey of my life, and this is my presentation title. While you are trying to solve for these challenges, you would definitely need a tribe of people who will solve these challenges with you. It cannot be done. Innovation never happens in silos. It never happens alone. You need a tribe. And why do you need a tribe? While you're building that tribe, how do you build a rewarding career in science? Rewarding for different people can mean differently. For me, rewarding means spending time with young people like you and really getting, to thinking about problems and solving them and bringing different people together and thinking about these. For someone else, rewarding may mean a paycheck of, say, 10 lakh a month, right? So rewarding is different, but defining that rewarding for yourself is really, really important. Let me walk you through my personal journey. I have tried to write some of the messages in Telugu. I don't know if it is coming all right here. So these are my parents. I'm a first generation learner. My parents were farmers in Uttarakhand. We migrated to Delhi when I was young. I was in class 3, and they both have studied till class 10th. My mother, then when she came to Delhi, she's a homemaker, and my father worked as an electrician with the municipal corporation. I am also a scientist, a physicist with a PhD from the University of Cambridge, UK. So I was born in a small village in Uttarakhand. These are our fields, some of them from the area I come from. I attended a Delhi government school when I came to Delhi. Then when I was in class 11th, I was lucky to be picked up by this organization called Udyan Care that supports girls from low-income families and gives them mentoring and career support. And I went to this school, which is in many ways is very similar to the social welfare and tribal welfare schools, because Delhi government started these schools where we had access to really good teachers, laboratories, libraries, and really it just lifted our spirits. And just like you people, we also had an entrance exam in class 6th, and we studied till 12th. Then I went to Miranda House, Delhi University, where I did a BSc in physics. After that, I was selected for an integrated PhD program at Jawaharlal Nehru Center for Advanced Scientific Research, which is a center in Bangalore. Many of you should write this, JNCSR. Please take a note, because after your pharmacology, many of you can apply for integrated PhD programs in molecular biology or ecology and genetics there, if that interests you. You can also apply for their summer research program, which is open now, which can help you acquire advanced teacher skills and understand some of the cutting edge science that is happening in the country. It is there I met some really good mentors. With their support, I went on to do a PhD at Cambridge. And at Cambridge, again, I got to become part of a very interesting tribe called Schlumberger Foundation. We had women from 86 developing countries, just like ours, from different parts of Africa to Latin America to South Asia. We are part of this big community, and we all had similar stories, right? Women growing up, whether in a remote village in India or a remote village in Africa, we all have similar journeys. And it's just interesting to see how that feeling is so, like the experiences are so relatable, and you can learn so much from each other. So, and then in 2019, I started Vigyanshala to bring the joy of science to many young people like yourselves. And also in 2017, I won the election for Graduate Union at Cambridge. So why I'm saying this in my slide is to emphasize in your career, extracurricular activities are equally important, right? So unless you are able to contextualize what you are learning with your extracurricular activities and the things that you will do for society with your learning, it can actually help you be more relevant and be more competitive compared to others who may not have. So for me, getting elected to this office was a very different experience altogether. I got to be a trustee of the Cambridge University, one of the first women from India to be at that position and to understand how a university like Cambridge is working with billions of pounds in its kitty, and how they are trying to now look at solving and bringing more talent like yourselves to university. So that really gave me the purpose that, okay, this is what I want to do, and that's how the career shift happened for me. So another important thing that I want to emphasize here is careers are never linear. They always keep evolving, and you have to start thinking about it, every action, everything that you do, how it is tying up with your ultimate goal. So the exercise that we did at the beginning, how are you connecting to that person that you want to be after 10 years, how your action today is going to be there, is really important. All right? Am I too fast, or is it okay? Okay? Okay, cool. So then now my partner Vijay, he's also a physicist, and as scientists, we have really developed a lot of joy in working with young people like you and developing interesting projects. So instead of working in our laboratories, we now work with you people on field and work on some exciting projects that can help you acquire skills. So this is what my day job looks like. In my scientific journey, I worked in a very interesting field called biomimetics. And again, this is super relevant for you people, because a lot of us grow up thinking, okay, I'm studying physics, I'm studying chemistry, I'm studying pharma, I'm studying maths, and they have nothing to do with each other. But the truth is, the boundaries are merging. A person who works in drug delivery needs to talk to a lot of computational scientists who are computing for them on everyday basis what are the potential drugs that can be useful for them. And then they combine those learning, and it helps them save time on thousands of molecules that they may have to synthesize otherwise. Now they can synthesize only a few molecules and test them, because they have synthesized all other molecules on computer and tested if they would work for a particular thing or not. So similarly, biomimetics is again is made up of two words. One is bio and mimetics, means learning from nature. And there are a lot of inspirations. So this is a slide just to show you different innovations that have found their inspiration in nature, right? So the lotus leaf have inspired super hydrophobic coatings that can be converted into surfaces that clean themselves. Similarly, your spider webs have inspired some of the most strongest materials like Kevlars that are used in bulletproofing jackets now. So similarly, we all see seashells, right? They are now inspiring hard coatings, because if you look at their structures, they are nothing but clay particles separated by proteins. Simple structure, but nature is such a powerful designer that it creates amazing engineering with very, very simple structures. And I think that's how science is also, right? We want to build things that are simple, that are doable, and that have applications very similar to what they find in nature, right? So it's a very nice emerging field, which many of you can actually go back and read about, and we'll find that there is a lot of nature that is inspiring research across drug discovery to new materials. I worked at the interface of material science and physics. So, so I use DNA, which people like to call the wonder material. And mind it, I have not studied bio beyond class 10th. So it was almost 10 years later when I started my PhD, I learned the fundamentals of what DNA is like, how do you isolate it, how do you take a plasmid and put it inside a bacteria and let it grow, and then you separate that. All those interesting things I learned. And had I thought that, okay, I'm just going to keep myself to physics and I'm not going to learn biology, I would have missed out on a very, very interesting opportunity. Though my work was understanding how these small ball-like particle would aggregate on top of an oil droplet. Again, this is a very important problem from the physics perspective, right? So we are studying thermodynamics, and this is purely thermodynamics-driven problem. But it can have applications into pharmaceutical, it can have applications into cosmetic industry, and it can have application into understanding how you can distinguish between a viral and a bacterial infection. That was an interesting project that I really worked towards the end of my PhD, and I absolutely loved it. ऐसा नहीं है कि I got bored of science and that's why I switched here. I loved my science, I still love it, but now the way I'm doing science is a bit different. I want to do it with you people so that you acquire the skills, and I learn how can I simplify much of this science to be done with frugal projects. Frugal means low-cost projects. So coming to career exploration, how did this interesting journey across, like saying studying physics, material science, and again PhD in interdisciplinary science, and now social entrepreneurship, it happened. 3 pillars, okay, for all of you that are super important. Curiosity. Can somebody read what is it written in Telugu, and if it is the correct word for curiosity? Huh? Is it correct? Jigyasa, right? In Hindi they call jigyasa. Second is persistence, right? Curiosity means be asking questions. Why? Why is it happening? How can I, like how can I make something better? So curiosity is really important. Second one is persistence. Like even when you feel you're failing, you have to keep going, keep going, keep going, right? Is the word for persistence correct? What is it? Can you speak louder, please? Is it correct? Okay, good. And the third one is tribe, right? So what does tribe mean in Telugu? Is that, what's the word there? Huh? Sangadam, right? Your community, the community that you are part of. So these 3 things are super important when you are thinking about a career. So let me describe the tribe word bit detailed to you. A tribe is basically a distinctive group of people who are driven by common purpose or ambition, right? So that when you feel you are about to fail, you have other people in your community who are going to uplift you. So please find that tribe for yourself. This is a very famous Sanskrit shloka that describes the importance of tribe very beautifully. It goes, Acharyat padam adatte, padam shishva samedhaya, padam sah brahmacharibhya, padam kalakramena cha. A student learns a quarter from their own intelligence, a quarter from its peers, a quarter from the mentors, and a quarter from the passage of time, right? So it's the quarter that you learn from your mentors and your peers is what constitutes your tribe, right? Your mentors, your peers are your tribe because they are going to help you multiply your learning with lesser effort, and also making sure that when you are facing challenges or difficulties, they are there to increase your motivation, right? So if you're thinking about career, a tribe is really, really important. I'll just give an example of tribe, okay, from my student days. So this is 2006, the year I went to college. Three of us, this is my friend Srishti, Renu and myself. Three of us came from families where neither of our parents had gone to college. So we had no idea when you go to a college what happens there, right? And we went to one of the so-called posh colleges in Delhi. So absolutely clueless, like where will we go, what we'll do after BSc. But together we kept exploring with our teachers, just like today. Our teachers also took us to some conferences. We got to talk to some really nice people, got enthusiastic about nanotechnology, and started studying. This is three of us in 2019. And all three of us are PhDs in physics today. Both of them got Marie Curie Fellowship for their PhDs. I went to Cambridge. So none of this our families could have envisaged when we were thinking that we want to go and study in Bangalore or we want to go to UK or Europe. Our families were very against it. They're like, no, we won't send our girls this far and all. But together, we negotiated, together we fought those battles that had to be fought with the families. Particularly for girls, I'm saying this thing. It gets really difficult sometimes if you are negotiating alone, right? So you need that support who are in a similar boat, who can talk this thing with you. And of course, we were supported by amazing mentors who again chipped in when it was required to convince and counsel our families. So don't let what your family thinks shrink the size of your dreams, right? So rather, use the resistance that you're getting, if any, from them to really increase the size much bigger. Us anger ko, us resistance ko, use it as your power and not as your weakness, okay? So that's a very important lesson I want you people to take with yourself today. So again, just an analogy. When you are with your tribe, climbing a mountain alone with a stone tie looks difficult, right? But if you have three people pushing it, the efforts get reduced, but the rewards get multiplied. So you will be able to go ahead in life and do better. Now to the last part of my presentation, where I want to talk about reimagining your career, right? So, and I'm going to focus on acquiring STEM skills early on so that when you apply for any position, whether it's a research position, whether it's a job in industry, or whether it's a grant for starting a startup, some of the things that will be super important are your STEM skills, right? So what is STEM? I will talk more about it in a bit, but a few things on the importance of STEM skills. So World Economic Forum is a big agency which sits in Geneva. They are saying that more than 80% jobs in the next decade will require some or the other form of STEM skills, right? And that means much of the jobs that we thought were very manual are going to be super automatic now, and you will require some or the other form. Even if you were studying biology, you would require advanced computational capabilities, right, to solve for the challenges that will come. So apart from the job sector, having people who have good STEM skills across a spectrum is very important so that India can truly make sure that all its people taste what development looks like, right? Until and unless people like us who are coming from mountains or villages do not have access to advanced STEM skills, the voices of our communities will not reach the forefront of science. Our challenges will not get solved, because what may be a challenge to us may be restricted to us only, right? Because other people don't even know. Their voices may not be even reach the policy table. Tomorrow, I hope many of you have your training in science and are going to work for, say, the government and design policies that will impact a farmer's life in a rural village, or design policy on energy that may give electrification to a village that did not have it earlier. But all of that scientific thinking will be rooted in your training. So it's really important to acquire these STEM skills early on. But what are these STEM skills, right? So just give me a minute, please. So if I have to expand STEM, it stands for science, technology, engineering, and mathematics, right? The disciplines. But if you were to think about in terms of competencies, so you should be able to think about a problem and think about solving it from a hypothesis-driven way, right? You are able, hypothesis means a possible solution that you are thinking and you are able to test it. That's how much of our scientific research works. The second thing is having an ability to do experiments, to do practicals, having that hands-on experience, right? Then to be able to think critically, what may be the problem, because how will you drive the hypothesis that this may be the reason? That will only come when you have thought about it critically. So that's a skill called critical thinking. Then similarly, mathematical and logical reasoning. So if you see a large amount of data, and we saw much of it today in many presentations, right? How do you draw inference on that data? That comes from your mathematical and logical reasoning. And then, of course, data generation and analysis. So if you have thought about a hypothesis, thought critically about it, thought of a solution, to validate that solution, you will need to generate data and you will need to analyze it and you will need to show to people that this works. For example, in the previous slide, previous presentation, we're talking about drug discovery, right? How does a drug work for someone? So we really need to do those experiments. We really need to gather that data, analyze and draw inference, and either accept or reject the hypothesis that we proposed, right? So all these skills are not just going to be restricted to scientists, but with the new era, what we call Industrial Revolution 4.0, these are going to be really, really important across sectors. For instance, before pandemic, not many of us thought that education could be so digital, right? And how somebody is learning or how they are interacting with technology, we could gather data. And that data can give us insights about what potential skills or sectors may be important for people, right? So all of that, again, got heavily digitized because the pandemic was there. But again, this would have anyway happened, maybe in a decade's time. But just because of the pandemic, it got accelerated. So that shows if something like education could be, so digitized with STEM skills, any other sector can be. Right now, we have technologies that are talking about farm to plate, right? Where a farmer grew, how did they grow it, what pesticides or chemicals did they put, is it organic, is it not? All of that you can track with the help of technology. So that's the impact STEM skills and technology are bringing into various sectors. So it's really important that you think about them early on. So how do you develop STEM skills? So this is one of our collaborators, Professor Nagarjuna at Homi Bhabha Center for Science Education. He says, let's not call them skills, let's call them STEM habits. Why not all of us start thinking about STEM skills as everyday habits? Why don't we ask questions? And once we ask questions, we bring 3 Ps. We bring our peers, our friends, we play with them, and we think passionately about the questions that are coming to us. We record our observation, we research about it, and we report it to a wider world to think of what we have noticed, right? So there's a very interesting app. All of you should just check it out. It's called the Cool STEM Games, and you will see many more home scientists like yourselves who are doing different things, are collaborating there. So my mantras for all of you is, wherever you are staying, your 4x4 room, make it your lab. Right? So, and think beyond your limitations, think beyond your knowledge and barriers of learning, and design your own training. If something you learned in the class, think about it. Can you study using some model system within your college hostel or within your room? And think about a question, discuss with your peers, and see how that question can be channeled into a more serious research question. And then all of you explore that question together, observe things that are, that can either prove or disprove of what you are thinking. And then you have platforms like Peta Studio, which is, again, I said cool STEM games earlier. There you can record this. So let me give you an example. Just like you, thousands of home scientists, as these people call themselves, have been studying various model systems. This is common housefly, this is a hydra, I think it's a water flea, earthworm, snail, mosquito, butterfly, and cardamom plant. So they study, people have just picked up these systems and they are trying to see how they can grow them without having a big laboratory or something, and how they can study various questions of biology at home. And this group meets every evening between 7:30 p.m. and 9:30 p.m. online, and young people like you in their second year, third year of their BScs, B Pharmas, B Techs come and share their learning. And together they ask questions and they create a space where all of them can learn. And in the last 2 years, I have learned a lot of biology, having not studied biology beyond class 10 with this group. So as young learners, this can be a point where you can start getting introduced to these communities, start building your skills. So when you apply for a master's degree or for a job or for a PhD or for anything that you are going, you can show these experiences that this is how I have followed my curiosity, these were the results, this is where I have documented my research, and this is how I'm going to do. That will help you when you are making, say, a research application for a summer research fellowship, because people will now see that you are already thinking about your career very seriously, you are acquiring these skills, and now you are ready to share it with the world. Similarly, to all the girls in here, I would like to invite you to be part of our program Kalpana. With Pawnee ma'am's help, we are now launching this program for every girl who is studying at the social welfare and tribal welfare colleges. And we really want to build your careers with you and for you. Just an example, we have Aparna, she's from ISER Bhopal. When she came into the program, she was at home, it was pandemic. She designed an electrochromic window, a window that could change color with the application of electric field, with a mentor who was sitting in Italy. So they were working online, and with that project, she got selected at Jawaharlal Nehru Center, where I was doing my master's, for a project. And now, because of that project, she's going to Canada to do her summer research with the MITAX fellowship. So this is how you build your step-by-step careers, right? Things just don't happen out of the blues. We have to start following and building that whole thing brick by brick. Similarly, Shramana, she's studying at Delhi University. She was in Kolkata during the pandemic at home, and she came to program Kalpana. She connected with other mentor who is working in bioinformatics and drug design, through computational tools, right? So they started working on very interesting projects. She worked with that mentor remotely for 1 year, and because of that project and the learnings and the skills that she picked, she is now in the inaugural cohort of Quad Fellowship, which will give her access to do a PhD in the United States with fully funded project. So this is the first time this scholarship was inaugurated, and she is in the top 25 students. Similarly, Diksha, she is from Pithoragarh. Pithoragarh is a town, very remote town in Uttarakhand near the Nepal border. And she came into program Kalpana. She was working with a mentor who is an ecologist in the UK. And because of his mentoring, she got really interested in ecology and started mapping the various species in her neighborhood, mapping over 80 different moth species with her friends. So they mapped lots of interesting species. And because of that project, today, she's now working with a London-based think tank on a year-long paid internship. So after she finished her BSc, she got this internship. And she's working with scientists from US, UK, and Africa, and studying the impact of climate change on agriculture and how we can mitigate it. So these are the interesting possibilities that the computers and the digital system can enable for you. So today you met so many scientists here, as Pawnee ma'am was telling, right? Some of these people will be your future PhD guides or postdoc guides or collaborators. So today is the day when you start networking with them, you start talking with them, go back, read their paper, anything that inspired you in the entire day today, pick that one thing that caught your attention, go back, read about it, dig deeper, and see if it really interests you and how you can continue to explore that field, right? Again, bringing it back to where we did that exercise, that if you can dream it, you can be it. So it starts with dreaming, but it progresses with action, right? So you have to act on an everyday basis towards your goals, towards your ambition that you want to perceive, that you want to build for yourself. And it starts with being curious today. It starts with building a community today, and it starts with being persistent every single day of your life. So with that, I would like to close. Thank you, and if there are any questions, I'm happy to take.
Thank you, ma'am. On behalf of T-Series and TT-Race, we would like to thank you, ma'am, for mentoring our young minds with your STEM program through Vigyan Shala. This session will definitely guide all our participants throughout their lives. The session is open for any questions from the participants.
You can ask me anything, seriously. I think I hope I didn't put you to sleep, but if there is any question, please feel free. Like literally anything that comes to your mind. Okay, I put you to sleep then. Come on, anything like about the projects that I said you can do. It starts with being curious today.
At least a question like, how do I enroll in Project Kalpana? Yeah? Maybe you can tell that.
Yeah, so with Pawnee ma'am, we are collaborating with your colleges, and we have sent notifications to your college, and you can enroll online. And it's an online program where we bring role models like you met today from across the globe, and we give you an opportunity to directly engage with them and ask them questions and really learn from their areas of expertise. So we have circulated the notification. Ask your teachers if you haven't already enrolled. We have about 1,000+ registrations from your colleges already, and our next session is going to start on the first week of March. And yeah, if you would like to be part of it, please feel free.
I would now request Dr. Nuruddin Khan sir and Dr. A.S.N. Pavani, OST Higher Education T-Series, to felicitate Dr. Darshana Joshi. Thank you, ma'am, and thank you, sir. May I now invite Mercy, ma'am, to introduce our last speaker for today's session, Professor Hanuman Sagar Boddu.
Good evening, everyone. I, P. Mercy, lecturer in microbiology from Social Welfare Degree College, Medak. It's my privilege to introduce our next speaker, Professor Sai Hanuman Sagar, sir. He is a professor of pharmaceutics at Advacnes University. He has expertise in formulation of drugs in the required dosage forms like tablets, syrup, solutions, injections, eye drops, and stability monitoring with respect to US, Russian, and Asian markets. He also has high-level knowledge in the development and characterization of various novel drug delivery systems such as micro or nanoparticles, polymeric micelles, and microemulsions. He has published over 100 research and review articles in various peer-reviewed journals and holds 2 patents. Dr. Hanuman Sagar received over 250,000 USD in research funding from various foundations. He is on the editorial board of the Journal of Pharmaceutical Sciences and Journal of Pharmacy and Bioallied Sciences. Dr. Badoo is a co-editor of the book titled Excipient Applications in Formulation Design and Drug Delivery. His current research interests are developing novel drug delivery systems for treating various diseases, especially retinal diseases. I now welcome Professor Sai Hanuman Sagar, sir, to deliver his talk on an insight into the back of the eye delivery, challenges, and its opportunities. We welcome you, sir.
A very good afternoon to all the students. Yeah, I see since morning you guys are sitting in the hall and it requires a lot of patience, and I appreciate all of you for, so patiently listening to all the talks. Probably the talks are of high level for your standard, but still I think at least some of you I think should be benefited out of these talks. And I'm not going to take a lot of time. I'm going to deliver a talk on an insight into the back of the eye delivery, what are the challenges we have, and then what are the opportunities. So it's basically related to the eye, okay? So let me start my presentation with a famous quote by Chanakya, who is a teacher as well as a politician. So way back in, 3rd century BC, he suggested or he quoted, Sarvendriyanam Nayanam Pradhanam. What does that mean? It means eye is the most important sensory organ. So what is the reason? What is the reason behind this? It's because 80% of the knowledge that we acquire is through the eyes, okay? So we can acquire knowledge through hearing, we can acquire knowledge through touch, okay? And then the most important thing is we acquire the knowledge through seeing, okay? So that's the reason eye is the most important sensory organ of the body. Okay, so 80% of the 80% of the knowledge is acquired through the eyes, and that is partly one reason why eyes are very protected. So protected, I mean, you see, like there is continuously blinking of the eyes, and there is also generation of tear fluids. Okay, you see the generation of tear fluids in the second picture. So what is the reason behind this? Why do you think the eyelids will continuously blink, or why there is continuous generation of tear fluids? So the main reason is to prevent the entry of foreign bodies into the eye. Okay, to prevent the entry of foreign bodies into the eye, and also there is one more mechanism by which eyes have an ability to clear the unwanted material, that is through nasolacrimal duct. So nasolacrimal duct is basically, here you see the third picture, wherein we have a small duct that connects the eye to the systemic circulation. Okay, so whenever there is excess fluid that is present in the eye, the nasolacrimal duct will open, and this can result in the drainage of the excess fluids into the systemic circulation. So these are the three important mechanisms by which, or three important mechanisms which protect the eye. So these are good for the eye, but the thing is, these protective mechanisms can pose significant challenges in terms of drug delivery. Suppose if you are trying to deliver a drug to the eye, so there is continuous blinking of the eyelids, and also the tear fluid generation. They try to clear the drug out of the eye. Okay, even the nasolacrimal duct also does the same thing. So in the next 15 minutes, my job here is to give you a brief introduction about the anatomy of the eye, then what are the diseases we are trying to treat here. Okay, mainly back of the eye disorders, means diseases that affect the retina. Okay, so we'll concentrate mainly on retinal diseases, then what are the risk factors and challenges what we have in terms of current delivery options. Okay, and then how we can overcome these challenges using nano carrier systems or using nanotechnology. So these are the things I'm going to explain in the next 15 minutes. So let's move on to the structure of the eye. I think most of you are already familiar with the structure of the eye. Mainly we can divide the eye into two segments. One is So coming to the structure of the eye, mainly eye can be divided into two segments. Okay, the anterior segment and the posterior segment. So the anterior segment is the front portion of the eye that is visible to a naked eye. Okay, mainly it consists of tissues such as you have cornea. Okay, the transparent tissue we have in the front. Then conjunctiva. Okay, which is going to cover the sclera. Then we have iris, lens, as well as aqueous humor. Aqueous humor is present behind the cornea. So these are the important tissues in the anterior segment. But we are mostly interested in the posterior segment, okay? That is the back of the eye. So posterior segment of the eye, you see like 2/3 of the eye, which is not visible to a naked eye, is called as the posterior segment. And the main tissues we have in the posterior segment are, we have sclera, which is the white color coating around the globe of the eye. Then we have choroid. Choroid means it contains mainly blood vessels, okay? The blood vessels that nourish the eye. So that is choroid. Then we have retina. So retina contains the very important cells of the eye, which are rod cells and cone cells.
which help in the vision process. Okay, these are the cells which help in the process of vision. So how does it work? So naturally, when the light enters the eye, they get focused onto the central part of the retina, which is called as macula or the fovea. So when the light rays hit the retina, a series of electrical as well as chemical signals get generated, which are transported to the brain via the optic nerve. So we have optic nerve over here. So this optic nerve transfers the signals to the brain where the signals get processed. Okay? Once the brain processes signals, then it can get the image. The image is again sent back to the retina. That's how the whole vision process works. So any disease that's going to affect the retina, okay? So we are mostly interested in the retina here. So retina is the tissue which is helping in the vision process. So any disease that affects the retina, obviously, is going to result in vision loss. So let's see what are the diseases that affect the retina, okay? So here we have, I'll highlight the important ones, most important ones. We have age-related macular degeneration. So age-related macular degeneration is wherein you have, there is excessive growth of blood vessels in the choroid region. We also call it as choroidal neovascularization, and it is projected that approximately 300 million people will suffer from this disease by 2040. And then we have diabetic retinopathy as well as diabetic macular edema. So as the name suggests, diabetic retinopathy is something which is related to, people suffering from diabetes tend to have this disease. It's a microvascular complication related to diabetes. And diabetic retinopathy will progress further into diabetic macular edema. So, and approximately like 233 million people are projected to suffer from this disease by 2045. And here you see like the major consequences of these two diseases are proliferation of retinal cells or the blood vessels, or we also have the inflammation of the retina. So these are the two major consequences which we have in these diseases. Likewise, we also have a couple of other diseases which affect the retina or back of the eye. We have glaucoma. It's a very common disease. It is nothing but increased intraocular pressure. So whenever there is fluid buildup in the anterior segment of the eye, aqueous humor, so it's going to create a pressure in the back of the eye. And this is going to result in the death of neurons that are present in the retinal cells. So that is also a disease that can affect the back of the eye, and like approximately 110 million people will be suffering from this disease by 2040. Likewise, you have uveitis, which is another complication, and it's the third leading cause of blindness across the world. Then we have retinitis pigmentosa. So all these diseases mainly affect the back of the eye, which is the retina. So people who suffer from this disease eventually tend to lose vision. And what are the risk factors for these diseases? Mainly we have aging. So mostly elderly people tend to have any of these complications. Then the other risk factors are smoking, obesity. Then we have genetic, family history. If anyone in the family is suffering from, these diseases, then their offsprings also will tend to have this disease. Then we have eye trauma as well as diabetes. So people suffering from diabetes might end up having diabetic macular edema or diabetic retinopathy. So now let's look at what are the ways to deliver the drugs to the eye. Okay. Here we have mainly the popular way of delivering the drugs to the eye is through eye drops. Okay. So you might have taken eye drops several times, and eye drops are good if it is, trying to treat inflammation occurring in the front portion of the eye, that is the anterior segment. Okay. But eye drops fail to deliver the drug to the back of the eye. So back of the eye is this retina. Okay. Why they fail to deliver the drugs to the retina, I'm going to show you on the next slide. So for treating retinal diseases, mainly we have three options. Okay, the number one option is systemic administration. So what is systemic administration? It's nothing but giving the drugs either in the form of a tablet or injection. Okay, so the drug gets distributed throughout the body and finally it reaches the eye. So that is one mode of administration. Then we have intravitreal injection. Okay, intravitreal injection is wherein the drug is directly injected into the eye. Or we have periocular injection. Periocular means outside of the eye. Okay, outside of the eye means like it's not injecting into the eyeball, but the drug is placed surrounding the structure of the eye. So I'm going to talk very briefly about each one of these, and I'll tell you what are the disadvantages that are associated with this mode of administration. So let's start with the topical administration. Okay, I told you conventional eye drops fail to deliver the drugs to the retina. Okay, so mainly conventional eye drops means the eye drops that we use are mainly meant for topical infections or inflammations. So what is the reason why they fail to deliver the drugs to the retina? So you can understand it through this slide. So whenever the drug is administered topically on the cornea or in the precorneal space, 90% of the drug that is administered, okay, or even more than 90% of the drug gets washed off, okay, washed off because of the tear turnover, because it's a protective mechanism, okay. So 90% of the drug either is washed off by the tear turnover or through blinking, or we also have the nasolacrimal drainage, which I told you in the first slide, okay. The nasolacrimal duct is going to drain the fluids into the systemic circulation. So only we are left with less than 5 to 10% of the drug. So this 5 to 10% of the drug is good for treating any diseases occurring in the anterior segment, but it cannot reach in sufficient concentrations in the back of the eye. Mainly the drug has to move either laterally or it has to move transverse. So lateral means like it has to cross aqueous chamber, lens and iris, then vitreous humor, okay? Then it comes to retina. That is one pathway. Then we have transverse pathway wherein the drug will spread around the globe of the eye, that is conjunctiva, sclera, from there choroid and retina. So that is the second mechanism, or it could be a mix of both. Okay, that means lateral and transverse. But all these mechanisms can only deliver 10,000th part of the administered dose, which is nothing almost. Okay, so 10,000th part of the administered dose reaches the back of the eye, which is not therapeutically effective. So we have to rule out the possibility of eye drops. We cannot deliver the drugs to the back of the eye using eye drops. So now what are the other possibilities? So I told you the first one is systemic administration. Systemic administration is giving the drug in the form of tablets or injections. So using the systemic administration, studies indicate that less than 2%, okay, depending on the nature of the molecule, so less than 2% or maybe less than 1% of the drug reaches the retina. Again, several doses of the drug should be taken systemically, which can result in systemic side effects. So that is also, it's not a good option, okay? So systemic administration is used, but still it's not a good option for treating the back of the eye disorders. Next, what is the other option we have? It's intravitreal injection. So this is the best option we have currently, okay, for treating back of the eye disorders. Either it is intravitreal injection or periocular injection. Intravitreal means directly injecting the drug into the vitreal cavity, okay. So you can see in this picture how we can inject the drug into the vitreal cavity using a 30 gauge needle. It's a very thin needle. 30 gauge needle is used for injecting the drug, or the same can be injected surrounding the globe of the eye. So this region. In this region, we can call it as peribulbar route or subtenon route, or we can have retrobulbar route. So depending on where the drug is placed, we have these modes of administration. And there are several drugs which are being administered in this way, like like a lot of antibodies, bevacizumab, ranibizumab. Then you have antibiotics, okay, like clindamycin, ganciclovir, then corticosteroids like triamcinolone, dexamethasone. So all these drugs currently they are administered using either periocular or intravitreal injections. And I need not say what are the disadvantages with this intravitreal injection. Because taking a simple injection itself, like a small needle prick itself, is very painful, right? So imagine taking an injection into the vitreal cavity, so which majority of the patients do not prefer. Okay, so there is a certain procedure, that means the eyeball is anesthetized before making the injection, but still majority of the patients do not prefer to have intravitreal injections. And again, intravitreal injections are also known to have couple of side effects, you know, like endophthalmitis, retinal detachment and all. So mainly if you see here, they are trying to deliver anti-inflammatory agents, because inflammation is one of the major consequences for most of the retinal diseases. Okay, so here you have to understand anti-inflammatory agents means mainly corticosteroids like triamcinolone or dexamethasone, which have very low solubility. Okay, getting them into an injection is also a big challenge. So this is one of the option. And as these intravitreal injections are painful, because these injections have to be taken once every 2 months, once every month or once every 2 months, depending on the condition of the patient. So it's very inconvenient for the patients. So the other option that we have is intravitreal implants. So I think already you might have studied about implants in your pharmaceutics course. So implants are nothing but these are tiny structures which can deliver the drug you know, for several months to years once they are placed inside the eye. Okay, so we have like a few implants which are there on the market using non-biodegradable polymers, okay. So either these are, you know, placed via a small surgery like you see in the first picture here, or they can be directly injected into the eye. Okay, so again, this is the best possible option we have currently to treat the retinal diseases. Okay, mostly these implants, they contain anti-inflammatory drugs, because we have to take care of the inflammation process. We have to take care of the inflammation process. So these implants, obviously, they have certain advantages, like they provide sustained release of the drug, because they can deliver the drug from months to years, okay, in a sustained manner. And obviously, that is a major advantage, but the disadvantage is still they are invasive in nature. Invasive means either they have to be injected or they have to be surgically placed. So still it's something which is, you know, most of the patients do not prefer. So now the biggest question that we have in front of us is, can we really deliver the drugs to the back of the eye using an eye drop? Because that is the most convenient way of delivering the drugs to the eye, which is going on for several years. Okay, is it really possible to make changes to the eye drop formulation so that the drug can get to the back of the eye or the retina in required concentration? So this is the biggest question we have. And the best bet for this question is the use of nanotechnology. Okay, the use of nanotechnology means using nanocarrier systems. So these systems, we have several systems, like simple nanoparticles we have, liposomes we have, then we have dendrimers, surface modified nanoparticles, nanomicelles. So there are several systems, okay? I have worked with majority of these systems, and for this research, I'm going to use nanomicelles. Okay, this is the system of interest for us currently, and I have made some modifications to these nanomicelles, and what we are currently adopting in the lab is mixed nanomicellar technology. Okay, mixed nanomicellar technology means for forming a micelle, we require one surfactant. Okay, when the surfactant concentration is beyond the critical micellar concentration, the micelles automatically form in water. Okay, when we dissolve surfactant, and when the surfactant concentration is beyond the critical micellar concentration, they automatically form micelles. Micelles means like these are spherical shaped vascular structures. So now, mixed micelles means when we are using more than one surfactant. Okay, more than one surfactant means we can use two. So what is the advantage of mixed micelles means? They form structures with better stability. You know, even with dilution, they remain stable. So that is the major advantage of mixed micelles. So we are, our plan is to implement mixed nanomicellar technology, and using this technology, we want to deliver the drugs to the back of the eye. And so this is the mechanism which I'm not going to talk about, how micelles can, this is a hypothesis in fact, how micelles can deliver the drug to the back of the eye, which I'm not going to talk right now. But the major goal of our project is to design and characterize a eye drop formulation, an acceptable eye drop formulation which can deliver dexamethasone for treating retinal diseases. So dexamethasone is a corticosteroid. It has very good anti-inflammatory properties. And already we have several products on the market which have dexamethasone, like suspensions. We have even an implant of dexamethasone, okay? But we don't have an eye drop formulation. Eye drop means like, a clear solution, okay? So dexamethasone is very widely used for treating anterior segment inflammations, like conjunctivitis, okay? So these diseases can be treated with eye drops. So what we have used is, we have used this mixed nanomicellar technology, wherein we have, screened several surfactants which are approved by the US FDA for ocular use. And we have decided like two surfactants. One is polysorbate 80 and polyoxyl 40 stearate. So these are the two surfactants that were finalized after several trial and errors. Because we want, a stable micellar structure, that is one criteria. The other criteria is it has to, it should be able to dissolve required amount of dexamethasone, 0.1% dexamethasone. And we named this formulation as DexSolve. Okay, DexSolve means solution. Okay, because the marketed formulation is a suspension product, okay, which cannot deliver the drugs to the back of the eye. But the product that we created is a eye drop solution, which we call it as a DexSolve. The manufacturing procedure is very simple, okay. I'm not going to again explain the manufacturing process here, but the process can be easily scaled up. Easily scaled up means it can be easily reproduced in a larger scale. So majority of the technologies, they fail to, they fail the, that means they fail when they are being scaled up. That means it's difficult to scale them up at a bigger scale. In a small research lab, they tend to work, or they tend to make wonders. But when the same system is reproduced on a larger scale, they tend to fail. That is the major, problem with majority of the nanocarrier systems. But this process is very simple, and it can be easily scaled up. So we have dissolved 0.1% of dexamethasone using 4.2% of polyoxyl 40 stearate and polysorbate 80. Okay? And then we have did some basic characterization studies, whether these nanomicelles are formed, if they are formed, whether they are stable or not. So these are very basic preliminary studies which we do for any eye drop formulation. So we looked at the particle size. These are 20 nanometers, close to 20 nanometers. This is the particle size curve, the Gaussian distribution curve. Then we also looked at the morphology. Morphology means like the structure, using transmission electron microscope, negative staining. So we can clearly see these dots here. They are of size 30 nanometers, 40 nanometers. So these are the tiny nanomicellar structures. So that means micelles are being formed, and they are able to deliver, they are able to dissolve dexamethasone because the solution is clear. And dexamethasone has very poor water solubility. Okay, you cannot dissolve 0.1% in water unless the drug gets into the micellar formulation. So these two studies confirm that micelles are being formed. And now we need to make sure the micelles are stable. Means once they are administered to the eye, they get mixed up with the tear fluids. Once they get mixed up with the tear fluids, the structure can collapse. Okay? So we have to make sure whether the structure is intact or not when they are administered. So how can we test that? Is by using artificial tear dilution test. Means we have artificial tears which are available on the market. Mainly these are used for treating dry eye conditions. And we have diluted the micellar formulation by 2 times, 10 times, 15 times. And we have seen whether the drug is getting precipitated or not upon dilution. And what we see is, like, when we measure the absorbance using a UV spectrophotometer, the absorbance is very less than 0.1, which indicates that the drug is not getting precipitated. Okay, that means the structures are stable when they are diluted with the tear fluids. So that is one criteria. The other criteria is the eye drop should be sterile in nature. Okay, sterility is an important characteristic of ophthalmics as well as injectables. That means we should be able to sterilize the formulation using any of the available techniques. So we used filters. Okay, we have like filters, syringe filters we call, and these are made up of you have several materials. Okay, PTFE we have, polyether sulfone material, nylon material. So these are membrane filters. These can be attached to simple syringe, and when we pass the solution through the syringe filter, the product will become sterile. So we have passed the solution, the Dexol, through these filters, and we have seen whether we are getting the entire drug filtered or not. So when we measure the filtrate, again we are seeing 100% of the drug. That means we are able to sterilize the formulation using simple filtration technique without any issue. So that criteria is also solved. And then we looked at the in vitro drug release study to see how the drug is getting released from the formulation. So you see here, again, this is the process by which we do the study. And if you look at the graph, the drug is not released all at once. It's getting released over a period of 8 hours. So slowly you see the drug getting released, 20%, 40%, 60%. So until 100%, you see it's taking 8 hours for the drug to get released. So the drug is getting released in a sustained manner. And then finally, we went ahead to test the formulation in rabbits. So we administered the formulation in rabbits to see whether the drug is getting to the retina or not. So we have used white rabbits, New Zealand rabbits, and so we have taken 5 rabbits, and we administered the formulation to the left eye of the rabbit, and the right eye was used as a control. And then after 1 hour after administration, the rabbits were euthanized, the eyeballs were taken, the tissues were separated, okay, like cornea, conjunctiva, aqueous humor, lens, retina, sclera. So we have separated the tissues and measured the drug concentration using a validated analytical method. Okay? So we clearly see that the drug is getting into the retina. Okay? So we measured the concentration of the drug, and it is around 43 nanogram per gram of retina. So which is a required concentration, that means required therapeutic concentration. So then the next thing is, we have seen whether the eye drop formulation is causing any irritation to the rabbit eye. Okay, so this is one more criteria because it contains a lot of surfactants. And surfactants, they have a tendency to irritate the ocular tissues, which we don't want. So obviously we did a DRACE test. Okay, DRACE test is the accepted test for looking at the toxicity of ophthalmics. And mainly we see after the administration of the eye drop, whether there is any changes, whether there are any changes occurring to the cornea, iris, or the conjunctiva. Okay, so cornea, so we basically score, okay, by looking at the eye with the help of a torchlight after fluorescence staining. So you see the scores are pretty much 0. That means we did not notice any significant difference between the treated eye versus the control. Okay, only we have seen some redness, but it's only temporary. Okay, it's only temporary redness. So this test was conducted for 7 days. Like we administered the drug 2 times daily for 7 days, and still after repeated administrations, we did not see any kind of, irritation or side effects in the eye. And after 7 days, again, we euthanized the rabbits, separated the tissues, and again analyzed the drug concentration using LC-MSMS, which is, liquid chromatography, tandem mass spectrometry, a validated method. And again here, you can clearly see the drug concentration in the retina is around 67.3 nanogram per gram. So typically, around 10 to 50 nanogram is good enough for having anti-inflammatory properties. So we are able to get significant, that means therapeutic concentration of the drug to the back of the eye using this nanomicellar technology. So this is still an ongoing study, and still we have to further characterize this formulation in an animal model, like a mice model, wherein we induce, age-related macular degeneration, and we see whether the drug is, whether the formulation is able to treat it or not. So that's a pending study. To summarize what we have done so far is we created a nanomicellar formulation, mixed nanomicellar formulation, and obviously this formulation is going to have much more advantages when compared to the injections or implants. So injections or implants, they are considered to be invasive in nature, okay, and they have very less patient compliance. Whereas mixed nanomicellar eye drop formulations can be self-administered by the eye, and they work as simple eye drops. So these are much more patient compliant or much more patient friendly. So we have to further characterize this formulation in a suitable animal model. So that is the study that is pending. And finally, I have the acknowledgement slide. So I have, the students who worked on this project. So these are my past graduate students who worked on this project mainly. Ms. Suhi Patil, she was the one who designed this formulation. Then I need to, thank Advacnes University for helping me with the funds to continue this study. And then the organizers of BioMe', especially Dr. Mamidala, as well as Dr. Pavani for, arranging such a wonderful seminar, okay, such a wonderful event. And these are the references. And now the floor is open for questions. Questions from the students? I think this is of your level, right? Did you guys understand at least a part of it? Yes? Then ask me a question. So for after cataract operation, generally we use anti-inflammatory agents. So we have to take care of the inflammation process. So that is, again, it's an anterior segment disease. So for that, we don't have any issues. So we have we have to take the medication regularly, anti-inflammatory agents, in order to prevent the cloudiness. Other questions? All right then, thank you for patiently listening to my seminar. And I think next we are going to have a session on meditation, which is very important, okay? After having so much of, brain exercise, continuously listening to the talk, you have to calm down. You have to calm down means you have to calm down the brain, okay, through meditation. So I think, all right then, thank you for patiently listening to my seminar. All right, thank you. Next we are going to have a session on meditation.
Thank you, sir. Sir, I request you to stay on the base. Okay. I would now request Jagadishwar Reddy Garu, OST Tribal Welfare, to felicitate our speaker, Professor Hanuman Sagar.
So this is an announcement to all the participants who have registered and sent you abstracts for the oral presentation. We request you to move on to the room towards the left side of the dais. The participants who have registered and sent your abstracts for the oral presentations, please move to the room towards the left side of the dais. The selected members. There are 9 participants who are selected for the oral presentations. Next, we have meditation session. In the same auditorium, we request you to please cooperate. Thank you.
I request all social welfare and tribal welfare participants to stay back, and the interested participants to join the meditation.
Okay, does anyone want to stay in the room at all? Yeah? Well, it's not going to be that bad, you know. We're just trying to get you to relax. Okay, ones who absolutely cannot sit or tired, have some emergency business to attend to, you can quickly and quietly walk out of the auditorium. Okay, we just have 1 more minute for people to exit the auditorium. After that, we're going to close the doors. I'm just joking. Please don't run. We don't want a stampede here. It's all right. Okay, since there's no one